will be leaving at some point, so I'll be here kind of, yeah, if you need anything, just shout. I'll have my camera turned off and everything, but I'll be monitoring and doing stuff in the background, but just let me know.
Okay, thanks, Ben.
Thank you.
No worries. He will be leaving at some point, so I'll be here kind of, yeah, if you need anything, just shout. Ooh, spooky. Yeah.
Oh yeah, so yeah, Ben, you'll be in charge of spotlights and stuff. Actually, maybe Pedro can be as well as the co-host. Oh yeah, so yeah, Ben, you'll be in charge of spotlights. Always quite disconcerting to hear your voice, right?
Just give you some encouragement so it kind of keeps coming through the speaker and you feel good about yourself. You're doing a great job, Joe.
Thanks, Ben. You too. You're doing a great job. You're doing really well. Nearly there. Just need to do one thing. Okay. We're nearly there. Yeah. Cool, we are on all platforms. Cool. Right, we should be live on all platforms again. I will hand over to Pedro, who is going to be our host for the next session. And we'll hand on into the night to a series of hosts, mostly based in North America. So in that sense, I'm going to hop off the call. You're going to message me if it all falls to pieces, but it should be fine. And I can't wait to hear about magnetotactic microorganisms. So, Pedro, over to you.
Thanks, Joe. So as Joe was saying, we're about to start the session about the magnetostatic organisms. It's my pleasure to be a host today. My name is Pedro. And well, enough talking. I will pass the room to Igor. He will introduce himself and talk a little bit about the work he's been developing for a while in the lab. It's up to you, Igor. Go for it.
Hello guys, my name is Igor. First, I would like to thank you for the invitation to be here today and to talk about a little of our research here in Brazil. So I have to share my screen. Share it out. Are you guys seeing?
Looks
good.
The whole screen. Okay. So today we're going to talk a little about magnetostatic microorganisms, and especially today we're going to talk about magnetostatic bacteria diversity and their spatial distribution in the Araguaia River floodplain, which is a site in the Brazilian Cerrado and Amazon, which is a very cool place to study. So my name is Igor, as I said before, and my supervisor, PI, is Professora Fernanda Abreu, and we work at the Universidade of Federal Rio de Janeiro. So the first thing that I'd like to say to you guys and ask is, what is magnetotaxis? And I like to show this with a video. So here we're gonna see how this behavior works. And this is an edge drop. Here we have a lot of magnetostatic bacteria. This is set in a Dick microscopy. Here is the edge drop, and then we switch the magnet that is placed on the stage of the microscope, and then the magnetostatic bacteria swim in direction of the magnet, or either it swims running from it. These bacteria, they tend to swim in anti-parallel to the magnet. And every time we switch the magnet, we can see that they drive or they concentrate. But this phenomenon it occurs in bacteria, but not only in them. They also occur in magnetostatic eukaryotes, which was Pedro's previous work on his thesis. And this is the same thing on the edge drop. Here are the eukaryotes, and the same thing as we switch the magnet, the eukaryotes swim away from the edge drop, and then they concentrate again. So this have this response to the magnetic field that in this case is induced by placing a magnet on the stage of the microscope. And how this happens, these magnetostatic microorganisms have two unique features: that they can passively align or orientate themselves to the Earth geomagnetic field, and through their active flagellar propulsion, they swim and respond to this passive orientation. So the main characteristics about this group of bacteria is that they're gram-negative. They can be unicellular microorganisms. Here we can see the flagella, or they can be multicellular, as they called MMPs, multicellular magnetostatic prokaryotes. And they are capable of synthesizing, or the more precise term, biomineralize a ferromagnetic nanocrystal, which is this guy here called the magnetosome. and it's surrounded by a biological membrane, as we can see here on the stem image. And this process is controlled by a set of genes called the magnetosome gene clusters, MGCs, and this trait caused by the biomineralization of the magnetosomes and the active flagellar propulsion corresponds to the magnetotatic behavior. And we can classify these organisms based on a lot of things. We can catalog these magnetosomes. They can be cataloged into their chemical composition, whether it's magnetite or graphite. They can be characterized regarding their chain organization or the crystal morphology. As we can see here, they have very unique shapes, and these shapes are species specific. We see that a cubohedral shape, octohedrals, prismatic, and anisotropic, which they resemble a bullet. And this magnetosome in particular can be used in a lot of biotechnological applications, such as cell separation, hyperthermia, drug delivery, food analysis, DNA and antigen analysis, can be used to image contrast in MRIs and etc., immobilization of enzymes. So this is very versatile and can be used to a lot of things. And the advantage is that it can be magnetically recovered. And talking about our work here, about the geographical localization. So this is the map of Brazil. And our area of study is here on the Araguaia River, right in the middle of our country. And we are located here in Rio de Janeiro at UFRJ. And why did we go there to study what we study? The Araguaia River, so it's oligotrophic, meaning that has low nutrients and has an acidic pH. And the Bananal Island, which is located somewhere here, is the world's largest fluvial island. And these are some photographs of the place that we went, some aerial images. So that's a very beautiful place if you want to go visit or spend your vacation too. And why study MTB in this place? So this is a giant field of work. We can generate large amounts of data from a field that we don't know, or we don't know these conditions. We can understand MTB spatial distribution and how the magnetosomes that they biomineralize are also distributed in this place. Species that are going to be prospected can have some biotechnological applications, and they belong to our national territory. And we can enrich our biodiversity from the Cerrado and the Brazilian Amazon. And the team that is indirectly involved in this particular research is our PI, Professor Fernanda Jefferson. He's already a PhD and he works with microscopy now. Juliana, that is Master in Science. And Professor Alex Price, that is our collaborator in the Linköping University at Sweden. And just to talk a little fast about, we sampled this bacteria, this sediment with water, and we basically divided this area. We cover approximately 1,000 kilometers of river extension, and from Pará, which is a very, it could be, I don't know, it's a very, very large distance. And we did this to understand how the abiotic factors are dispersed and what they influence, but that's not the main point that I want to show you guys here. What I want to show you guys is that what we found in this place. So we divided in the bacteria from the main channel and the bacteria that we found in its lakes and tributaries. And for our surprise, what we found. Let me stop the laser. I need to click on the video. So the bacteria from the main channel, they're majorly coccoid, which means they are spherical. And the same thing here, we place the magnet, and this is the edge drop. Then when the north pole is directed to the edge drop, then they swim towards it. And when the reverse the magnet, then the bacteria swim away from it. And these ones are majorly found in the river main channel. And for our surprise, there is a completely different population of MTB that are in the lakes and tributaries. And what we see is that they have other shapes and morphologies. Some of them are ovoid, some of them are rods, and some of them are vibrioid and spirilla. And the same thing that I've talked to you guys, when we reverse the magnet, they're going to perform a turn, what we call a U-turn. And then it's all fun when you guys see this. Once we revert again, they will swim away from it. And then when we reverse again, they go back to the edge drop, concentrating again. So, as I said before, oh, I'm sorry. So I said before, we sampled in 14 points, and the first 4 were in the river main channel, and the morphology was coccoid bacteria with some vibrios, and the other sites on lakes and tributaries were majorly rod-shaped bacteria, ovoids, spirilla, and vibrioided cells when you see this in this set of images. And when we understand this occurrence and spatial distribution, we understand that the magnetotatic cocci, which are this one, they are distributed when we do this diagram chord plot, we understand that they are majorly located in the river main channel, and the other morphologies are located in, here in green, in the lakes and tributaries, the rods, the spirilla, and the vibrioid. And we can see when we perform a sort of set of statistics analysis that some abiotic factors are important for driving this diversity. And there are some clusters when they are very proximal to each other about their morphology composition. And just to illustrate what these magnetotatic cocci are, we have they are very diverse. They have a high diversity. They are prevailing in the main channel. They majorly synthesize octahedral magnetosomes, which are in this area, as you can see. And here in this inset is the greater magnification. In some of them, you can see the flagella, when it is this arrow, this purple arrowhead. We have a great diversity also of magnetotatic vibrios. The same thing here. So in some of them, you can see the flagella on the stem images, large diversity of magnetosomes. And on lakes and tributaries, we also have a large diversity of these microorganisms. These are the magnetosome chain within the cell, the large modification. Some of them can be analyzed, hundreds of magnetosomes. These are rod-shaped bacteria. These are the spirilla. And some of them, you can see the flagella, and these are the ovoid bacteria. And these magnetosomes, they don't have any particular kind of organization. These are concentrated in this pole of the cell. And we can perform a lot of statistical analysis and catalog these magnetosomes regarding their morphology. But the fun thing is that we can see and predict their size doing linear regression, and some of them fit quite well. But the anisotropic magnetosomes, which are these 3 sets, they don't quite fit here. They are more like a sigmoidal distribution, which is fun to understand. And the same thing happens when we use the abiotic factors to understand how these magnetosomes are spatially distributed in this space, and they also form some kind of narrow clusters depending on their composition. And when we talk about the magnetosome, one of these particular traits is that it is a magnetic monodomain. And what that means, it means that this mineral, this ferromagnetic mineral, is permanently magnetized. So what happens is when we plot the length of the magnetosomes per shape factor, which means they are more elongated, or their dimensions of width and length are equal, we see that they are distributed in a dot plot, and they are clearly positioned in a monodomain from biogenic magnetite. And fun things when we see that is that are some giant crystals that are hard to find in normal samples, and they are present in the sample of the Araguaia River, which are giant octahedral magnetosomes that are ranging from almost 200 nanometers. And here is their distribution, regarding the dot plot, which fits a log normal distribution. And the things that we found so far in this particular site of study is that we didn't find any magnetotatic multicellular prokaryotes which are highly related to high salinity place. We didn't find any eukaryotes or magnetotatic consortia, but we could understand their spatial distribution in the sites of the river main channel and its lakes and tributaries. We saw that the magnetotatic cocci are the most abundant morphotype presenting in this particular environment. And as we're talking about the freshwater river, we can exclude the salinity factor, which many authors extensively describe how this affected MTB diversity. And then we can understand how some other factors like the potential reduction can influence the pH, the dissolved oxygen, and even the water transparency can influence and shape the MTB population, the populational dynamics. And the Banana Islands, which I talked to you guys earlier, that is the world's largest fluvial island. It's a natural MTB hotspot, which was new discovery. And future perspectives talking about this work is that we're going to understand, as we have this large area of study, how the microbiome of MTB is structured using a gene mark as the 16S RNA gene, and search for the biomineralization genes that are involved in this process. And we are already conducting a species prospection to study future biotechnological applications from bacteria that are from Brazil, and to confirm the chemical composition of the magnetosomes by microanalysis. And here are some photos of the field trip. And here are some of the people that went with us. This was a multidisciplinary travel with lots of areas of study, including fishing and sediment core sampling. Here we are in the river. Here are the people and the guy that drove us in the boat. Here we are drinking some beer in a small town in the middle of the state of Mato Grosso. And I would like to say special thanks to our lab founder, Professor Ulisses Lins, which is no longer with us. He unfortunately passed away 4 years ago. And our friends here is Pedro on his PhD thesis, after he defended his PhD thesis. Our friends here from the lab. Here are our social media. And I would like to thank our financial support and the infrastructure used in this project. And that's it, guys. I hope you all enjoyed it.
Thank you, Igor. It's always amazing for me to see what you guys are up to in the lab. And yeah, we're a little short on time, but again, I'd like to thank you. I really appreciate what you have been doing, and can't wait to see what's come up next from this project and from all the other projects you're involved in.
I'm pleased to be here today, and I would really like to thank the invitation to be here and talk a little bit about our work.
See you, Igor. Let me allow our next speaker to be here with us. We'll have Lucia guiding. Lucia will talk a little bit more about the potential applications of magnetosomes and magnetostatic bacteria soon. If you want to start introducing yourself, Lucia, be my guest. Thank you so much for accepting my invitation to be part of this session today. Oh, you're muted, sorry.
Sorry, I think you can hear me now, right?
Yes, that's right.
Yeah, so thank you for inviting me here. It's a pleasure for me to talk about magnetotactic bacteria also. So I'm a PhD student in the University of the Basque Country, and I mainly work with the biomedical applications of magnetotactic bacteria. I will try to share my screen for you to see. Okay. And I will start my presentation. Okay, can you see my screen?
Yes, it's working.
Okay, so Igor already introduced you magnetotactic bacteria, but I will talk you about our work, which is in the biomedical applications of them. Bacteria in general have a long history in cancer therapy, because already in the 1890, Coley discovered that some cancer patients recover after suffering bacterial infections that destroys the tumor. So over the last century, many genera of bacteria are being investigated to treat cancer because they have so many advantages that make them ideal for this purpose. For example, they have the ability to infect and kill specifically cancer cells because, for example, they can compete with them for the nutrients, and some of them secrete some exotoxins that activates the immune system so that the body fights against the tumors. Moreover, they are motile. So most of them, they have their flagella so that they can swim and go deep into the tumoral tissues. And also, which is really interesting, is that mostly we use all the anaerobic strains or microaerophilic strains of bacteria are being investigated for this, because normally the tumors have like a really strange vasculature, so that there is like a low concentration of oxygen, so that these bacteria will be attracted to this, specifically to these tumoral areas, which is great. But mostly the bacteria have a major drawback, which is that we couldn't detect them inside the body, and it's impossible to guide them to the desired area. But we have the magnetotactic bacteria that have the magnetic chain, which makes them special, mostly because we could detect them by using, for example, magnetic resonance imaging once they are in the body. And we could also, and more importantly, guide them to the tumoral area really easily, because we could guide them by using external magnetic fields. So in our group, we propose the use of magnetotactic bacteria for cancer treatment, and I will explain you today how. Magnetosomes by themselves have also been proposed for biomedical applications because they have many advantages also compared with chemically synthesized magnetic nanoparticles. For example, the level of purity and crystallinity of magnetosomes is really high, and they are really uniform in terms of size and morphology. They have interesting magnetic properties, and I think the major advantage compared with chemically synthesized magnetic nanoparticles is that they have the biological coating, which is composed of lipids and proteins, which makes them more compatible with cells and makes them easy to functionalize with, for example, some anti-cancer molecules or something like that. And compared to chemically produced nanoparticles, they are environmentally friendly, which is great also. But they have also some drawbacks. For example, the production of magnetosomes is quite low, and they are not able to self-propel. So, which guides me again to magnetotactic bacteria. So we think in our group, and I hope that you think it too after my talk, that magnetotactic bacteria are ideal candidates for cancer therapy nanorobots because they have flagella so that they can swim inside the body towards the tumor. They have also chemical and oxygen receptors so that they could swim towards the hypoxic regions of the of the tumor. They also have the magnetic chain, most importantly, which makes them magnetotactic, so that we could guide them, as I explained you before, by using magnetic fields. And they could also be functionalized with, for example, drugs for anti-cancer treatment, so that they will only they will transport the drugs to the tumoral area only, and do like more specific treatment. So there are several biomedical applications that have been proposed for this bacteria, but here I put the main three, I think, which are drug delivery, magnetic hyperthermia, and MRI imaging. In our group, we mainly work with magnetic hyperthermia, and is what I'm going to explain now. But after my talk, after I finish up with magnetic hyperthermia, I will show you some cool videos and images about drug delivery too. So magnetic hyperthermia, for those of you that are not familiar with this technique, is a therapy for cancer treatment that is based on the injection of magnetic nanoparticles or magnetotactic bacteria, in this case, in the tumoral area, and the application of an alternating magnetic field. So when the field is applied, the magnetic nanoparticles, they release heat so that they increase the temperature, specifically in the tumor, up to 42 degrees Celsius or so, which is enough to debilitate or kill cancer cells, and it doesn't affect healthy tissues. So it's great because it's more specific than chemotherapy, and for example. So it's been used for glioblastoma, I think, since 2011, but with chemically synthesized nanoparticles. But in this case, we propose magnetotactic bacteria for that. Okay, so we did a study where I'm not going to go into detail with this because I know most of you are microbiologists, me too, but we measured the heating efficiency of bacteria compared with magnetosomes. And surprisingly, we saw that they are more efficient in heating, so increasing the temperature of the tumors, the bacteria, than the magnetosomes, which is great because this is another advantage of them. So we did an in vitro experiment with using magnetotactic bacteria and lung carcinoma cells. So we incubated them together. For this, we used MSR1, which is Magnetospirillum griffiths valdense. We incubated them with the cells for 24 hours, and then we checked the cytotoxicity of the bacteria in the cells. Then we applied the magnetic field in clinical conditions, and we also checked the viability of the cells to see if the treatment was or not effective. Okay, first of all, some nice pictures, because we wanted to see if bacteria and these cells interact with each other, because if not, that would be nonsense. So here we see the scanning electron micrographs, where we see the magnetospirillum in orange and the cells in gray, so that they interact kind of in the surface. But with fluorescence microscope in the left, we also saw that they get into the cells, because we can see the bacteria in red with rhodamine, and in blue, the nuclei of the cells. But then we confirmed this by using transmission electron microscopy, where we can see the bacteria inside the cells. And here, for example, we can see in the right image, the nuclei of the cells and the bacteria inside. So this is great. This is the first step. And then we wanted to check the cytotoxicity of the bacteria in the cells. For this, as I told you, we incubated them together for 24 hours. And we checked if cells died after this incubation period by using flow cytometry and propidium iodide that only stains the dead cells. And we saw that both control cells and bacteria-loaded cells had like a similar percentage of death. So we concluded that in principle, magnetospirillum are not toxic for the cell, which is great because it leads us to the next step, which is applying the magnetic field and checking again the death efficiency, or let's say the hyperthermia treatment efficiency, by using propidium iodide again. And we saw that 2 hours after the magnetic field application and 24 hours after the hyperthermia treatment, the percentage of dead cells increased like significantly, which is great because that means that the hyperthermia with magnetotactic bacteria actually works, at least in vitro. Then we will have to try it in vivo, but that's a step further. Okay, so we also saw like there is an effect of with hyperthermia for the growth rate of the cells. So when they were incubated with bacteria, and the control cells had a similar growth rate, but when we apply the magnetic field with the bacteria, this was really slower, especially after 48 hours after the treatment. So again, this is a good news, but then we will have to follow our experiments to see if we can someday like have this treatment really in clinical trials. Okay, so I told you that I will show you some cool videos, and I promised it. So here, as I told you, bacteria could also be used for drug delivery because they can swim. In here, you can see magnetospirillum also in my microchannel, where we put a magnetic field horizontally, so you can see how they swim in the direction of the magnetic field, which is cool because that means that we could guide them inside the body. And this is an experiment done by another group of Felful and their colleagues, where we see another species of magnetotactic bacteria, which is Magnetococcus marinus. What they did here was to attach some liposomes that had anti-cancer drugs inside to the surface of the bacteria. So it's more like another step for drug delivery. So once the liposomes are attached in the bacteria, we could guide them to the tumoral area, and if everything goes well, they will only release their content once in the tumor. So this is like a beginning of I think a big thing. So hopefully in some years we will see all together in a cancer therapy treatment. I don't know. So I would like to thank all my colleagues and our collaborators for all the work. And yeah, I want to thank you again, Pedro, for letting me speak today. And I hope that I have convinced you about the applications of magnetotactic bacteria. And if you have any questions, please ask me. Thank you.
That was an amazing talk, Lucia. Lucia,
Thank you.
a lot of nice stuff that you have been doing for some time. Yeah, we have some time for some questions. I can't see any here now, but I have a lot of questions, so I will take some. So so you're saying that there's a lot of promising things that you guys have found out so far. What is the next step? I mean, what do you think is putting us a little back to go to clinical trials or even to some more, I don't know, other types of experiments that could lead us to a direction where we can actually use this for therapy?
So there are many things that we can do. For example, I think that there are some groups that are already trying this with mice in terms of magnetic hyperthermia, but I think that also we will have to see if the magnetospirillum, for example, could be like functionalized with, I don't know, anti-cancer drugs and so on. And I think there's a lot of work to do still on the navigations of this bacteria inside the body. Like I showed you a video in a microchannel with water, which is easy, but then inside the body and the blood is quite different. So I don't know. I think it would be the next step. Yeah.
Yeah, definitely. It will be interesting to see their behavior in more complex environments, right? How they will behave and all this. Yeah,
yeah. I think a lot of groups are studying that. We are doing it at the moment, but yeah, there's a lot of investigation there.
Nice. Can't wait to see what's up next for you guys. I thank you so much for a talk. That was amazing to have you here in this session.
Thank you.
I will put the next speaker up. Our last speaker of the session will be Dennis, that will be talking a little bit more about how it's the scenario of the evolution and the diversity of the magnetotatic organisms so far. Then it's coming up. Here there is.
Hi.
Hello, Dennis. Well, I will let you the room, let you introduce yourself and talk a little bit about the evolution of magnetotatic bacteria.
Yes. My name is Denis Gruzdev. I'm co-founder of genomic analysis startup Cyber. And today I will talk about diversity and evolution of magnetotactic bacteria. And I will start from the history of in the middle 60s, Salvatore Bellini discovered unusual movement of bacteria. He observed large number of bacteria swimming in a single direction, and they have magnetic behavior. And he suggested that this behavior is due to the presence of a magnetic compass inside the bacteria. And 10 years later, Richard Blakemore independently rediscovered magnetotactic bacteria, or MTB, and was the first who demonstrated Bellini's magnetic compass within cells. He observed tiny magnets organized in chains, and these particles, as you know, called magnetosomes. MTB is a common name for a large bacterial group that have ability to form magnetosomes and have magnetotactic behavior. MTB are diverse in their cell morphology. Different spirilla, vibrios, cocci, multicellular bacteria were shown. Shapes and composition of magnetosomes, magnetosome crystals, are also different. Magnetosome crystal can consist of magnetite or greigite, and usually magnetosomes are aligned in single chain. However, MTB with multiple chains and clustered magnetosomes were observed. Magnetosome formation is controlled by specific magnetosome genes. In the genomes of all MTB, magnetosome genes are present as operons or clusters, and all MTB have a common 9 essential genes located in MAMA B operon, and they're necessary for sorry.
Yeah, if you have some slides, we cannot see it at the moment.
Oh my God, sorry. I can start.
We can start over if you
Okay.
want to. You have time.
Briefly, I can repeat that magnetotactic bacteria is bacteria that can produce magnetosomes. They have usually they have a different cell shape, including spirilla, rods, vibrios, and etc. And usually magnetosomes also different in their composition, including they can consist from magnetite or greigite and have the different shapes. And the magnetosome biomineralization process is controlled genetically, and 9 essential genes present in all genomes of MTB. And in all genomes of MTB, sorry. And as was said by Lucia, magnetosomes have a huge biotechnological potential and could be used in several biomedical applications. But the main problem for the wider use of magnetosomes is their limited production. The fact is that cultured MTB produce only 15-20 milligrams of magnetosomes per liter of culture. And what are the ways out? The first is the optimization of MTB growth condition, but over 30 years of attempts to optimize, the breakthrough did not happen. The second is genetic engineering and the transfer of magnetosome genes into an easily cultivated bacteria. And the third is the search and the xylation pure culture of MTB, producing a greater number of magnetosomes. For example, Magnetobacterium bavaricum produce up to 1,000 magnetosomes per cell. As for genetic engineering, there is some progress. So in 2014, German group transferred the genes for magnetosome synthesis into Rhodospirillum rubrum, and it began to make magnetosomes. But unfortunately, these magnetosomes had too much distribution in size and were synthesized only in the light. This This indicates that we still poorly understand the process of magnetosome synthesis, and to understand it, we need more information about the genomes, diversity, and evolution of MTB. And well, now directly about the study of diversity MTB, I conventionally divided the last 50 years into 3 parts. The first of them ends at the beginning of the 10th of the 21st century. During this period, the study of MTB diversity is limited to the isolation and description of pure cultures of MTB and the analysis of clone libraries of the 16S RNA gene. Already 25 years ago, we knew that MTB are found among alpha and delta proteobacteria and the not yet named phylum Nitrospirota. All known MTB of the alpha proteobacteria class belong to 2 orders, Rhodospirillales and Magnetococcalis. This is the most studied group of MTB, and bacteria of the genus Magnetospirillum are model organisms for studying magnetoreception and synthesis of magnetosomes. And MTB from the order Rhodospirillales are spirilla ovibrous that synthesize one chain of cuboctahedral or prismatic magnetite magnetosomes. MTB from order Magnetococcalis often include coccoid bacteria that produce magnetite magnetosomes, which can organize in one or more chains or not at all. This order is very interesting in the light of the study of evolution. First, this order will soon be reclassified as a new class within proteobacteria, and absolutely all bacteria in this group can produce magnetosomes, which is the exception. MTB from delta proteobacteria or Disulfobacterota are the most diverse in cell shape of magnetosome types. There are bacteria that can produce both type magnetosomes, magnetosome crystals, magnetite and greigite, and even both types of magnetosome in one cell, like Disulfanthus magnetovalimortis. This group also includes amazing multicellular prokaryotes. And the second period is mediated by the beginning of work on metagenomic and single cell studies. During this period, our understanding of the MTB of the phylums Nitrospirota and Proteobacteria was significantly expanded. MTB were identified in the phylum Omnitrophota, previously known as OP3, and in the class Gamma Proteobacteria. Known MTB from Gamma Proteobacteria are mesophilic, microaerophilic rods, and they bimineralize either cuboctahedral or elongated prismatic crystals of magnetite in their magnetosomes, like the alpha proteobacterial MTB. A large morphological diversity of MTB in phylum Nitrospirota was shown. Unfortunately, not a single MTB of this phylum is related in axenic culture, but we know of huge rods, small vibrous, and cocci that can synthesize from 10 to 1,000 bullet-shaped magnetosomes. It's also worth mentioning the discovery of MTB in the phylum Omnitrophota, but it's all complicated. And during this period, enough information was also accumulated to formulate the evolutionary scheme, which was supported by most scientists. It was concluded that genes for the synthesis for the magnetosome synthesis appeared once in the common ancestor of proteobacteria, Omnitrophota and Nitrospirota, and these genes disappeared in some classes of proteobacteria. Also, during this period, the accumulation of metagenomic data in open databases takes place. This led to the identification of magnetosome genes in contigs of bacterial genomes from the phytoplankton Mycetota and Latestobacterota. And phylogenetic analysis showed that these bacteria most likely synthesize greigite-type magnetosomes. But unfortunately, we still have no idea what these bacteria look like or what shape of their magnetosomes. Well, and the third period came quite recently and is associated with the introduction of metagenomic approach and culture-independent analysis into microbiological practice. So in 2018, the article was published describing 28 reconstructed from metagenomes MTB genomes, which exceeded the total number of MTB genomes sequenced all previous years. Also, MTB were first discovered in Zeta and Lambda protobacteria classes of protobacteria. But unfortunately, we also still do not know anything about their morphology. But it was predicted that they should produce magnetite magnetosomes. And at the same time, the new hypotheses for the evolution of magnetotaxis were formulated, and it is a very complex process. As we think the evolution included vertical inheritance from one common ancestor with multiple duplications, losses, and horizontal gene transfers. And 2 years later, the article was published on the reconstruction of MTB genomes from open databases, and several dozen MTB genomes were reconstructed, including those belong to bacteria from phyla Nitrospinota, Gidrogenidentota, and the Lucy microbiota. And performed phylogenetic analysis revealed multiple discrepancies when comparing the topologies of phylogenomic and the magnetosome trees, which indicate a great influence of horizontal gene transfer on the MTB evolution process. And a few months later, the analysis of 168 metagenome assembled MTB genomes were presented, and 4 previously unknown MTB containing phyla were identified, including Riflebacterota, Fibrobacterota, Bdella vibrianota, and Uba 10199. And again, we have only genomes. Also, in this article was an interesting but controversial theory that the last bacterial common ancestor could synthesize magnetosomes. And it is very interesting theory, but we need more data and here a lot of work. And well, I want to sum up with a picture with the current situation. It should be said that right now is the golden age of studying the MTB diversity, because most MTB containing phyla were discovered in the last 3 years. But as you can see, we have a huge bias toward genomics. That is all we have in genomes. It should be said that the diversity MTB is much greater that is known today, and so the amount of genomic data will increase for objective reason. But I really hope that you and I will be able not only to get hung up on genomes, but also to describe the morphology of cells, the magnetosomes from new phylogenetic groups, which ultimately will help to fully understand the process of evolution and process of magnetosome synthesis. And thank you for your attention.
Thank you, Dennis, for the great talk. I mean, I'm a little biased here, but I think this is fascinating and still so much stuff for us to find out. Not only what are the other shapes of this magnetotatic organisms, but also the big question, where they came from and what they are doing here exactly, how they're important for the biogeochemical cycles, all this stuff. My question to you, that I guess it's a tricky question. So we saw the last work from your group that Maria is developing, and that we have a lot of really ancient branches with magnetotatic bacteria. So where do you think this will go? I mean, are we getting closer to find that they are even more ancient than the Delta Proteobacteria class that we find out before?
Thank you for your question. It is really important to understand that if we see magnetotoxic bacteria in phylogenetic group that near to the root of bacterial tree, universal bacterial tree, this is not meaning that this phenotype and genotype was received through vertical evolution, through vertical inheritance, because now we know that magnetosome genes can be transferred through the phyla, interphyla horizontal gene transfer. It is very interesting because we do not really know how they looks like. I am not sure that they produce magnetosomes. Maybe it can be some different process. And I think that not all magnetotoxic bacteria that can produce magnetosome, use them for the magnetotaxis, for example. Maybe they can have another function for these organelles.
Yeah, I think this is really interesting what you were saying, because I completely agree with you. I don't think the magnetosomes arrive as they are right now today. So maybe a more ancient structure was developed before with some of the genes that are doing something else. And
yes,
today we have the modern magnetosomes. But yeah, this is really interesting. I have no clue what we're going to find out next, but I'm looking forward to it.
We have a few surprises for you in the next year. Yes.
Looking forward to see what you guys will bring. Thanks again, Dennis. I will wrap up this session a little bit. Do we have some of the hosts around here?
Hello.
Hello, Ben.
Hi. So yeah, you finished? No more questions? We're ready to wrap up the magnetosome session now.
Yeah,
well,
I think we're good.
Great. Well, thank you, Pedro, and thank you, Dennis. Thank you to all the speakers of this session. It was very, very interesting indeed. Yeah, I always get fascinated by these magnetotoxic bacteria. They're so cool. I've never worked on them myself, but yeah, it's nice that we get an opportunity to discuss them in this forum. And so thank you for chairing it, Pedro. Just looking at the time, yeah, we're getting close to the next session now, which is binning singletons. So while we prepare for that, I'll say, yeah, thank you, Pedro. It's been a pleasure. And I'll just remind everyone of what's going on this evening. Well, it's evening here in Stockholm where I am, but everywhere else, we're kind of going across the time zones now. And it's getting exciting because although it's still the 16th of September here in Stockholm, it's not quite International Microorganism Day for us. International Microorganism Day is the 17th. More and more countries are getting into that kind of crossing the boundary and being able to celebrate for real. So I know Australia and a lot of Asia now coming into International Microorganism Day. So that's exciting. We'll get there. We'll get there soon. So as binning singletons come in to the room, let's just see what we've got coming up. I mean, for the next hour, we have Binning Singletons. We're going to be learning a lot about them and what they do. Then we're going to go into vaccines and vaccine hesitancy, and then microbial arts and crafts workshop. So that's the next 3 hours for us on the live stream. Please, please, please feel free to engage with us. Feel free to ask questions. We're streaming live now. Wherever you're watching us, we're streaming live and you can ask us questions there. You can go to our website, or you can just ask us in social media. We'll be monitoring everything and you will get your questions answered here. And so without further ado, I would like to invite Joe James to step forward. Hi there, Joe. How are you?
I am doing well.
Fantastic, fantastic. Well, we're now pretty much in your session, in the Binning Singleton session. So if it's okay with you, I will hand the floor to you, and I'll be here kind of hiding in the background if you have any technical issues or questions. But otherwise, yeah, over to you. The floor is yours.
All right. So hi, my name is Joe James. I am the founder of Binning Singletons, which is a mentoring organization, and we've been involved with a couple of microbiology associated organizations and meetings for the last few years. So today I'm really excited because everyone who's speaking today is someone who has participated in our program. In fact, I think all of them are from our World Microbe Forum program that we had back in June. So I'm going to do a very quick talk first. We actually had somebody drop out because they weren't feeling well, so I'm going to do a little bit of a quick presentation, and then we'll get to the lightning talks, so to speak. Mine hopefully will be as fast, if not faster, than some of theirs. So what I'm going to talk about first is networking and mentoring, and here we go. Hopefully, let me pull this up. Give me 1 second. Sorry about that. Full screen. All right. Does everybody see that? Y'all good?
Yeah,
okay.
Perfect.
Looks good.
So let's go backwards. All right. So the title is Growing Peer Cultures, of course, a pun because I'm a dad and I like dad jokes. And so we're going to talk a little bit about the importance of near-peer mentoring. I think this is important, really, even for the non-micro, but maybe especially the non-micro people. Even if you're in a job anywhere, really, or in school, a lot of times mentorship is really focused on that first thing on there. We have the types of mentorship we have traditional. And there's a little bit less on coaches and sponsors. We all kind of know who these people are, but we don't always think of them in a mentoring kind of way. Then we'll talk about peer mentors, and then we'll hopefully talk a little bit about a perspective shift. So, like I said, mentors can be classified in these 3 ways. You got your traditional mentors, your coaches, and your sponsors. And these are not original thoughts to me. Exactly, I took a lot of this. You'll see there's an article on ASM that I found very helpful. So that's kind of what I use some of my inspiration from. So traditional mentoring, this is often a top-down thing. You have like a senior person, a boss, a lab leader, a PI, and then you have that person's usually an individual that's associated with the organization. And so this is when we think of like the long-term formal guidance. So this person kind of like is your boss, your mentor, your teacher, the one who kind of gets you through everything, but you also like kind of look to them for everything in a way. And I think that's the way a lot of people look at traditional mentoring. And I think there's obviously some things that can be gained from having a person that is your kind of dedicated person, but I think there's more that we're missing if that's all we do. So one thing that's nice is that they do help you recognize and achieve these defined milestones. And that success, though, is context specific. We need to remember that traditional mentors, it's really about socializing the individual to the organizational norms. A good professor in academia, quote unquote, good professor, does a really good job of churning out a lot of students. In a lot of cases, people think of that are people who would go into academia. There are obviously a lot of people who do more than that, but I think a lot of people kind of get hung up on that idea that they're preparing you for everything, and really there is a specific context that we need to look at. Moving on to coaches. Coaches are people who spend less time with these individuals, but they make a significant impact. We're all familiar, if we've all done sports, you have a coach, maybe you have practice twice a week, maybe you have a game once a week. You don't interact with those people, say, as often. Say if you're in 5th grade and you have a teacher and you see that teacher all day, that you see, your soccer coach twice a week, your soccer coach is really only focused on one thing, whereas your teacher is focused on a lot of things. So that's kind of the general idea of coaching, and I think most people can understand that. And so these coaches focus on just development of particular skills, things that don't come up as much. So if you're looking for a job, if you're writing a manuscript, designing an experiment, often learning a new lab technique is somewhere where you would find a coach. Our program, Bidding Singletons, I think of as an example of coaching. So we have mentors who are really interested and excited about helping you. And there is a time frame that's involved, but it is not an infinite thing. It's a short duration because we help people through a meeting. And we have specific goals. We're going to help that person have success at the meeting through networking, understanding the logistics of how to navigate meetings, and then building a sense of community. So we have some definite goals, and then we have a time frame that's defined, and everybody kind of knows what that is going into it. Sponsors are your senior scientists, or if you're not in science, these are just people who have garnered substantial political capital. This is kind of the who you know part of things where mentorship is involved. So they're going to be committed to the career development of this individual and help to influence and advocate and advance mentoring. So this could be somebody who can get you on a committee. This is somebody who can invite you to a talk, to give a talk somewhere, invite you to review something for an article, invite you to a planning committee for a meeting. These are people who can help you advance your career, and them putting your name forward can really go a long way. So I want to dig into the effectiveness of the traditional mentoring for a little bit. There was a study came out in 2019, and it showed that the PI, their involvement in the mentoring laboratory activities did not significantly predict the students' skill development trajectories, which is a little bit mind-blowing if you think about how we usually think things go, which is that if you have a very involved PI, they're going to really help you with your skill development. And what they found instead was that postdocs and senior postdoc students, when they actively participate in these lab discussions, that the junior students are 4 times as likely to have positive skill development trajectories. Yeah. Oops. Okay, so these near peer mentors, they make a really, really big difference. Near peer mentees, these people who had mentors that were, so if you're a PhD student and you have a PhD candidate, or maybe a postdoc in your lab as well, those people would be your near peers, and they can really help you out with some professional and personal development. They can help you navigate things, so it reduces some of your stress and then helps transition. So there's an article there that came out in 2018. So let's do a little bit of a perspective shift. And this is Baranda Montgomery has a really great article I think you should read that I'm referencing here. And this is on individual-centered mentoring, where career advancement is personally defined. So remember before we talked about normalization to organizational norms. And this is really about the personal definition of what career advancement means. So they're going to focus on the individual needs and these personal aspirations, which are not going to necessarily be the same. And one thing that she advocates, and I agree with, is developing a network of mentors. So in this case, you see that we have a lot of different types of mentor nodes and resources. So a mentor node, for example, if you look at the individual mentee and they have a nice dark blue or nice solid blue line to the one that says mentor node there, that might be their lab, the lab they're in if you're a PhD student. And some of the ones with the dash lines that you see there could be different professors that are within the same department, but maybe not necessarily your PI. Your resource node there that has a dash line there, that might be your local chapter of your scientific society. And then, there's the mentor node you see in there that's not connected. So that might be somebody that you could connect to that's in your university or your institution that you're maybe not seeing. Then the outside resources, so that's where, the mentor node on the outside, that might be, for example, you might find a sponsor out there who was your PI's PI back when they were in grad school. Or you might find something like with Benning Singleton, somebody from outside who's going to help you for a short time, and they're going to act as a mentor and as a coach instead of maybe, that traditional mentor. So in this one, I filled in a lot of these examples. I have NSERP on there, which is a really great program that helps underrepresented minorities do summer research. I have PhD Balance there as a resource node, Black and Micro, a few others. But I think this kind of helps explain and illustrate that you really need to develop this network of mentors rather than just look to this one particular person as this is my mentor and that's where everything comes from. You really need to cultivate this network of people who can help you for different things, because a mentor isn't always, somebody who's a mentor for one person in one role might act as a mentor for somebody else in a different role. So your PI, for example, might act as a coach for somebody else to learn a new technique or to help them with the job hunt or help them with writing an article. And then that's a more short-term kind of a thing. Or that person may, years from now, you may reach back to your old advisor who is now no longer your traditional mentor, but could act as a sponsor because they've gained some more capital than you and they can help you progress in your career. So that's the general idea. So my take-home advice, despite what you've seen on here, if you're a PI, don't be afraid to be a mentor. Just because there's that statistic that having the postdocs and the senior PhD students help in the lab discussions, it results in that 4-time increase in skills trajectory. That really starts with the mentor, so the traditional advisor. So if you create an atmosphere where in a culture where that kind of interaction can happen, then you're going to help foster your students' growth. And also remember, you will be a different type of mentor to different people, and that's perfectly fine. We don't need to all think of mentors in one particular way. It's not always the same thing. And I stress this a lot, but ask your mentors what they need. Don't necessarily go in saying, okay, I'm a mentor, this is what we do, period. Here's how we help people. Really what should happen is that you need to find out what it is they want, what it is they're trying to accomplish, and then you can guide them towards accomplishing what they want and seeing what you can do to make that happen. And if you're a mentee, please tell your mentor what you need. They don't know. They, in general, are going to be acting in good faith and they want to help you. That's why they're in the job they're in, hopefully. And if you tell them what you need, most of the time I found that people are happy to help. If you are somebody who wants to go into industry, find this out hopefully before you join a lab, but make sure that your advisor can help you find somebody in industry, and don't be afraid to cultivate those mentors outside of your traditional lab structure who can maybe help you accomplish your goals if you want to go into industry or something else. So build that network of mentors, and really your mentors, your traditional advisors should help you to maintain and expand that network that you already have. I believe that's a really important part of being a mentor. So that's all I have for now. Kept it under time, hopefully, and I will end up mine. You can email me at joe@bendingsingletons.com. We're on Twitter at bsingletons, and we have a website, of course. I would like to thank International Microorganism Day for having us, and I will move this on very quickly to Justine Debelius, who's at the Karolinska Institute. And her talk is Who's to Blame? Thinking about Disorder in Human Microbial Communities. And hopefully I can make this go as seamlessly as possible.
Awesome. I am super excited to be here. And hopefully I can figure out how to actually present my screen. It's a success already. So I love following on the heels of Joe's talk about communities, because I think microbiomes are sort of all about communities, even if it comes from a different perspective. So my niche in this is that I'm kind of a computational person, and I like big human data sets, and I like to look at them from a community perspective. So I think that there's often this view of bacteria as either good or bad. And there doesn't always seem to be space for the bacteria that sort of wants to sit at home and not get involved in superhero battles. And this good or bad dichotomy is sort of old school. It goes back to this 19th century view of how do you know an organism causes disease? Well, you have to find it in sick people. You got to be able to culture it. You have to use it to reinfect people. And then after you infect them, you got to be able to find the same thing. And it's a great approach if your tools are a plate that you can use to culture bacteria. But we're not living in the 1890s anymore. And so we need to look at this in different ways. And more recently, we've determined that a lot of bacteria are hard to culture. They don't like to grow on traditional media. They don't like a lot of air. Some bacteria need other bacterial interactions to be able to function in the community, or they don't behave that way without additional metabolites. And of course, our microbiomes are huge and diverse, and we actually find bacteria that are thought of as pathogenic in healthy communities. And that's kind of more what I want to talk about today. And so our bacterial communities exist in communities just like we do. And I want to talk about some problems that recently occurred in my local community that are similar to things that happen in your microbiome. And so we had two problems this last week. Somebody ate a very expensive pair of wool socks in my apartment of 3 humans and a dog. And then we also wanted to make a chocolate cake, and it was kind of a struggle. So when it comes to the issue of the sock, there is obviously a culprit and someone in the community who is most inclined towards sock consumption. However, this culprit would like to speak for herself. And as with other organisms in a broader community, there's a metabolic and a behavioral issue from other community members. And so she's not a bad dog. She's a very good dog. She just needs support from additional members of her community. And a classic example of this microbially is Clostridium difficile infection. So C. diff is responsible for a huge amount of hospital-acquired antibiotic-associated diarrhea, and it can be life-threatening. But the thing about C. diff is that it exists as part of many people's healthy gut microbiomes. And so it becomes an issue when the metabolic state of the community gets shifted or antibiotics get introduced and C. diff gets out of balance, just like my dog. And so in the case of a potentially pathogenic organism, it is the organism that becomes a problem, but it's also the community. A second problem at my house was this issue of chocolate cake. So we wanted to make cake. We had a recipe, and it requires 7 ingredients. And so we were going through the ingredients and we had 3 in the cupboard. We needed 3 more. And it didn't matter who forgot their ingredient. Without one of those ingredients, we couldn't bake our cake. And so in a lot of communities, we see this loss of diversity. So it doesn't matter what organism you're missing. If you're missing enough organisms, you can't make a cake. You have problems and deficits in your metabolic state. And you see this happening in things like inflammatory bowel disease, again, sometimes associated with Clostridium difficile infection, as well as a response to certain host stimuli. And so based on these, we have multiple ways for microbial communities to get out of whack and to see weird things happen. And I'd like to conclude by saying that they're not bad microbes, just like all dogs are good dogs. They're just in a community that's out of balance. And I'll hand this back to whoever's next.
Thank you, Justine. That was really cool. I like the analogy with the dog. That was really great. My kid is in 4th grade, and she always appreciates it when people put their animals up on the calls. So that's great. All right, next is Kaylee Arnold from the University of Georgia, and we're going to talk about insects and their bugs, parasites, and bacteria. Take it away, Kaylee.
All right, hi everyone. I'm really excited to join y'all for International Microorganism Day. All right, and that looks okay?
Yep, we can see it.
Okay, awesome. All right, so yes, so my name is Kaylee Arnold. I'm a PhD student at the University of Georgia in the US. And yes, today I'll be talking about insects and their bugs, so their gut parasites and their gut bacteria. So the microbiome. So what is it? So if you're here today, I'm sure you've heard of the microbiome, or you may have heard it in health magazines, on the radio, or in the news. Well, your microbiome is just the collection of microbes like bacteria, fungi, viruses that live on and inside your body. And most of these microbes are good for you and necessary for your health. And so we have microbes all over our bodies, but and inside our bodies, and I study the bacteria found in guts. So gut bacteria and other microbes are important for many processes in your body. So they can help with many things like digestion, nutrient uptake to stay healthy, stress tolerance, and then especially parasite tolerance. So the bacteria and other microbes in your guts can help you from getting sick from parasites. So now let's talk insects. So disease vectors refer to all animals that can spread diseases. Mosquitoes are the most common ones, but there are many more, including kissing bugs, which is what I study, fleas, lice, and other non-insects like ticks and freshwater snails. So these animals can spread infectious diseases like malaria, Chagas disease, the plague, Lyme disease, and guinea worm disease. And these animals are responsible for at least 17% of all infectious diseases. So now let's talk insects and their gut parasites. So many infectious diseases are spread by insects or disease vectors. And these diseases are caused by different parasites, which can be viruses, bacteria, protozoa. So now these parasites tend to live and develop in the guts of insects, and they're spread either through their saliva when they feed on an animal or human, or in their feces, as is the case for kissing bugs. And so as I mentioned, so insects, like most other animals, have bacteria and other microbes that live in their guts. Now these microbes help with many functions like digestion and even reproduction, But importantly, we study their gut microbes to understand how these microbes either facilitate or even prevent parasites from spreading to humans. So now my research is kind of focused on thinking about the parasites and the bacteria that live in the guts of insects. So my research is focused on identifying which bacteria is found in the guts of insects. And then I look at how any changes to the environment or any changes to insect behavior, so when, where, and which animals or humans they're feeding from, and how that impacts or changes the type of microbes we find in insects. So if and how any external changes impact the gut microbes of insects. And then further, I seek to understand if any of these potential changes to the type of gut microbes found in these insects may facilitate and allow these insects to spread even more parasites. So now why do we care? Well, we care because insects are responsible for a lot of infectious disease transmission, both between humans, between different animals, and especially between humans and animals. So it's really important that we study these gut microbes, we study their gut parasites, and hopefully can understand how different factors, how different environmental changes, behavioral changes impact these gut microbes and ultimately their ability to spread parasites. And then also there's a lot of ongoing research that is working to alter different types of gut microbes in these insects to directly prevent insects from spreading diseases. And then here's a photo of me. So I do work in Panama. So this is me collecting some kissing bugs out of a palm tree to eventually go and dissect and look at their gut bacteria. And that is all I have for you today. Thank you.
There I am. Thank you, Kaylee. All right. That was really cool. Thank you very much. All right, next up is Elizabeth Diet, who is, I say from Microorganism, I believe she's actually associated with an academic institution as well, but I was on the Microorganism podcast. And here to talk about 5 tests. You coming through?
Sorry, we lost you for a second there.
Oh.
Sure. Could you just introduce the speaker?
Sorry about that. Yeah, I just got a notification that my internet connection was unstable, which is unfortunate. Yeah, next up is Tess Diet, who I always say is from Microorganism. I know Tess is actually from, I think, the University of California, Riverside. Is that correct? Yeah, but I always say Microorganism.
That's what I say too.
Okay. She's going to talk about 5 ways that microbes affect your communication. And off to you.
Excellent. Thank you, Joe. Okay, so as Joe said, my name is Tess Diet. I come from the University of California, Riverside, where I study plant microbiomes. But today I come to you as the founder of Microorganism and the co-host of the podcast, The Microbe Moment. And I'm going to talk today about 5 ways microbes affect your communication. So keeping in with the theme. But first I want to tell you a little bit about my blog. It's called Microorganism. We're science stories and news for the microbial enthused. And we have a whole bunch of different blog posts from history to scientist spotlights to a segment we call The Bomb, the best of microbiology, which is our favorite articles on microbiology. We also do recipes and talk about fermentation. And then recently we opened up the Society of Symbionts, which is our virtual school for microbes to learn to be good symbionts to the earth. So this is a little bit more about what we do. It's a little bit of everything from science fiction to history, to recipes to holiday favorites. So I have a lot of fun doing what I do. And with that, I just want to thank International Microorganism Day for having me and for Binning Singletons and Joe for having me part of the session. And also to thank my team, Jonathan Mitchell, Julie Grubat, and Celia Shee, who helped me put together all of the stuff you see today. So I'm going to talk about 5 ways microbes affect your communication. And the first way is small talk, that sort of jib jabber that you have with people when you first meet them. It's usually, hi, how are you? What's the weather? What are you eating? So those are the 2 topics that people always ask me. And when I answer those 2 questions, it always has a microbial component to them. So what's on the microbe menu? We have cheese, we have bread, we have chocolate, yogurt, kefir, coffee. All of these are made possible because of microbes. And I think that is a beautiful thing. If there's a food on here that you don't like, or if there's no food on here you don't like, I would be surprised. They make some of the best food I think we have on the world. Next, I want to talk about weather. So petrichor is that beautiful earthy smell that you get after it rains. And this is actually caused by a microbe called Streptomyces. And they produce this metabolite called geosmin, which causes that smell. And then the final piece that I want to talk about with weather has to do with snowflakes. You know how every snowflake is unique? Well, that is made possible by Pseudomonas syringae, which is a microbe that may be hiding in your snowflake. So Pseudomonas syringae has a protein that's called INAZ that helps form these snowflakes into the unique structures that we see and love. So that's number 1 and 2, small talk, both in food and weather. Now we're going to move on to number 3, which is alcohol. And anyone who's ever had enough alcohol knows that it does impact your communication. Some people become angry. We don't like those people, but other people become more flirty or more loose with their lips and tell secrets. So alcohol has a way of definitely impacting our communication. And just another little plug here, I've created this microbe-inspired cocktails ebook, which is about a dozen different recipes that are all inspired by microbes. So if you'd like that, all you have to do is sign up for our mailing list and we'll send that to you. So number 4 gets a little bit more serious. We were talking about mental health. So if if you don't have good mental health, this can definitely affect your communication. You may not want to go outside. You may not want to communicate with people. And while mental health is a multifaceted area, they are finding ways that your mental health is connected to your gut microbiome. And you have this axis they call the gut-brain axis, which is this informational highway from your gut to your brain. So as an example of how microbes may be impacting it, we have Lactobacillus rhamnosus, which can produce GABA, which is linked to decreasing stress responsiveness and anxiety and depressive-like behaviors. So my 5th and final one is halitosis, which is stinky breath. So stinky breath is another thing that can be very multifaceted. It can be caused by a number of things, but one thing that it may be caused by is a sulfur-producing bacteria that live in your tongue or in your throat. Luckily, in the world of virtual communication, this aspect doesn't actually affect your communication, but once we go back into the world again, this may be impacting your communication more than you'd like. So with that, I just want to conclude and say that microbes have a limitless potential to affect our communication and our daily lives. So just to wrap up what we discussed, we talked about how microbes are in food. We talked about how microbes can affect weather, and this has to do with small talk and communication. We talked about how alcohol impacts communication and how we have this because of microbes. We talked about how mental health can also be impacted by your gut microbiome, and microbes can affect the smell of your breath. And just one final plug, I do have another talk in an hour. So if you're free, this is Microbial Arts and Crafts. It will be here as well. And this is a collaboration, again, with about 7 other fabulous microbiologists. And we're going to do a lot of really fun things. So I hope to see you there. So thank you. And that's all from me.
Thanks a lot, Tess. I'm sorry I pronounced your last name wrong the first time.
That's okay. Everyone does. Names are hard.
I think we will definitely be back on here with my 4th grader for the arts and crafts, because I've never seen anyone love arts and crafts as much as I can. So
we're going to have some fun stuff. We have a live action drawing session. We have fermentation demonstration.
That'll be great. Okay. So next up, so everyone, we actually are going to probably finish up a little bit early. So I will try to come up with some questions for you. But if you have questions for each other, or Ben, if you want me to talk a little bit more about the Binning Singletons thing, I didn't really focus on that for my part. But if anyone wants that explained a little bit better, I can do that. But I really wanted to make sure that everybody else had their chance to talk. So next up, we have Sarah Woodward, who is at the University of British Columbia. And she's going to talk about a travel guide to the gut, how enteric pathogens navigate the gastrointestinal environment. So take it away, Sarah.
Awesome, thanks. So I'll just see if I can figure out how to share it. Okay. Can you see the right thing, or can you see my note that tells me to tell you that my name is Sarah? Does it look okay? All right, good. All right. Hi, everyone. My name is Sarah Woodward, and I'm a microbiology PhD candidate at the University of British Columbia in beautiful Vancouver, Canada. And I've really enjoyed the talk so far, and I'm going to talk to you about the incredibly dynamic and complex ecosystem we carry around with us every day, which is the gut. We've heard some great things about some of the bacterial communities that live in the gut, but I'm going to try and convince you that the gut itself, or the context in which those communities are existing, is also really, really important. So before we get started, I just want to introduce diarrheal disease to give us some context for this talk. So diarrheal disease remains a major health burden. It's the leading cause of death in children the age of 5, and every day diarrheal disease is estimated to kill more than 2,000 children. That's more than AIDS, malaria, and measles combined, and it accounts for 1 in 9 child deaths worldwide. Now, there are many microorganisms that can cause diarrheal disease, including both viruses and bacteria, and I'm going to refer to these sometimes as enteric pathogens. So enteric meaning that they're in the gut, and pathogen meaning a microorganism that causes disease or damage. So I'm particularly interested in those bacterial agents, some of which I'm sure you've heard of, such as E. coli, Salmonella, and Vibrio cholera. And these are predominantly transmitted through the ingestion of contaminated food and water. So while we know quite a bit about how these enteric pathogens cause host damage and the molecular mechanisms that they use once they reach the cells lining our gut, we still don't have the full picture. We know that ingestion of these bugs leads to disease, but we don't know a ton about the population dynamics or the way in which these bugs are actually reaching their gut niche in the first place. And to figure that out, we need to open the mystery box, which is the gut. So we don't tend to give the gut a lot of thought. It's just a tube where food goes in and stuff comes out the other end. But it's actually not a passive environment at all. It's a complex and dynamic series of parallel ecosystems. So for example, the gut physiology itself actually changes drastically from the long villi of the small intestine that are helping us with absorption of nutrients to thick mucus and deep crypts of the large intestine. There's also regional differences, from immune cell hubs to different microbial populations. And what these regional differences do is they create gradients. So down its length, we can see, for example, less undigested nutrients as you'd expect, or an increase in mucus thickness, for example, or some things that can fluctuate along its length, such as the pH or the acidity within the gut regions. In any one section of the gut, there's also these radial or cross-sectional gradients. If you imagine looking down the middle of a pipe, for example, there's an increase of oxygen closer to the cells that line your gut compared to in the middle. So it's unsurprising then that enteric pathogens have evolved to use the presence or absence of these regional signals to determine where they are in the gut, or even what stage of disease the host is in. And this tells them, importantly, where they need to go and what they need to do when they get there. So some of the behaviors that are directed by sensing these gut signals include growth of a population, motility or moving to a particular area, attachment to your host cells, and even virulence, or those host damaging behaviors such as hijacking our host cells. But the good news is that because of all of these regional differences that bacteria have to navigate to cause disease, the gut is actually highly restrictive. For example, major barriers to causing disease for an enteric pathogen include passing through the highly acidic stomach acid, which is able to kill a very large number of bugs. There's also peristalsis, or that luminal flow that helps us not only push our food along the GI tract, but also helps to flush away bacterial pathogens. And of course, those resident microbes you've been hearing about, which are able to resist colonization by enteric pathogens, in some ways just by taking up real estate and nutrients in the gut. So you can think of these each as barriers or literal hurdles that pathogens need to overcome in order to host and cause disease. If we can identify and manipulate these barriers or even alter the regional signals that pathogens respond to, this can allow us to better prevent and control diarrheal disease. But understanding gut barriers to colonization can even help us to improve probiotic therapies, or that is the introduction or colonization of those so-called good bacteria, which we have a notoriously difficult time getting to stick around in our gut after we've ingested them. So I hope I've convinced you to think a little bit more about your gut and why it's important to understand not only how the enteric pathogens damage our host cells, but how they get to those host cells in the first place and whether we can intervene in the process. So with that, thanks for listening. Special thanks to Joe and Benning Singletons for the opportunity, as well as the International Microorganism Day team. And of course, I'd also like to thank my colleagues in the Finley Lab, where I'm doing my PhD here in Vancouver, including Jorge Pena, who's going to speak next. So, and if you have any questions, feel free to reach out to me. Awesome.
Okay. Yeah, with that, we're going to go right to Jorge, which actually is here on my screen, but I don't know where he is on your screen. From the University of British Columbia, and title is Everybody Talks, Bacterial Communication in the Gastrointestinal Tract, which I don't know if Everybody Talks is a reference to a song title, but if it is, I get it. So
I'm impressed you got the reference. That's pretty. Thank you so much. Am I sharing already? Can you see everything? Okay. Okay, just give me 2 seconds. Okay, well, thanks so much, everyone. And like, as just like said, I'm going to talk to you a little bit more about like bacterial communication in the gastrointestinal tract. Jorge Pena, and like, I'm happy to talk to you. So like, thanks everyone from the International Microorganism Day and Benning Singletons for like inviting me to give this little talk. So I don't know why I even had to give like an introduction at this point, like with all the beautiful talks that we just heard. Like, I think that's not much that I can say, but as we have heard so far right now is that like everyone, we know that bugs can just be found everywhere. And like, since microbes can be so well adapted, they basically can be found on any environment that you can imagine, from like leaves from a plant, their roots, the soil, to your like skin, any environment in earth that we can find, even our food, as like we have heard from Tess, like our cheese, our coffee, our beer, to like even the most unhospitable like environments, such as like the bottom of the ocean floor, the permafrost, even in volcanic environments too. But one thing we don't talk too much, or we don't hear too much, as Justine said, is that like bacteria are just not living on isolation. They actually live on very complex communities where they can actually be interacting with each other, where they can even be like cooperating, so they can actually help each other to then actually thrive in this like really like harsh environments. Or they can even be competing, so they can actually, like as Sarah was pointing out, thrive when there's very hard competition to like actually colonize a niche. They need to compete so they can actually thrive. Or even cheat, which I found hilarious. They can actually like take advantage of others so they can actually flourish and establish. Now, since they live on really complex communities, they actually, it's been found that they can actually talk to each other. At least some bacteria, especially like gram-negatives, can do a process known as quorum sensing, which is a way of bacteria-bacteria communication. And when they grow, they can secrete really, really small molecules. And as they start growing and accumulate more in the environment, they can use this as little cues that allows them to respond what's happening around them. So it could be a cue of like, who's around me, or like, are there many of my brothers and sisters around me, or is there a lot of people that I need to compete with? And then use this as a cue to then regulate different group behaviors, such as like production of like toxins, or like motility, like the production of biofilm formation, bioluminescence, and others, which would be like behaviors that are very expensive for the bacteria to produce by itself. But when they group like work together as a team, then it's something that it can be like a competitive advantage. But now, as I told you like earlier in my first slide, like we know that bacteria lived in like many complex communities. And we know that like they can talk about, like they can use quorum sensing or like this communication signals to talk with each other. But are they actually talking in our gut? and it's something that is like still not very well like explored. And it's very interesting because like the human microbiota is still one of the most richest and most densely populated communities inside of our body, but still we don't know actually like who are they actually like these molecules or like produce, or can they be secreted, where are secreted, what they're actually doing. And there's many questions that I'm trying to like answer in my PhD, but we do know that actually they can be measured and detected in the guts in very like and different amounts. Then as well that it's believed that they can play a role to like actually contribute to our health and maintain like a healthy composition of our gut microbiome. So now, why it's so important to like explore this like communication signals in our gut? And it's because they can actually confer like a really interesting approach. For example, some pathogens can use these little communication signals to like coordinate how they can cause virulence or harm into ourselves. So if we can understand better how they can use these signals to like modulate this virulence, we can like maybe make different like treatments that can cause like the inhibition of this communication signals, not allowing this bacteria to mount these group behaviors and just act them as individuals, and then just allow like the immune system to naturally clear this infection instead of having to use antibiotics. Or at the same time, we can use them as a different way to like to have a disease microbiome that it could be like associated with disease, and then help them to like push the creation of a more healthy or a more balanced microbiome by allowing different like microorganisms to thrive and like grow. So with that, I hope that I can like at least convince you, and like takeaways from my talk is that like bacteria can live in very complex environments, and they can actually be talking to each other and responding to what's around them. They can use this small molecules known as autoinducers to like communicate with each other. And as well that like exploring and like these tools can offer a tool to then use it to like use this as a tool to like I don't know, like I'm lost of words, but to like incorporate it as like another molecular toolbox to like create healthy environments. And with that, thank you so much for everyone, especially everyone in the Finley lab who has let me explore a lot of the questions that I like proposed today, and as well to Binning Singleton's like showing a very nice environment for me to show my science, and as well my International Microbiome Day for like a place to like express my talk. So thanks so much, and if you have any questions, please feel free to contact me. Thank you so much.
All right, thank you very much. We do have a question for Tess about the microbe-inspired cocktails, and what is the most alcoholic one? Tess, do you know that answer?
I do, yeah. So we made a cocktail, and it's called Toughest Tardigrade, and it is a beer float cocktail that includes a shot of vanilla, or 2 shots actually, of vanilla vodka inside a 15% stout. So it's fairly alcoholic, and when you mix your alcohols, you will definitely feel it a little bit faster, which is why we named that one Toughest Tardigrade.
That's pretty good. So this was suggested. I think it's actually hopefully a good one. Well, let me give a little bit of a background first. So Binning Singletons is something that we came up with back in 2019. Actually, I came up with it a year earlier and wanted to try it, and then we actually did it in 2019 at the ASM Microbe meeting. And so the idea was that a lot of people go to these. I don't know if you're not in science, you probably have been to some kind of a meeting or conference with people from your industry, and you get there. And there's just all these people, and you don't necessarily know them all, but you may have heard of some of them depending on your field, and you might be intimidated because honestly, when you suddenly show up and there's thousands of people, it can be a little intimidating, and imposter syndrome can really kick in. So our idea was to help people kind of jumpstart and raise the floor of what their meeting experience is. So rather than having to go through those first 2 or 3 times on your own and learn a bunch of stuff kind of the hard way, and maybe have negative experiences and never come back. We want to make that first experience pretty good. So the idea was, so we used a lot of puns from microbiology, and so that I know that this audience is really the microbe interested public, not necessarily microbiologists. So our puns kind of go hard in on the micro, so they're not necessarily as useful as an explanation. But essentially what we do is we want to group together. So if you've gone to one of these things and you've ever felt left out, or even, I think we've all felt this if you've been a new kid at school, or you got to high school and you had different friends and whatever. What we try to do is give people a community, so group people in groups of like 2 or 3, and then give them somebody who has done the meeting before as a mentor to kind of help guide them through it. And so that would be our Ben the Singleton's idea. The next thing we do is what in our description is called horizontal transfer, which comes from the fact that microorganisms often can transfer genes from one to another that did not evolve within that line. And so this is where sometimes you get bugs that acquire an antibiotic resistance that they didn't have because they're around something else that has that. So suddenly an organism that wasn't really that bad before, now you get an ear infection and you take your antibiotics and it doesn't work because it has got this horizontal transfer of this nice gene. For it, it's a nice gene. For us, it's not. So our idea is that this horizontal transfer takes place between both the members, so the peer-to-peer kind of mentoring kind of thing, but also that the more experienced person can offer their advice. Also importantly, unfortunately, we didn't get to do this at the World Microbe Forum, but we kind of did it virtually with some of our Zoom calls, is you want to bring all these people together who feel this way, and it helps people realize that they are not the only ones who feel this way. And it's like actually some really, really cool, smart people that maybe you were intimidated by are also there. And you're like, oh, well, okay, cool. I mean, I'm in this group too, so we're all good. And then the next we jokingly call it the exponential growth phase, which if you are growing bacteria, there's a phase where they just grow and double every, if you're E. coli, you can double every 15 minutes under the ideal conditions. So, you suddenly have a whole bunch more E. coli than what you started with. So our idea is that these people now who've worked together and kind of spent a little bit of time together now can really grow at a much higher rate than they would have before. And so really that's kind of the general idea of what Benning Singleton is. Again, for me, it was really as a result of having gone to many, many meetings by myself and feeling like I didn't really belong there and everyone else was smarter than me and they knew what they were talking about and I was faking it. And so it was kind of a way to help get over imposter syndrome. I don't know if you can see right above my head. There's a quote by Charles Darwin back there that says, but a lot of you have heard it. It says, but I am very poorly today and hate everyone and everything. One lives only to make blunders. And I have this up in my office because if Charles Darwin can have that day, then I can have that day. And I have that day a lot. So I think it's important for everyone really to understand that, even if you feel like sometimes you're not measuring up to what some idea of what you think a scientist is, that actually, or if you're in another field, whatever your profession is, you're probably doing better than you think. And if you find a community of people that you can share with and talk with, you'll find out that you're actually probably doing a lot better than you think, and that it's really a lot more fun if you go through this together. So I very cordially like to say that we are cultivating a culture of community. Again, cultivating culture, all those nice bacteria jokes. But so one question somebody wanted to ask is what kind of impact Benning Singleton has had on your route. I'm not sure I like the question too much because I don't know that I want to take a lot of credit for really changing anyone's life, but hopefully people have had some small benefit from being with us. So if anyone wants to jump in on that, and then I'll also take other questions and pass them on. Anyone? Justine? Oh no,
I was just saying it's awesome. Two thumbs up.
Oh, Kaylee has her hand up. Okay. Kaylee.
Sorry.
I'm misreading Zoom responses. It's my fault.
Well, so I'll just say quickly is that so I'm kind of new to microbiology. I was trained as an ecologist and in marine bio. So kind of just having this avenue to have like less imposter syndrome and kind of feel a little more kind of connected to like microbiologists has helped a lot.
I want to like say too that like I've been to like a conference before, and it's kind of like so challenging when you don't know anyone and you're literally like the one odd one in the room, and you just, I mean, I didn't even know how conferences work. So like it makes such a difference when like you can have like a community to thrive, right? Like as I like said something on my talk, like that makes a huge difference. And it was so much easier to like, oh, I want to talk to this person or whatnot, like having like that group as a support. And the other thing is it just opens like opportunities too. Like I feel like it helped me to expand my social like media and like reach, and like as well gives you more opportunities, such as like that's why I'm here, right? Like, so thanks.
Yeah, I'll just echo that. I think, I mean, when I heard about binning singletons, I absolutely fell in love with the idea. And I think sometimes when you hear these ideas, you can think, well, you know, maybe they're not actually going to give you what they're saying and it's all just talk. But I going to the World Micro Forum and being part of binning singletons, they had actual moments and networking hours where you could talk to people. And it was just so nice to hear other people say like, you know, I'm super introverted, I hate talking to people, and just being able to relate to other scientists who also feel the same way. It just makes you feel a little less alone in the virtual world.
Yeah, I have nothing original to add to that, but I agree with everything everyone has said. It's been a fantastic networking opportunity, and even just kind of going to those other broader conference networking events as a little binning singletons team was key, because otherwise I don't think I would have gone to them. And just like learning how to navigate some of the huge conferences like that, it's been fantastic. So yeah.
I particularly enjoyed when we all kind of descended on Johnny Chang as he was drawing art. So I have my shirt that he drew for me. You saw in Jorge's presentation, he also had one. I think like at least 50% of the people in that session are from binning singletons, because I made sure to share it. So that was pretty great.
That was kind of crazy too, because I had to wake up like at 4 a.m. to like go to the
Yeah, I actually thankfully had gotten mine at the session the day before, because I had something else I wanted to do and I couldn't get in, because even though I got up at 6, I still couldn't get in because he's such in demand. But his art is awesome, so it's worth it. Okay, so Ben, do you have anything that you want us to talk about, or do you have any questions that we haven't seen? Because I'm not seeing YouTube or any of the other formats.
No, we don't have any questions coming in at the moment, but we've got how long is it? A couple of minutes left. So I guess are there any more questions from you guys? Is there any kind of final remarks? Otherwise, we can close the session. It's been absolutely fantastic, I can say. I really enjoyed listening to you all and kind of learning more about binning singletons. It's really inspiring. So thank you for that. Are there any kind of closing remarks? Anything anyone would like to say?
I know that I can say that having these kinds of presentations where, you know, junior early career people are presenting their work, as somebody who's been in my job for 21 years now, it almost increases my imposter syndrome, because I'm like, oh my God, they're doing things that I can't even imagine, and it's amazing and really awesome and cool. So I think it's really good for, because I know a lot of us, now if you haven't been to a lot of conferences, you maybe haven't felt this, but sometimes if you go to the similar conferences, you kind of hear the same talks from the same people over and over again. And I think it's so hugely important to get those talks in where the junior people, the early career people, get to talk about something that they're the ones working on it in the whole world, because it's a small little thing, but it's like their central focus, and they're going to teach you something you don't know. And I really hope that in the future we I know that ASM has done a pretty good job of this, of adding some talks for some junior people, but I really hope that a lot of conferences go this way. I know that you've got the headliners and people want to hear the big hitters, but there's some really, really good work being done out there by the really career people, and I would love to see it. So that's my little soapbox.
I can also say something maybe. So I was the one asking the question on what was the impact of winning singletons on your route. I didn't know it before. So I have to say that I find it amazing. I want to join, but I'm a postdoc. I feel I'm a bit too old to be a mentee, too young to be a...
I was a mentee.
Yeah?
Yeah.
Okay, maybe it's not so...
We had a tenured
late.
professor as a mentee one time. So
really? Okay.
yeah. No, I mean,
So maybe I think I'm more of a mentee than a mentor, but I have to say I totally relate to the feeling of you're the odd one, or you don't know how to act, or if you can approach someone, or you feel insecure about saying something. Everyone feels like so much smarter than you. So great initiative. I just wanted to say this. And I also want a t-shirt. They are cool.
so this one is a one-off. I have the only one of these, but
that's the original one, I feel.
Yeah. Well, this one's different than all the other ones. I think you can see Sarah and Jorge,
yeah.
the ones we did for World Microform. I actually have one that I have whipped up for International Microorganism Day, but I have to clear the logos with IMD folks to make sure I can put it on there. So, but actually, I can show it real quick if anyone wants to see it, because I think it's...
Yeah, let's see it. Let's see it.
Sure.
Where did I put it? All right, so there it is.
Oh, nice.
So I've got the
nice.
singletons microbes on there, and then I put the International Microorganism Day microbes on there as well.
That's nice.
So I actually have... Oh, something just... I broke something. It went away.
No, it's fine.
Very cool.
So yeah, I will totally look up to it.
So that's what. Oh, and then I did put my oh, where'd it go? Yeah, I put my it's a front and back shirt, and so I have my... Where's my share screen? There we go. Okay. There we go. Yeah, on the back I put my nice cheesy slogan of cultivating the culture community. But again, you can see I have the logos there, so I got to make sure that that's actually kosher before I actually print it.
Excellent, excellent. Send it over. I'm sure it won't be a problem, but yeah, it'd be very nice to see that on a real t-shirt. And yeah, with that, I'll say thank you again to everyone. Thank you very much, Joe, for sharing, and thank you everyone for your amazing talks. And I guess we'll move on now. It's that time of the evening. Hours flown by. It's absolutely flown by. And now we're going to move on to the next session, which is vaccines and vaccine hesitancy. And I see everyone waiting in the waiting room. So I'll say goodbye to you guys. Thank you so much. And we'll do a swift, smooth changeover as we now welcome to the call Janet Hart, Leela Savela, and Henning Anderson. Welcome, welcome, welcome. How are you doing?
We're doing great. Well? It's a little bit gray in Boston, but we're doing good.
Good, good. I'm really kind of getting confused. I wanted to say to you that how are you doing this evening, because it's evening here, but you know, it's International Microorganism Day. We're talking to people all over the world. You know, you can't say that. So yeah, good to know where you are. And yeah, without further ado, I think I'll let you get started straight away so we can get hearing about what you're talking about. So Janet, I think you're going to start. Is that right?
Yes.
Yes. And so you're a professor of biology at Massachusetts College of Pharmacy and Health Science. Is that correct? Yeah, it is. It is. But it's a pleasure to have you here. And yeah, I'll hand the floor over to you straight away.
Great. Thank you so much. Let me just kind of organize my tiny little desktop here. Yeah, happy to be with all of you today. We're here in Boston again, as I mentioned, a little bit of a gray evening. So good morning, good afternoon, and good evening, no matter where you are. So as was mentioned, I'm a biology professor here in Boston in the United States. I thought I would disclose I'm not associated with any industry or governmental institutions. I'm here solely as an educator and a mom. And together with my students, we really hope to help people understand the science around microbes and vaccines. And that's what our talk is all about. And we also hope if you have some questions, we'll do our best towards the end to answer them. All righty, so let's get started. So microbes are everywhere. You may have heard that in other talks for International Microorganism Day. Some are harmful, but most are actually quite helpful. In 1 drop of surface seawater, there's about 10 million viruses. Fortunately, very few actually make large animals like fish or humans sick. Almost all of those viruses actually attack marine bacteria. Viruses, of course, unfortunately, can also infect other animals, birds, reptiles, insects, and of course, us. Viruses have probably existed for millions and millions of years, evolving with other life forms, and that's millions and millions of years of developing and adapting and getting better at what they do. And for the microbes that are the bad guys, unfortunately, that's making us sick. So this talk is focused around stories. Of course, stories are really important for us as human beings, no matter what culture we come from. So I wanted to start by sharing this important story from not so long ago. This gentleman, Morton Chapman, he writes, "I can remember no experience more horrifying than watching by the bedside of my 5-year-old stricken with polio. The disease attacked his right leg, and we watched helplessly as his limbs steadily weakened. On the 3rd day, the doctor told us that he would survive and that paralysis was the worst he would suffer. I was grateful, although I continued to agonize about whether my wife and unborn child would be affected. What a blessing that no other parent will have to endure the terror that my wife and I and thousands of others shared that August." So we've been working very hard to eliminate polio from the planet, which we can do since it only infects human beings and doesn't hang out in any other animals. The only pathogen we have ever gotten rid of in the wild globally has been smallpox, and that's because it also only infects human beings. And vaccines were key to making sure that that would happen. In fact, we've seen incredible reductions in illness and death in the 20th century for many diseases, and that's because of vaccines. You can see here on the left a bunch of different diseases. This is just a small sample. You can see, right, that we were able to, again, bring down smallpox as well as diphtheria compared with the 20th century levels of disease to 0 in the 21st century. We also dramatically decreased in the United States the amount of paralytic polio, measles, mumps, and rubella, as well as congenital rubella, which causes birth defects. But when diseases become less frequent due to high vaccination rates, we can become a bit complacent and we forget just about the impact of these diseases. And that's why certain kinds of infections like pertussis can come back. And we've seen this in the US, like is shown here. So stories and scientific evidence are really very, very important in helping people make informed decisions to protect their health. So here are a bunch of vaccine preventable diseases. Some of these are viruses and some are bacteria. And I'm so grateful that I've had access to vaccines that I could protect my kids from all of these different kinds of diseases. I didn't have to worry about my kids getting a lot of these. My son didn't lose his hearing after getting a mumps infection. I can protect both my son and my daughter from deadly cancer and meningitis. And my kids are much less likely to develop the painful condition called shingles later in life because they've been vaccinated against chickenpox. So I'm not just protecting my kids though. The great thing about vaccines is that it protects their friends, teachers, and everyone else around them. I wanted to point out a couple of these vaccines, starting with rabies. If you get infected with rabies, you will die, 100%. It's got a 100% mortality rate, which is very scary. And rabies does exist in many, many parts of the world. And every year people do die from rabies. But this vaccine, the rabies vaccine, is pretty amazing because it's one of the rare situations where a vaccine can actually be given after you're infected. And so it saved many, many lives. Hepatitis B is so common, we think upwards of a third of the population of the planet is infected with this type of virus that attacks your liver and can cause liver failure and cancer. So a hepatitis B vaccine has been amazing in really preventing that kind of outcome. Rotavirus is so contagious that every single child on the planet eventually gets infected with it unless they are vaccinated. And it's the third leading cause of infant death worldwide due to diarrhea. So I wanted to step back a little bit in time and share the fact that immunization actually is a concept with a pretty long history. The top picture that you can see in the top right of your screen is from Mexico in the 1500s, and it very sadly shows the devastating effect of smallpox on the native population. The picture down in the lower right corner is Fulin, the Shunzi emperor in 17th century China. Unfortunately, he died of smallpox, but when his son became emperor, his son actually survived smallpox as a child, and his son then became a big champion for supporting inoculation in the population. Between 1910 and 1940s, we had some pretty incredible advances in really fighting a lot of major pathogens, major diseases that were causing a lot of death and a lot of illness. We were able to develop vaccines against tuberculosis, yellow fever, diphtheria, tetanus, pertussis, influenza. Between 1970 and 1990, we had a lot of victories. We were able to, in the United States, get rid of polio completely. We were able to eradicate smallpox, as I mentioned, from the entire globe, except for a few laboratories, but it's no longer in the wild. I'm old enough that I was vaccinated against smallpox, but most people today don't need to be because we got rid of it. We also had some problems though. We had some adverse events. There was an earlier, it's not the same rotavirus vaccine that's being used now, but there was an earlier version that caused a rare but very serious side effect in infants. We'll talk a little bit more about risks versus benefits of vaccines a little bit later. We also had some setbacks. We had some major resurgence of measles in the US and in Europe, and a few other places around the world. We had a little bit more recently, we've had some failures of vaccines. There were some HIV vaccines that showed some promise early on that ended up not being able to work properly to help protect people against HIV, and that was obviously very disappointing. We've also seen massive improvements in the way that we deliver vaccines and make safety and efficacy a really top priority. And then I also wanted to talk about a really important topic, and that's equity. Measles is no longer a common disease in the United States, but globally, it's still a major cause of death of children under 5. And that's highlighted by this slide from data from the WHO, the World Health Organization, that shows just how something that really should be eliminated through vaccination has not been because of access. We have not been able to get the measles vaccine to enough parts of the world to really bring down this rate of disease and death in children and adults. So I wanted to share just a little highlight from the report that this is taken from. This is from 2019, not so long ago. And the report states, measles outbreaks continue to spread rapidly around the world, with millions of people globally at risk of the disease. In the first 6 months of 2019, reported measles cases were the highest they have been in any year since 2006, with outbreaks straining healthcare systems and leading to serious illness, disability, and death in many parts of the world. The Democratic Republic of Congo, Madagascar, and Ukraine reported the highest numbers of cases. However, cases dramatically decreased in Madagascar as a result of a nationwide emergency measles vaccination campaign, highlighting how effective vaccination is in ending outbreaks and protecting health. The largest outbreaks were in countries with low measles vaccination coverage, which left large numbers of people vulnerable to the disease. At the same time, protracted outbreaks are occurring even in countries with high national vaccination rates. This results from inequities in vaccine coverage, gaps and disparities between communities, geographic areas, and among age groups. When enough people who are not immune are exposed to measles, it can very quickly spread. The United States reported its highest measles case count in 25 years, and in the WHO European region, there were 90,000 cases reported for the first 6 months of 2019. The reasons for people not being vaccinated vary significantly between communities and countries, including lack of access to quality health care or vaccination services, conflict and displacement, misinformation about vaccines, or low awareness about the need to vaccinate. In a number of countries, measles is spreading among older children, youth, and adults who have missed out on vaccination in the past. So I personally have been very fortunate to travel the world in my life, and health equity is something that I care very deeply about, as I know many of my colleagues do. And so I wanted to share a little bit of one of my own stories. Here I am a long time ago with fewer gray hairs, with my little baby girl. Isn't she cute? And she spent the first 5 years of her life being a little Parisian girl, eating croissant and running around beautiful places like this. Being a mom, I have a smile on my face in this picture, but trust me, I was exhausted trying to keep up with her. And I remember her getting her vaccines, but there was one, the pneumococcal vaccine, we somehow forgot. And I honestly don't remember why. Fast forward to age 5, and after a really bad prolonged cold, she ended up very seriously ill. And in this hospital, in the picture on the upper right of the screen, she was sick from, you guessed it, Streptococcus pneumoniae, which is the bacteria that causes pneumonia and meningitis, and was the pathogen that the vaccine she missed should have protected her against. It was honestly very scary to see her hooked up to IVs and very sick. But fortunately, there was a happy ending to this story. Here she is with her little brother not so long ago. So let's talk a little bit more about vaccines, the different types of vaccines that are out there, and maybe dispel a few myths. All right, so the idea behind vaccines is to fake out your immune system. Give it something that looks like the bad guy to react to and build up some protection against. Infections are always a race between your immune system and the nasty pathogens, and pathogens, unfortunately, often win that race. So vaccines give us a really crucial head start in that race. So one of the older kinds of technology we use for vaccination is something called a live or attenuated vaccine. These are very effective, but they often have a little bit of a trade-off because there are sometimes more reactions, right? Maybe more soreness at the point that you get vaccinated, maybe more likely to cause a fever. And so we've also developed what's called an inactivated vaccine. And this tends to have less what we call reactogenicity, right? Those side effects that can be uncomfortable. But because we're being a little more gentle with our immune systems, we often have to give booster doses. We also have toxoid vaccines. So many pathogens have nasty tricks that make it harder for your immune system to fight them. Tetanus and botulism produce toxins that are some of the most deadly substances on Earth. These are both bacteria. These bacterial toxins are sub-immunogenic, which means your immune system doesn't really realize that they're there. And so you and your immune system are pretty helpless against them. So by developing these vaccines, we can give your body, again, that head start. So if you are exposed to these pathogens and their toxins, you can fight them off really, really well. Many people were really understandably concerned about the newer mRNA vaccines, but they're not as new as people thought. There's actually been research going on for at least 10 years, and they had been tested in humans for other kinds of diseases before COVID came onto the scene. We also have some vectored vaccines, which again, they're about precision. They're about all the things that we've learned over many, many decades about the immune system and about different kinds of pathogens. And we've become really good at being able to make a very precise type of vaccine that's very safe and also really does the job it needs to do. So I wanted to give you a little bit of an analogy. All right, so here we see a picture of a 1970 Honda Civic, which is a great, reliable car. Honda Civics are great cars. It will, if this is all you have, it will get you absolutely to where you need to go. Very reliable. But what if instead of this 1970 Honda Civic, I offered you a 2021 luxury Tesla? Would you still want the 1970 Honda? You know, we drive around in new cars all the time and we feel safe. Why? Because they've been tested extensively and the technology, comfort, and safety are constantly being improved. And the same is true for vaccines. So try not to be too scared if something is new. It doesn't necessarily mean it's not going to be really safe. Vaccines are given to healthy people and they undergo much higher, more rigorous standards than anything else we put in our bodies. So let's move on to the sad story that we've all been living through, and that's the story of the virus SARS-CoV-2. This is an excerpt from a paper I remember reading in January 2020, very naively being a little concerned, but not realizing just how much this was going to upend the lives of every single person on the planet. There's reason to feel hopeful, right? We've been able to develop globally several different vaccines. And when you start reading some of this data, this is from Our World in Data, which is a great site to get information about, and data obviously, about the coronavirus and COVID-19 disease. You know, and you read this and you see, okay, 40% of the world's population has received one dose of COVID vaccine and 5.2 billion doses have been administered and 31 million are administered each day. And then you look and you see, less than 2% of people in low-income countries have received at least one dose, right? So again, we have some work to do. When you look at this particular map, right, share of the population fully vaccinated against COVID-19, this is a major, major problem. You know, I deeply appreciate being in a country where I have access to this and many other life-saving vaccines. But if we've learned anything from SARS-CoV-2, we're all interconnected as human beings and we depend upon each other in many ways. Many of us in developed countries need to continue to push our governments to share more vaccines with developing countries. And this is essential to bringing the virus under control. There are a few things that I say with certainty about this crazy virus, but one is that it's not going away. It's in too many places, in too many people, and people can spread it before they know they're even sick or infected. We have to learn how to live with this virus, and we can only do this if we stick together globally and help each other. So speaking of sticking together and helping each other, I'm going to hand things off to two of my awesome students who are going to talk about the journal club that we worked on last spring to help each other understand the evolving science around this virus. So Leah and Henning, take it away.
Hi everyone, my name is Leah and I am a third year here at MCPHS in the Medical Molecular Bio program. And for our project that we did for our journal club last semester, we were actually able to take information every single week from some of these amazing organizations that you see on the screen. For me personally, I took information from the World Health Organization and I reported on it every single week. And so it was really great because we had 5 students all reporting from these great sites and great data sharers, and we were able to talk about what we were finding. For me, I reported on the WHO's weekly epidemiology update, and so I was able to look at graphs and really determine what those graphs were showing. Some of my other peers, they reported on the CDC, as well as the New England Journal of Medicine, 2 really great places for information. And we were able to have these really great discussions talking about what was going on that week in the world of COVID, just looking at vaccine hesitancy and how we could combat that. So we really did a lot of work together and really trying to understand different things that were happening in the world. I really loved hearing about things. I learned so much every single week. And by talking with Dr. Hart about all the things that we brought to the table, we were able to have really great facilitated discussions around the information we were sharing, as well as really thinking about why things were happening and how we could share this in a really great way, which Henning will be talking about in a little bit. But Henning, is there anything else that you'd like to add about our journal club?
No, I think you covered it perfectly. So just to introduce myself in advance, my name is Henning Anderson. I'm also a third year in the Medical Molecular Biology program. And I was the person that reported on the CDC's weekly morbidity and mortality report. And I had a very similar experience to Leah. Like, we learned so much, and it was very great to see a lot of information in a concise location and just chat about it for an hour and kind of like combat like the misinformation or anything else that we are hearing, especially as students and full-time students at that in person.
Great, thank you so much for sharing that. You know, this is really the inspiration for this was, you know, something that I've learned over many years being a scientist, and that's, you know, good science is a team sport. And so, you know, it's really bringing together multiple reliable sources of information and going as close to the source of science directly to scientists that really helps to, you know, to get at the truth in science. All right. So moving on. So our journal club discussed, right, the science of SARS-CoV-2, and we looked for inspiration to help us explain science to our friends and family. And one example is that we came up with is this soup analogy. So mRNA vaccines, at least from my perspective, have relatively simple ingredients. There are a little bit of instructions, right, to make the virus, and part of the virus, I should say, that's protected in a little lipid bubble. So because it has relatively simple ingredients, I'm kind of making the analogy that mRNA vaccines are like chicken broth. But there are other types of vaccines, some with similar ingredients and some that have extra ingredients. So, you know, I kind of gave them a little bit of, you know, a similar base, right? They're all chicken soup, but some have vegetables and some have noodles. So some soups taste good to me, just like some vaccines taste good to my immune system. But sometimes our immune systems don't like the taste of certain vaccines, which is where allergies and rare reactions can come in. For some vaccines, may be better or less reactogenic, right? Again, that term that has to do with reacting in some way could be something mild, like, feeling sore in your arm or getting a fever, could be something more serious. And for some people, there might be some vaccines that are going to taste better to their immune system, less likely to have an allergy or a reaction. And for others, it may be a different type of a vaccine that might taste better to their immune system. Another way of looking at this, my husband and my son both happen to be allergic to penicillin, but they're not allergic to all antibiotics. Just because they're allergic to penicillin doesn't mean they don't take another kind of antibiotic when they're sick. So if you have concerns, and I think that's very, very reasonable to have concerns about myocarditis or blood clots. By the way, your risk of myocarditis and blood clots if you get infected with the virus is much, much higher than those risks from the vaccines. But if you do have those kinds of concerns, then maybe a different vaccine would be better for you. Not everyone has the luxury, of course, of choosing which vaccine, though, as we saw in previous slides. We still have a long way to go in terms of vaccine equity. But I will tell you from my own personal experience, back in May, any of the vaccines that I had seen the data on that were approved in the United States, it's called an emergency use authorization, or EUA. I would have taken any of those vaccines that were given to me after, what we had been going through for so long, over the course of 2020 and then the beginning part of this year. Here's a different analogy to help us understand how the immune system works. So antibodies and B cells that make the antibodies are part of our first line of defense against infection. It's normal for the amount of antibodies to go down after vaccination over time. This isn't something new for COVID vaccines. If our bodies cranked out high levels of antibodies for everything we've ever been exposed to or vaccinated against, our bodies would be clogged with those antibodies. We'd be a walking antibody. This is why our B cells are around to keep the memory of those exposures. And then they ramp up the production of antibody quickly when we actually need it. Again, that race analogy. So it's not unexpected or unusual for vaccinated people to get infected with SARS-CoV-2 and even get some mild symptoms. Over time, it will take a little bit longer for those B cells to crank up antibody production. And this gives the virus a little bit more of a chance to make more of itself and make you feel some symptoms. But remember, we are still at a big, big advantage in this race if we are vaccinated. So B cells and antibodies are a bit like the Navy and its torpedoes. They try to get the floating bad guys, the viruses, before they reach land, your cells. When antibody levels go down, a few bad guys might make it into your cells and you might start to feel sick. But those memory B cells will still do a great job keeping the infection in check. Also, we have a whole different branch of our immune system called T cells. T cells are like your army. If the bad guys do land, if they make it past the Navy and they do end up landing or entering your cells, T cells will help destroy those infected cells and lessen the impact of the bad guys, prevent you from hospitalization and death. So far, the vaccine doses that people have gotten are highly, highly effective against hospitalization and death for the great majority of us, against all variants we have come across so far, including Delta. When talking about things that you put in or do to your body, it's important, of course, to think about risk versus benefit and balancing those things. Remember that anything in our environment has risks associated with it. OTC, or over-the-counter medicines like aspirin, have some risks associated with them. Aspirin in small children in certain circumstances can cause a very rare but serious side effect called Reye's syndrome. prescription medicines, of course, if you've read the instructions and the little packaging associated with prescription medicine, often there's lots of different things they warn you about. I love to eat out. I don't like to cook, but eating out has risks. You're trusting someone else to wash their hands after they go to the bathroom and cook your food properly. Foodborne illness is a major cause of illness and even death globally. Unfortunately, there's a lot of things we do to our environment that is not good for our health. We do things that pollute the air we breathe or the water we drink. Clearly, that's not going to stop us from continuing to breathe and drink, right? So again, a good way to frame the discussion of vaccines is to really weigh benefit and risk. Going back to the car analogy, right? There's a very high rate potentially of getting hurt in a car accident when you get into a car, especially here in Boston. Not great drivers in Boston. But most of us, right, still get in a car because the benefit getting to where we need to go outweighs the risk of potentially getting in a car accident and getting injured. Even though my kids are at low risk of dying from SARS-CoV-2, they're not at no risk. And long COVID is a real phenomenon that we're only beginning to understand. So we're going to talk now a little bit about my son's story. People sometimes forget that scientists are moms and dads too. This is a picture of my baby, my son, from a long ago. Isn't he cute? Basically, these days he talks down for me from above because he's 6 feet tall and he has a deep voice. But he was very cute back then. And basically, you know, when I found out that last November that there were very safe vaccines that worked really, really well, I knew that vaccine hesitancy was going to be an issue. So I decided to use my expertise. I have a PhD in microbial genetics from the University of Paris. And I decided to use that to help my friends and family on Facebook try to understand the science so they could make positive, informed decisions regarding their health. So I started to create these Facebook posts that then morphed into these types of Instagram projects that Henny's going to talk about in a moment. And just to show you this one, you know, I basically was sharing that, you know, I was very happy when my little guy was little and I could protect him from polio, measles, or meningitis. Of course, I was less successful at protecting him from spaghetti sauce and yogurt that's plastered all over his face. Fast forward, right? My son's now 18. Here's him playing hockey and scuba diving and fencing. And this is a picture of him getting his first dose of the Pfizer vaccine. And I wanted to share that story again, right? Because stories are important. And it's important for people to hopefully realize, right, that we scientists want to do what's best for all kids, including our own. So to talk a little bit more about this Instagram project, I'm going to pass it back to Henning and Leah.
Awesome, thank you. So in addition to the collaborative journal club, which Leah talked about, where we reported on all these reputable sources, another big part of our work was obviously with the social media awareness regarding vaccines and vaccine hesitancy. So Dr. Hart definitely did a great introduction about her Facebook group, which I'd implore any viewer to join because she pumps up some amazing content and very relevant content of that. However, as Generation Z students, we wanted to bring awareness into different social media platforms, and thus our Dr. Biology Mom Instagram was made. So during the course of the 4-month time span, which Leah mentioned, we worked to convert some of Dr. Hart's Facebook groups into simple and concise infographics, and then we posted them onto Instagram to reach this separate audience and those demographics of those individuals who use Instagram way more than Facebook, such as myself, and I know Leah as well. And obviously these infographics did come out great, if I do say so myself. And we used Canva, which is a graphic design platform, in addition to cartoon caricatures of ourselves, which were made with Bitmoji. And I have a few featured right there of Leah, myself, and Dr. Hart. And they were very, very fun and easy to understand for readers of any age or educational level wanting to learn about COVID-19 or vaccines. So I think it's a very great platform to reach a whole bunch of different people. This Instagram project, along with Dr. Hart's Facebook group, will continue, and we're also looking for different ways to reach other audiences, such as converting our infographics into different languages, which is like a brand new idea that we just started thinking about and talking about how to like implement that going forward. So I definitely drop a follow onto our Instagram using that that's on the screen right there. And maybe if you want to repost something, you can use some of those hashtags that we have there as well.
Thanks, Henning. 69 followers. I know it's not very impressive, but but we've been working hard, and the point for us really, is to learn and to try to help, do what we can. As I mentioned before, the best science is done together in a group. I have a lot of my biology professor friends on Facebook to help make sure that I'm giving accurate information. Towards the end of this talk, I'm going to share a few of my favorite teams, science teams, that I get my information from that I really highly recommend. All right, so I wanted to kind of wrap up this talk talking about something very, very important, which is human behavior. I have some amazing colleagues here in our psychology department. I look to them a lot for advice and knowledge. And I've been learning a lot about some really interesting things that contribute to the really complex issue of vaccine hesitancy. It's very reasonable for people to have concerns and questions. And nobody should ever be shamed to have those kinds of questions and need to get them addressed before they feel comfortable getting vaccinated. But there are a couple of things that can interfere with our ability to make decisions that are good for our health. Confirmation bias is one of those things. It's an aspect of human behavior where we like to look at things that make us feel good, and we don't like to look at things that make us not feel good, which is natural, right? Unfortunately, this is something that social media can take advantage of, right? They have algorithms that when you like something or you watch a certain video, they're collecting data and they feed you more of the things that make you feel good, right? The things that you like or that you stop up on various social media platforms. And I think we have to be a little careful about this. I've been really trying lately to be a little bit more, push myself, to make sure that I'm seeing a broad range of perspectives from reliable people whose credentials I know and can, feel a little bit more trusting of. There's another concept related to human psychology called complexity bias. And this is something that is discussed often in medicine. When doctors are being trained, there's a little saying, when you hear hooves, think horse, not zebra. And basically the idea is if you walk in, if a patient walks into your emergency room in Boston with really serious flu-like symptoms, don't immediately assume they have Ebola, right? We tend to overcomplicate things. Probably why, we do tend to like maybe conspiracy theories, why lab leaks make newspaper headlines, but the much more likely spillover of a virus from animals doesn't make those headlines. Complexity bias is a thing. There's also something called omission bias, and as a tendency we have as human beings. And the way that I like to describe this is that even though doing nothing, right, being passive can be really risky in certain situations, it often feels better for us to be passive than to be active, even though taking action is less risky. Okay, that sounded like a mouthful. Let me explain that in terms of vaccination, right? A lot of times people feel very hesitant, right? Even though not getting vaccinated is more risky than getting vaccinated, right? In terms of your chances of getting very seriously ill, hospitalized, or dying, it's hard to get over that hurdle. Taking action to do something less risky is harder than not taking action when the situation consequences are even riskier. Hopefully that made sense. But, you know, often scientists, we make the mistake that just because we love facts and data and tables and graphs and figures, right? We think if we just give that out to people, you know, they'll decide to do what we want them to do and get vaccinated. But that's basically called the knowledge deficit model. That's not really going to help in a lot of situations to convince people. Vaccine hesitancy has a lot to do with trust. And there are some populations, at least in the United States, that have very valid reasons not to trust medical professionals or scientists because of historical or even current lived experiences. Taking action for our health is often tied up in cultural beliefs, personal values, our sense of identity, with politics sometimes sprinkled in. So it's really important not to judge people, to really listen, to share stories again, and to really be patient as people try to figure out what the right thing to do is for their health. So here are some of my favorite storytellers. Again, what I tend to gravitate to, because I feel like this is where the best science happens, is groups of scientists from respected institutions who have dialogues to get at the truth, to look through data and discuss it, and try to come up with some sort of consensus after not just one preprint, right, is released into the media, but after hopefully you know, several kinds of studies and accumulation of evidence happens. So in the top left is my favorite virology podcast. This is run by Vincent Racaniello, who's a professor of virology at Columbia University, just to the south in New York City. And he has a really great team of really lovely people. And they do a fantastic job explaining some pretty tough science. And they have really awesome guests too, people who have so much insight to share. Again, I like to go as close to the source of science as possible to really get, you know, good science. On the right, this is the University of California, San Francisco Department of Medicine. They have Grand Rounds. This is on YouTube. And they have a really nice mix of doctors and scientists. And they talk about a lot of important topics, again, together, where they can bounce ideas and share opinions with one another. And then lastly, in the lower left corner, again, one of my really favorite sources, storytellers, is the American Public Health Association, or APHA. They've been doing this series called the COVID-19 Conversations with the United States National Academy of Medicine. And they've been really wonderful at exploring, again, some of these really crucial issues, not just the science and the medicine, but public health, global equity, and really trying to help with health disparities, which exist here in the United States and obviously all over the world. I have a couple of recommended stories. So if you're interested in trying to understand a little bit more about vaccine hesitancy, there are two different books that I highly recommend. They're really good reads. One is The Panic Virus by Seth Nunkin, and the other is Stuck by Heidi Larson. She's a professor of epidemiology at the London School of Hygiene and Tropical Medicine and one of my heroes. If you want to know a little bit more about the spillover of microbes into humans, here are two really fantastic, well-researched sources. One is actually released a long time ago, but still very, very interesting and helpful to read today, and that's The Coming Plague by Laurie Garrett. And then David Quammen's Spillover is also a really, really good read. All right, so with that, I will stop screen sharing so I can see people's lovely faces. And yeah, I guess if there's any questions, we're happy to do our best to try to answer them.
Well, thank you very much, all of you, for a great insight into this. I think it's something that we've all been battling with a little bit over, you know, the corona period, but it's nice that you've put so much effort into kind of researching it. And I guess one of the questions I have is, in all your research, have you found a good way of kind of combining the, you know, the scientific literature, you know, the way vaccines work with the analogies, and then also with these psychological biases? Have you found any kind of formula or sequence or way of kind of combining those to actually have a good productive conversation with people who are hesitant about vaccines?
Yeah, that's a great question. I think it's a process that we're constantly learning. You know, my training is in bacterial proteomics, but, you know, being a mom again, you know, I love to share science with my kids, and I have 2 kids here. Sorry, young adults, I should say. I tend to think of my students as my kids. My kids are my genetically related kids, are also young adults, but you know, but basically really trying to learn how to, you know, to be a good communicator. You know, that takes time. As you mentioned, integrating an understanding of psychology. And I mean, it's really fascinating to explore the intersection of public health and genetics and microbiology and psychology. I'm a big believer that there's always something to learn from other disciplines. I've done some collaborations with professors in the field of ethics. And yeah, I think having an open mind and trying to learn as much as you can from different disciplines, and then just, you know, doing your best to really, you know, work to integrate those things together in discussion, right, with students or colleagues.
Yeah.
Do you guys want to add anything earlier? Do you want to add anything else to that?
I think that by having the conversations and being in programs like Henning and I are in, we have a really great understanding of how it works at a small level. And so being able to kind of build on that, and by looking at the science from reputable sources, and then also having conversations with professionals who are in the field, I think that really helps, because you, like for me, for example, like I've studied biology for the last 4 or 5 years in high school and now in college. I feel like I know a lot, but there's so many other people that know a lot more than I do. And so I learn from them. And by hearing the different analogies and the stories that Dr. Hart has shared, I think that it kind of puts the pieces together so that you can have a story too, and kind of understand how it works. And so I've personally had a lot of conversations with people close to me about vaccines and how important they are and how safe they are, because I've been able to have conversations with Henning and Dr. Hart in our journal group. We discuss so much. And I feel like in those situations and circumstances, it really helps you to make a formulated decision and helping share the wealth of information in a productive way.
And this is honestly, you know, my goal is not to become social media famous at all. I'm a very private person. You know, I just want to help, you know, and obviously it started with just my friends and my family, really trying to reach out to them. But, you know, I also really want to help my students become, you know, better ambassadors for science and health. And, you know, and so this has been a really rewarding project to be able to help inspire young people, right, to really know where to go for accurate information, how to put it all together, and how to communicate it to keep their friends and family safe.
Yeah, so kind of just piggybacking off both of those, I'm probably one of those young people who would most likely benefit from that. At this point in my life, I have an attention span of like a goldfish. So using like the infographics that we've developed, I think is definitely like the best way that I absorb information. And even though we're only 69 followers strong, as Dr. Hart pointed out, which hopefully will change after this, little shout out to the audience. But like just reposting those and allowing my friends to see them as well definitely gets the information out, because the amount of time that I would spend on social media compared to reading the morbidity and mortality weekly report is astronomically different. And seeing that accurate information all in one place while I'm just scrolling through my feed, I think is very beneficial.
and we try to be really, really careful, again, because we're using a lot of different sources. We're trying to be really careful to not share anything, that's, or to say when we're not certain, right? To be honest and transparent when we're not sure about something, when we don't have enough evidence yet. And when we do, we get excited about that and we want to share that too.
Great. Yeah, I hope we get some more followers on there after this. Yeah, definitely reach out to us on that. You did mention Twitter. I'm on Twitter at the moment. I could give you a shout out, but on Facebook and Instagram, maybe we can do it later on.
Yeah, we were going to try Twitter, but yeah, I mean, last year for all of us, right? It was a rough year work and study wise for students and teachers all around the world. So, Instagram was about as much as we could manage. Yeah. I'm a little scared of Twitter, to be honest, but I'll try to keep an open mind. Actually, we might do TikTok. Actually, we're cooking up some ideas with TikTok.
Okay, TikTok.
Don't tell my genetically related kids. The first time I tried TikTok and posted a video, I was lip syncing to Lizzo, and they were so embarrassed.
That's what TikTok's for, I think, isn't it? I mean, it's for.
Exactly.
We're embarrassed too.
Yeah, everyone's embarrassed on TikTok and they don't care. I think that's the beauty of it. Okay, let's just agree to meet on TikTok then, and we'll embarrass each other there. I have another question for you about your background and when you're talking about this kind of stuff, because at the beginning, kind of noticed you were careful to say that you're not affiliated with any companies or anything. So. Does your background have a positive or negative effect when you're talking to people? Do people say, well, of course you're going to be pro-vaccine because of where you come from? You're probably like a pharma company shill, and you kind of you're taking does it have a positive or a negative effect when you're talking to people?
Yeah, I mean, I think it is really important to disclose, whether you do have that kind of affiliation. It doesn't negate what you're saying, but it's important to be really honest and upfront with people. For me, I always try to approach it. I mean, it's important for people to have some expertise, right? I mean, I can tell you my PhD is in microbial genetics is super helpful during the past, year and a half, as I've tried to navigate a lot of things that often, even to me, are very confusing. And so figuring out, right, again, who was I going to listen to and how I was going to try to, bring together that information. We talk a lot in public health about the importance of trusted messengers. And so again, there are a lot of people on social media, some very well-meaning, but often it can be about egos, right? And that's again why I like groups of people, groups of scientists that talk together. I mean, for me, I think I'm really good. My friends, my family, the people who know me on social media, I'm probably a good person to reach out to them. They trust me, right? We have a relationship. But for Leah's friends and family or Henning's friends and family, right? They're the trusted messenger, right? They're the people who are really going to be able to, to help people understand as they're developing their expertise, right? And as a teacher, I, want to help them to know how to plug into those sources. So does that answer your question or?
Yeah. I think that's really valid. I think I've seen it as well. You need, whenever you're communicating about things like this, especially with someone who you think or is likely to disagree with you fundamentally, then you have to definitely have that element of trust to start the conversation, which is even more difficult to develop online, right? Do you have any tips or advice for how to be a trusted messenger online?
Oh, yeah. Honestly, I agonized for a little while before I started, I created the Biology Mom group on Facebook last November. I agonized. I'm like, do I really want to add my voice to all the craziness that's out there on social media? And then I realized, I have this expertise. I care. I want to make the world a better place. I want to end this as fast as I can, as we all, pitch together. And also, if I didn't speak up, then I'm letting people, and there are people, a lot of people out there actually, who are purposefully putting out misinformation because they profit from that. And so, if I don't say anything, I'm letting those people dominate the conversation. So yeah, I think it's really hard on social media, and each type of social media platform has its own flavor, right? To go back to the kind of soup analogy. Again, I just hear scary things about Twitter. I don't know why. It seems like a scary place. I think on social media, as much as possible, if you can plug into sources where you can have a back and forth, like a dialogue, I think that could be helpful. If you plug into sources, again, that have, they're clear about what their affiliations are, what their expertise is. I'm very wary of websites or social media where you don't know who's writing it. You don't know who's representing it. Be a little wary of egos, I would say too, on social media. Again, there's some very well-meaning people, and I watch some individuals on YouTube that do science education or health education, but every once in a while I'm like, oh, they didn't get it right, because they're by themselves. Whereas in a group, you're much more likely to be able to work through your thinking and have misconceptions corrected, if that makes sense.
Yeah, it does. I think it makes a lot of sense. I think, it's good advice for everybody, but maybe that's also a way of kind of ending conversation with people when you don't agree. You say, look, this is the way we look for information. Like when you're looking at your own information, make sure that you do the same as I'm doing. Make sure you look for groups of people who are having discussions. Make sure you watch out for egos. Make sure so that you actually encourage people to find out for themselves and kind of do the proper research, because it feels like that can be missing when you have the misinformation.
Yeah, so much has been sensationalized, and there's some amazing journalists, and I really, really have a lot of respect for that field. I don't think it's an easy field at all. But at the same time, that's not where I want to get my scientific information. I want to get it from scientists. And for me, education is the key to everything. I grew up during the HIV epidemic in San Francisco, my hometown. And, part of the reason I became a biologist is because of that. And I very quickly realized that actually, science is great. I love it. But for that pandemic as well, education is really the key. So that's where I'm really passionate and try to help, again, empower my students to really become also, engaged health educators, science educators, ambassadors.
Great. Education is the key. I think that's an excellent place to wrap up. And I'd just like to say, thank you very much to you, Janet. Thank you, Henning and Leah. It's been an absolute pleasure. We'll move on to the next session, but I'll definitely look out for you on social media, and we should definitely meet up on TikTok when we get our accounts started, and that's going to be fun. So thank you very much for today, and
thank you for inviting us.
Thank you so much.
No problem. Bye now. Bye. Okay, fantastic. Very, very interesting discussion. As I was saying, I think it's something we've all been battling with over these year and a half, 2 years of COVID, about talking about vaccines and vaccine hesitancy. Now we are moving on to our next session, Microbial Arts and Crafts workshop. And so I'd like to hand over now to our next hosts for this hour-long session. We have Elizabeth and Angelica here with us today. Hello, welcome back, Elizabeth. Hi, Angelica. How are you both doing?
Hi, everything is fine. Thank you.
Good.
Yeah, I'm good. Thanks for asking.
So I hear you've got some very exciting things for us planned. So I'm just going to kind of disappear into the background and kind of, watch along. And if you need me for anything, just shout, I'll be here, but I'll leave the floor to you.
Okay, thank you. So hello, everybody. As Ben just said, my name is Elizabeth or Testiet. I am the founder of Microbigals and the co-host of the Micro Moment. And today we have an arts and crafts workshop, which is going to have 3 different little workshops in it. But first, I'll let my co-host Angelica introduce herself.
Hi. Well, first of all, I hope you are enjoying this International Microorganism Day and the program that TEMS put together. And we both want to welcome you to this microbial arts and crafts session, which is going to be really cool. What we're going to show you is the result of a joint collaboration between 7 women involved in science communication. We are from different places around the world. So it was fun to put this together.
I think we're in like 3 or 4 different time zones and maybe 4 or 5 different countries.
Yeah, so it was hard.
It was hard, but it was a challenge and it was a lot of fun to kind of get to know everybody from around the world.
Yeah, definitely. It was lots of fun. So my name is Angelica Jara Servín. I am a PhD student at the National Autonomous University of Mexico. And you can find my work in social media profiles under the name of Pop Microbiology. And well, just to let you know who else participated in this session is Charlotte Ruffton. She's a PhD student from Newcastle University in UK. And she's the owner of a project called Stitching Science that you should definitely check out. Christina Campbell, who is a freelance science communicator and a medical writer, was also involved in this. And she prepared an interesting video for us. And we have more people. Tess, would you like to tell us?
Yeah, so a big portion of what we're doing today is a BioCrust illustration and sort of info session. And so the illustration comes to us from 3 science artists. We have Eliza Wolfson, who is a freelance scientific illustrator. We have Nomi Matthew, who's at the University of Lucerne in Switzerland, and she has her own science artist business, as does Lisa Vanda Art, who is another freelance science artist. So during the video, we will have everyone's Twitter, everyone's everywhere you can find people. So if you like their artwork or anything, most of them also sell different products. So you can go check out their stuff.
Yes, exactly. So let's begin. And we're going to start with our first video recorded by Charlotte. She's going to teach us how to cross-stitch a very interesting bacteria. And while she's cross-stitching, she's going to explain us why these bacteria are important. So let's check it out.
We don't hear the audio on there. When you click the share button, did you select the little button in the bottom that's share computer audio?
Okay, let's try again.
Yeah.
There, I'll be telling you.
Good now?
It works.
Yeah. Okay, perfect. Yeah. Thank you.
Hi there, my name is Charlotte and I'm a microbiology PhD student at Newcastle University in the UK. Now, I absolutely love my research, which focuses on the superbug Clostridioides difficile, or C. diff. But when I'm not in the lab, I really enjoy communicating science. One of my favourite ways of doing that is through art. My favourite medium is cross-stitch, and I really enjoy making and stitching designs I communicate the science I'm passionate about in both a fun and a beautiful way. I run a small business on Etsy, Stitching Science, where I sell cross-stitch patterns for you to follow and create your own science goodies, as well as completed designs. So this International Microorganism Day, I'll be telling you a little bit about C. diff, while showing you how you can cross-stitch one for yourself. Let's get started. You're going to want some fabric to stitch your design onto. There's lots of different fabrics available that are suitable for cross-stitching, each with different textures, fibre contents and colours. My personal preference is Aida, which is the most widely used for cross-stitching. It has a wide, open weave with easy holes that help make your cross-stitches neat and consistent. Aida comes in a number of fabric counts, defined by the number of holes and therefore stitches per inch. I like to use 14 count Aida, which means for each inch of fabric there will be 14 stitches. However, you may opt for a smaller fabric count if you prefer that your stitches are larger, or vice versa. Aida is readily available in a number of different fabric counts and colours, both in local craft stores and online. The next thing you'll need is a needle. For cross-stitching, I like to use a tapestry needle, which is blunt. One, because you generally part the threads of the fabric rather than pierce the materials you stitch. And two, because I'm incredibly clumsy and may have spilled blood on my projects before. I really am being literal when I say blood, sweat and tears go in my work. Next up is your threads. These come in a number of different brands which have different systems of numerically categorising their thread colours. I personally use threads from the brand DNC, which are the most accessible to me in the UK. So the thread numbers provided correspond to the exact DNC colours used. However, you can just gauge it by eye and use whatever you can get your hands on. Feel free to change up the colours too, there's no strict rules here. You'll also need some scissors to trim your thread with. I like to use embroidery scissors, which are really sharp and mean you can trim your threads without them fraying. And finally, this is somewhat optional, but I recommend a hoop. Hoops help to keep your fabric taut while you stitch, and are also a way of displaying your work when it's finished. However, Aida is starched, so it's fairly stiff as far as fabrics go, meaning you could get away with stitching without using a hoop. There are also alternative ways of displaying your work too, such as using photo frames. My preference though is to use a hoop, so that will be used in my tutorial, but do what works best for you. This piece frames nicely in a 3-inch hoop. Now that the materials are out of the way, let's crack on with the actual stitching. The pattern we'll be following is shown here. Feel free to take a screenshot, but a high quality PDF will also be made available for you to download. In this pattern, each stitch is represented by a symbol, with each symbol corresponding to a shade of green. It's up to you where you decide to start. Some stitches like to start in the centre, which is indicated by the intersection of the red lines, though I usually start at an edge and work my way in. I'm going to begin with the body of the cells, represented by the fan symbol. I like to take 2 strands of thread, fold these over, and then thread the needle. This means that each stitch will consist of 4 strands in total, which I prefer for a fuller effect, but you can opt for different strand numbers depending on the finish you prefer. The more strands you use, the less of the Aida colour you can see peeking through. I think 4 strands is a nice compromise between getting a nice full colour while avoiding the appearance of overcrowding. Once your needle is threaded, you want to make a start on your first stitch like so. Push the needle through the holes of the Aida at your starting point and then push back through the diagonal hole. Remember I told you to fold the threads up before threading your needle. Now, pull the needle and thread through the loop created to secure your thread. Next, push the needle back through the holes on the opposite side and create another diagonal stitch. And ta-da! That's your first cross stitch. Now you just need to follow along with the pattern, with each symbol corresponding to 1 cross stitch. You may want to print the pattern out and cross the symbols out with pencil as you complete them to help you keep track of your progress. This pattern also includes some thread needle through the corner of the cross. Bring it over in the centre. Using a sharper needle can help with this. And then bring it back down through the corner. Then cross the thread diagonally. This creates a triangular 3/4 stitch shape rather than the square full stitch. When you reach the end of your thread, to ensure your hard work doesn't unravel, slide your needle underneath the back of a few of your stitches and pull the thread through. Snip the excess and you're ready to start with your next thread.
As I continue to stitch, let me tell you some more about C. diff and why we should care about it. C. diff is a bacterial pathogen, meaning it causes disease. In this case, C. diff is an infection, or CDI. CDI usually occurs when a healthy population of gut microbes has been compromised in some way, for example, if taking antibiotics. While antibiotics are fantastic for treating bacterial infections, they're not specific to the bad guys. Unfortunately, our beneficial gut microbes are also killed by the drugs we take to cure infections. This is a problem, as it leaves an ideal home for less desirable microbes to move in, such as C. diff. Without a robust population of gut bacteria on hand to outcompete C. diff and fend it off, it moves in and makes itself comfortable. It starts to replicate and produces toxins which damage the gut and cause the symptoms of CDI, which include diarrhea and even holes in the gut and severe gases. Unfortunately, the current treatments available to tackle CDI are more antibiotics, which comes with caveats. By continuing to use antibiotics, more good gut bacteria continue to also be killed off as a result, creating a vicious cycle that maintains an ideal environment for C. diff to just come back once the antibiotics are stopped. In fact, around 20% of infections recur in the first instance, and this chance of recurrence increases with each subsequent infection. So it's crucial that we find new ways of tackling this bug, methods that don't also target the gut bacteria we want to keep around. Another characteristic of C. diff that makes it so tricky to deal with is its ability to form spores. Spores are dormant forms of C. diff. Think Sleeping Beauty, except when this beauty wakes up, you're in for diarrhea and a pretty bad time. Though spores don't do much, in this form they have several protective layers and are incredibly resistant to common disinfectants, heat and radiation. This allows C. diff to hang around in the environment and can spread to others if they, for example, touch a surface containing spores and then their mouths. If this happens to a person who doesn't have a healthy population of gut microbes, then the spores can wake up in the gut and then cause an infection. However, despite the important role spores play in the transmission of CDI, the mechanisms of spore formation are not yet fully understood, nor do current treatment options target this feature. Researchers like myself are working towards better understanding this process, and maybe in the future, this information could be used to develop new drugs that can target C. diff specific features, preventing it from returning and spreading to others, but without hurting the good bacteria. It's a super exciting avenue of research, I hope you agree. And now, back to the cross-stitching. Now that the bodies of the cells are finished, it's time to outline them. To do this, a back stitch will be used. For this, I like to use 2 strands, so this time take 1 strand of thread, fold over and thread your needle as you already have done. You may want to use a sharper needle for this step, as back stitching can sometimes require you to pierce the ader. Start wherever you'd like along the outline of the cells and stab the needle through the fabric. Pull it back through about a stitch length away and make sure to thread the needle through the loop created to ensure it doesn't unravel. Next, push the needle back through the fabric a stitch length away and then take it down to the end of the previous stitch and pull through. Then repeat until all the cells have been outlined. After you finish the back stitching, the piece is complete. Yay! To tidy up the back, you can just trim the excess ader, or to minimise fraying, you can run some stitches around the outside of the excess ader, then pull the fabric together like a drawstring bag. If you decide to follow this tutorial, I hope you enjoyed learning how to stitch this deceptively cute design. If not, I hope you enjoyed watching nonetheless and learned something new. You can find me on social media at CGRoughton on Twitter or at Stitching Science on Instagram. Feel free to come say hello, and if you decide to have a go with this pattern, let me know how you get on. I hope you enjoy the rest of International Microorganism Day.
Okay, so we hope you enjoyed Charlotte's video and you're willing to take a chance and try to cross stitch your own Clostridium difficile at home. As you saw, Charlotte used different shades of green to create her pattern. And believe it or not, those different shades of green could easily come from pigments obtained from microorganisms. So that's what we're going to talk a little bit about. I'm going to share my screen. And we should just also mention that Charlotte has the, what is it called? She has a PDF of the pattern if you're interested in it as well. And we will share that around social media for anyone who's interested. Yes. Okay, so I don't know if you have ever had the chance to appreciate microorganisms growing in a petri dish. If not, here you can see one. We have different microorganisms and we see them showing a wide variety of colors. We have yellow, turquoise, and some sort of orange, red. And someone along the way decided that extracting these pigments may be a good idea and we could give them a use in the industry. And that's something that happens nowadays. For example, there's a red pigment that we obtain from bacteria called Vibrio. These bacteria are usually found in environments with high salinity, so you can find them in the ocean. And the pigment that they produce that is red is called prodigiocin. This prodigiocin is extracted from the bacteria, and then it is used to dye different fabrics, such as wool, nylon, acrylics, or silk. And depending on the fabric, the shade of red that you can obtain is different. For example, silk, as you can see here, is darker than the color you obtain in cotton, which is more pink. Another color that we can obtain is orange. The orange pigment is obtained mainly from a bacteria called Serratia marcescens. This bacteria grows like this in the lab. You can see it at the bottom right of the screen. And actually, this bacteria you may have seen because it grows in damp spaces. So if you ever see a pink film on your bathroom, that's probably Serratia growing there. And the procedure is the same. You have to extract the pigment from Serratia, and then we use them to dye acrylic, polyester, silk, but mainly cotton. And the other pigment that I want to talk to you about is called anthraquinone. And it is obtained from a fungus that is called Fusarium oxysporum, and it's the one that is growing in the petri dish that you can also see in the screen. This compound is actually transparent, but when you add different chemicals, it can turn into different shades of color that go from pink to purple. And this compound is used mainly to dye wool. So who knows, maybe the clothing that you are wearing right now was dyed with a microbial compound. So if you're curious enough, you can check it out. And now we are going to pass to our second video, and Tess is going to explain us a little bit about that.
Yeah, so we're moving right along into our biocrust illustration. So this is it's a live illustration that Eliza, Naomi, and Lisa collaborated on together, and it's sped up really fast so that we can see it in real time. So we asked them if they could draw a biocrust formation, and they did a really fabulous job. This is a super dynamic, really interactive illustration, I think, and it's a lot of fun to watch. So while we're watching this, we'll kind of go over some stuff about biocrusts and what they are and their importance in our society. So first off, biocrusts are sometimes called the living skin of the earth. They are particularly found in dry lands, but they can occur all over the world. They do wonders at stabilizing soils. This is one of the main things that they do for our soils, but we'll get into some other points as well. And they are mainly photosynthetic communities. They're made up of cyanobacteria, microfungi, bacteria, lichens, and mosses. So cyanobacteria, algae, lichens, mosses, and liverworts are photoautotrophic, meaning they produce energy on their own from light. So as I mentioned, biocrusts can be found all over the world, including hot and cold deserts, as well as alpine regions and polar regions. They survive harsh conditions many other organisms cannot survive in. They form these beautiful little tiny forests that I almost imagine little fairies fluttering through and calling home. So the biocrust formation is influenced by a lot of things: climate, soil, water availability, the geographical location. So each one is very unique and hosts their own sort of microbial communities. The biocrust can be very different, even if they are in the same location. I read this one paper that looked at 4 different sites within the Mojave Desert, and they found that each one had a distinct microbiome compared to the others. And biocrusts cover, if you're wondering, biocrusts cover about 12% of the global terrestrial land surface and about 30% of the global dry lands. Some biocrusts can be as thick as 10 centimeters, but many of them are only a few millimeters in thickness. So Angelica is now going to talk to us about one of the main microbes we find in our biocrusts.
Okay, so first of all, we're going to make a zoom to the soil that we are seeing here being drawn. And the first thing that appears in a biocrust is a bacteria called cyanobacteria. And those cyanobacteria are these long and green colored bacteria that you can see right now in the screen. And cyanobacteria are a very special bacteria because they can carry out photosynthesis to obtain energy. This is a process that is known for plants because plants do that to obtain energy, but cyanobacteria carry photosynthesis out also, which is pretty amazing. The most common cyanobacteria that you can find in biocrust are called microcoleus and nostoc. These are the ones that appear and start to develop and grow. And the main objective that they have is to synthesize on the inside polysaccharides and then excrete them to the environment. And these polysaccharides that are being colored in pink are the ones that start to glue all the soil particles together. So you begin to have clumps or soil and bacteria. And this allows for different microorganisms to gather and the biocrust starts to grow.
So we're going to talk a little bit about another microorganism called lichen. So lichen is a symbiosis in the microbial world that happens between a fungi and an algae or a cyanobacteria. So they live together and form a symbiotic relationship, meaning that they sort of help each other out in different ways. So some lichens can grow as big as 3 feet tall, and other lichens can be thousands of years old. In fact, there's one in the Arctic that's called Rhizocarpon geographicum, which is thought to be 8,600 years old. So this is a pretty old organism. Lichens are not only found in biocrusts, but they cover 6% of the world. And chances are you have them right outside your door. If you've ever seen rocks or trees with strange crusty greens or yellows or oranges that kind of look like paint splotches, chances are this is a lichen. And from afar, they don't look like much, but if you take a magnifying glass up to them, you'll see that there is an intricate world in that little splotch of fake paint. So, however, lichens are very fragile. With just a little bit increase in pollution, they'll leave an environment. So if you live in a city that happens to be very polluted, you may not find any lichens in that environment. Lichens can be beautifully diverse, as we're seeing in this illustration here. The algae or cyanobacteria is the portion of the symbiosis that creates the that does the photosynthesizing, creating the energy for the organism. And the fungus kind of forms the structure. So that's a lot of the elements that you're actually seeing. And then I just want to talk a little bit about the different kinds of biocrusts that we have, because they can come in different kinds depending on what is the dominant organism. So if cyanobacterium is the dominant organism in the biocrust, we call this cyanobacterial biocrust. And we also have moss biocrust and lichen crust. So it all sort of depends on what you see in part of the biocrust. And then finally, I just want to mention a couple lichens that you can find in biocrusts, which include Soria, Placidium, Colema, and Fulgencia. So, Angelica, why are biocrusts important?
Yeah, well, before telling you why they are important, I want to tell you about some other organisms that you can find in biocrusts. Like we said at the beginning, biocrusts are a mixture of different organisms. So cyanobacteria and lichens are not the only ones that inhabit these biocrusts. You also find different types of bacteria. The ones that are found in biggest proportions are actinobacteria, chloroplexi, and proteobacteria. And all of these are really important in the biocrust formation because they produce different compounds such as vitamins or antibiotics that help the biocrust grow. And also they allow the biocrust to defend itself from different pathogens that may arrive from the surrounding soil in the desert or in any other ecosystem. Besides bacteria, cyanobacteria, and lichens, you can find also mosses. Mosses are those small flowerless plants that you find in damp spaces. For example, if you go to the forest, you can find mosses growing on the surface of the trees near a river because the environment needs to be damp. And they also grow in biocrusts. And all this mixture of microorganisms are crucial for the ecosystems such as deserts because they play a lot of different roles. They contribute to the carbon pool. And well, as we all know, carbon is essential for any living organism to grow. And specific cyanobacteria like nostoc and lichens as Colema, they contribute with nitrogen fixation. Nitrogen is one of the compounds that is scarce on deserts. So the more we have, the better. And the fact that we have an increase in the carbon and nitrogen pool allows different plants to grow. So it's normal to see plants growing near biocrusts in the deserts. And while biocrust also stabilize soils and prevent particles from blowing away when there is a dust storm. And this at the same time prevents soil erosion. And finally, biocrusts also help to retain moisture in the desert. And as we all know, sometimes water is difficult to find in these environments. So that's why biocrusts are really important and we need to be more aware of their role in nature. So as you see, they are beautiful. Here they are being drawn. So now we're going to talk about which are the threats that biocrusts have. You are muted, Tess.
Can you hear me now?
Yes, but now we can't see the video.
Yeah. There we go. Okay. Little hiccup. No worries. Yeah, so biocrusts are extremely fragile. We talked a little bit about how fragile lichens can be, which is a huge component of these biocrusts. But look at how beautiful they are. And it doesn't take much to destroy all of this beauty. And one way that this can happen is through trampling or grazing of wildlife. Here we have a picture of a dog that's coming in and is about to destroy the biocrust, which is why it's so important when you take your dogs to national parks and they say a leash law, keep your dogs on leashes to help protect the biocrust. But this is not just dogs, it can be coyotes, it can be other wildlife as well. Sheep are also another big one, or cows as well. And why this is so devastating to these ecosystems is because it can take 30 to 40 years for a biocrust to come together and maybe even longer for it to come back to its full functionality. But it's destroyed in an instant. Trampled biocrust can increase soil erosion. It also impacts the carbon and nitrogen concentrations in the soil, which can have compounding effects throughout the ecosystem. Because they are so slow growing, they are very fragile, and it's hard to research as well. And it also makes it hard to show their importance with the world. Another thing that ends up dismantling a biocrust is humans, and our own footprints that are trampling on these biocrusts can cause a lot of devastation. And human impact is projected to decrease biocrust between 25 and 40% in the next 65 years. So it is an ecosystem that we definitely need to start protecting.
What you see that is appearing now on the screen is an invasive grass. These grasses are also one of the major threats that biocrusts need to face because they tend to grow a lot to take up all the nutrients that you can find on the soil, and they grow until they occupy an incredibly amount of the surface that is available. And another thing that is really dangerous about these grasses is that they can act as fuel and start fires like the one that is happening right now. And these wildfires, like any other fire, can spread a lot and eventually kill all the microorganisms in the biocrust that took so long and made a lot of effort to grow until they become a healthy and big biocrust. The problem with these grasses is that they are usually not original from the desert. Usually it's us humans that take a grass that belongs to a different ecosystem and we take them to a place where they don't belong to. So they end up wiping out the plants that are native for this ecosystem. So it's something that we really need to care about also. We must not take seeds from different plants to our country when we come back from vacation, because the chances are that you may end up killing a lot of wildlife and biocrusts, like in this case. You're muted again and the video is gone. No, I can't hear you yet. Nope. No, I can't hear you. No, yeah.
Can you hear me now?
Yes. Just
Yes,
the video is missing. Yeah.
Yeah, I'll share it back. Cool. Thank you for bearing with us in all of this. Yeah, so we don't want to end this on a super downer, and we want to talk a little bit about the resiliency of biology. So even when trampled and burned and disrupted by shovels, biology fights on. And these biocrusts, although fragile, are resilient. I read this one study that looked at the long-term effects of trampling on biocrusts. And this is a study that's called Response and Resilience of Soil Biocrust Bacterial Communities to Chronic Physical Disturbance in Arid Shrublands, which was published in ISME by Cheryl R. Kuski. And they looked at the desert sites in the Colorado Plateau in the United States of America. And particularly, they looked at sites that had experienced foot trampling for 10 or more years. Their results showed there was definitely a huge change in the ecosystems. They could show you that these biocrusts were not at their full potential. They could show you that there was less DNA in trampled biocrusts, and they could show you that there was less chlorophyll A, which is an indicator of photosynthesis. But they did find something that I think is a little hopeful in all of our biocrust destructions. Despite the traumatic destructions they found, there was still life in these biocrusts. And they were even able to find a keystone species in the biocrust known as Microorganism vaginitis in the trampled soils. So this means given enough time, the ecosystems have the ability to restore themselves. They are resilient and capable of forming over time if we let them.
Yes, exactly. And that's why scientists have put a lot of effort on studying how to restore and maintain these biocrusts. What you are seeing now is a trowel or a small shovel, and it probably belongs to a scientist that is arriving to the biocrust to take a sample, to dig a sample, to study a little bit more about these biocrusts. Scientists have found two different approaches to help restore biocrusts once they have been damaged. The first approach consists on taking the top 0.5 centimeters of the biocrust and then crumbling the sample into pea-sized fragments. And these fragments are then taken to the damaged biocrust and used as an inoculum. And they put the pea-sized fragments on the biocrust and just let time go by, so the microorganisms have enough time to develop, to thrive, and to interact with the microorganisms that are left in the damaged biocrust. This sounds amazing, but there is a downside, because for this to happen, you need to damage a healthy biocrust. So it's not win-win. You are damaging a biocrust that was already healthy in order to save another one. This is why a different approach has been taken in the last years. And it consists basically on growing cyanobacteria, especially Microcolius and Nostoc, the first cyanobacteria that appear in a biocrust formation. Scientists take these cyanobacteria and start to grow them under control conditions with the right nutrients, the right temperature, and the right pressure, atmosphere, anything they need. And they grow them, they wait for them to develop enough so they have a lot of biomass. And once they have a considerable amount, they can take these laboratory grown cyanobacteria and take them to the field, use them as an inoculum, and just the same, wait until these cyanobacteria grow and help the biocrust to recover and to be healthy again. The problem with this is that you need to culture these cyanobacteria in the lab for 4 to 5 months, and you need to know at least the basics in microbiology. So we still need to do some more research.
Yeah, the last 2 decades have really been the time that scientists and researchers have dived into these biocrusts. So it's really a kind of new and exciting field to be in. And this last little piece of the video is showing acid rain and climate change having an impact on these biocrusts. So even if we start to restore these biocrusts on the land, our impacts on the climate may be destroying them anyways. And so it's just such a multifaceted problem that we really have to think about and come up with solutions to protect these very fragile yet beautiful ecosystems.
Yeah, that's right. So if you ever run into a biocrust, please treat it nicely. Don't step on it or anything like that, because they are really important for the environment. And if you like the drawing that you saw, as we said at the beginning, you can contact or follow in social media the artists that made it. On the right part of the screen, you can see the names: Eliza Wolfson, Naomi Matti, and Lisa Vanderart, with their corresponding Twitter accounts. So we hope you enjoyed watching this video, and now we're going to pass to another section about pigments, because we are really into art right now. Yeah, poor bacteria. I think you can almost hear them screaming with all the acid rain.
Yeah. And if you like this video, I believe it is on Twitter. It's on a couple other social medias too. So just go ahead and find them and you'll be able to watch this again. But they had a lot of fun making it, and we had a lot of fun kind of having this moment to share BioCross with you. And yeah, we hope you enjoyed it. So I'm going to switch gears a little bit as this wraps up, and we're going to talk about the rainbow diversity of microbes, which I think has been touched upon a little bit today, throughout the day, actually. So let me just get that set up for everybody. Okay, so this is color by microbe. So we're just going to talk a little bit about the diversity of colors that microbes have when we plate them on Petri dishes. So when I say microbe, what color do you see? Some people might say they're invisible. They don't have any colors. Others might say, oh, they're that green speck on my moldy bread that I didn't eat fast enough, or that pond scum that you see in marshlands. If you have ever experienced microbiology and done any plating, you'll probably say white or some sort of shade of white, because a lot of them are a white or a cream color. But when I think of microbes, I think of this wonderful diversity in all the different colors, and even colors that we can't even see, which I just think is incredible. There's like just a limitless amount of ways that microbes can surprise us. So in Color by Microbe today, we're going to talk about the microbes that can create the different colors of the rainbow. So red, as Angelica mentioned before, is Serratia marcescens. So when we're doing fabrics, you might come up with orange, but when you plate it on a Petri dish, you have this really rich, almost blood-like red. Orange belongs to Actinoplanes descendens. So this is like almost a Nickelodeon orange is what it reminds me of. It's very vibrant and very full of life. Yellow, we have Micrococcus that comes up with this very yellow color. It's almost mustardy in its color. And a lot of them almost don't look real. They look like it is just a piece of paint or a little piece of paint that got dribbled on a Petri dish. Green is, as many people see, is a common fungal color. Here we have Aspergillus parasiticus, showing a very prominent, almost forest green color. And then for blue, we can use Streptomyces collicolor as our royal blue color. And then finally, we have violet, or our purple color. And this is almost like a metallic-y royal purple. I've seen it in a couple different shades of purple as well, but this comes from Githanobacterium lividum. So that's all the ways that microbes, you can kind of paint with microbes on a Petri dish. And I think earlier in the International Microorganism Day, I don't remember exactly which hour, but they did have a Petri dish drawing example. So that was really fun to see. And I encourage, if you missed it, to go back and see the recording of that. And then finally, there's glowing microbes. And this is Vibrio fischeri, which are common symbionts of the bobtail squid. And they form the bioluminescence that these squids use in the ocean. So very, very clever are our microbial friends. And then lastly, I just want to leave you with this. If you are never heard of auger art, I strongly encourage you to go jump on social media. Instagram is great for this, #auger art. And you'll see so many amazing artists out there that are painting with microbes on Petri dishes and making these really fabulous works of art. And there's always competitions all the time on auger art as well. So if it's something you're interested in and want to get into, there are a lot of ways to sort of challenge yourself and compete in this very niche artistic style. And so that is our end of that, but we have one more little workshop for you.
Yes, and this is a video, our third video, about an activity 100% related to microorganisms. That's fermentation. This is the video that Christina recorded, and please grab a piece of paper so you can write down all the ingredients that you're gonna need.
Campbell, science and medical writer and author of the Well-Fed Microbiome Cookbook. And here's my assistant, Cece. And today, in celebration of International Microbe Day, we are going to demonstrate how to make fermented dilly carrots. So this is a way to put microbes to work for you in your own kitchen. The microbes today that will be working are a broad group called the lactic acid bacteria, and they look something like this. And these bacteria, many of them lactobacilli, transform a plain old carrot into something that's very tangy and delicious. So this is the finished jar of fermented carrots that we'll be making today. So also they're a great beginner's ferment because they're quick and easy, and because carrots have sort of a consistent texture, the results are usually very crunchy and a very good thing to start with if you're new to fermenting. So here's what you need. You need a 2-cup measuring cup. You need a jar. some sort of weight. And what we've done here is put some glass beads sealed inside a Ziploc bag, and that will help weigh down our carrots once they're in the brine. We need, of course, fresh carrots. So we've prepared them here, and they are just unpeeled, and they are cut up into kind of as uniform a size as we could get them. And you want them uniform size so that they ferment evenly, and you won't have one carrot that's thinner that gets more fermented than a thicker carrot in your jar. We also have fresh garden dill, sea salt, and we're using a very coarse salt, as you can see here. It's a very thick one. You can use any sort of sea salt. You can use fine grained as well. We also have optional spices. So today we're going to use a bay leaf and also some mustard seeds. And then to put those spices in, we have a paper tea bag that we'll just put them in to go in the jar. And we also then need a jar cover. So that's for at the end, a breathable cover. You don't have to use a custom-made cover. You can just use a cheesecloth or a clean tea towel for that part. So we're going to start by preparing the brine. So what we'll do is put 2 cups of water into our measuring cup. And we may use warm or hot water and fill it right up to the 2-cup mark. The warm or hot water just helps the salt dissolve better, but by the time you use the brine in your ferment, you will want it to be room temperature. So then we'll add 1.5 tablespoons of salt into the water and stir it up. And when that's all stirred up, that will be our brine. And so in the meantime, while that's dissolving, and then later while it's cooling, you can chop up carrots. Again, I have just scrubbed these and left the skin on. So the microbes that we want in our ferment are already living on the surface of the carrot and on the skin. And what we're going to do is, by adding the brine, we're going to make some of the microbes bloom and make the ones we don't want kind of die out, go away. So we've got our brine now. So now we prepare our jar, and note that the jar we're using is just a clean, dry jar. It doesn't have to be sterilized or anything like that. As long as it's clean and dry, it will be perfectly adequate, because again, we're creating the right conditions for sort of friendly microbes to grow, and that will keep the ferment very safe. So to begin filling our jar, we're going to take the dill, a nice big bunch of it that's been washed, and just kind of coil it inside the jar at the very, very bottom. And then on top of that, we're going to have the spices. So we can fill up our tea bag with these optional spices. So if you can help me do that. So we put our mustard seeds in, and our bay leaf. Just pull that drawstring, and then we can pack that as well down in the bottom of the jar. And that will add some nice flavor to the ferment. Very subtle flavor, really. So you can add different spices if you like. Peppercorns are also nice. And now we can add our carrots, and we're going to pack in the carrots. So again, I've cut the carrots to a pretty consistent length. And so we're going to pack them in.
You can see they're nicely packed in the jar.
Okay, finish packing. See if we can get all of the carrots in there. Pack them as tightly as possible. Just Just jam them right in. Great job, Cece. Now, the way you know you've packed the jar well is try turning it upside down gently. And if none fall out, you've done a great job. So now we're going to add our brine to the jar. So what we do is just slowly pour the brine in, and it's gonna fill every little crevice in that jar, and stop pouring about 1 inch from the top, just about right where this ledge is in the jar. Keep going, keep going. So you want to make sure that all your carrots are covered. And oops, I still see one poking out, so let's add a little more brine. So the reason for this is that for any traditional brine ferment, you need 3 elements. So one element is the substrate. The substrate in this case is the thing you're fermenting, it's the carrot. You also need a brine. Of course, we've added the brine, and that's gonna create the environment for the microbes to grow and transform the carrot. And the third element you need is actually the lack of an element, it's a lack of oxygen. So we want the brine to cover up all the spaces in here so that there's no oxygen. Because if there is oxygen, then the wrong kind of microbes could grow. So to be extra sure that we won't have any oxygen, that's where our weight comes in. So we're just gonna place the weight on top of the carrots and right into the brine. Yeah, some might come out and that's okay. As long as your carrots stay put. And usually at the beginning, your carrots will be packed nice and tightly, and there's, it'll feel like you don't need the weight, but over time, the carrots will contract a little bit, and that's when you'll find they'll start poking out of the brine. So that's when it's handy to have had the weight on top after a few days. So there we are. And all we have to do now is to cover it with a breathable cover. And you could possibly secure it with an elastic band. And this ferment should be ready in about 1 week. So again, here's the finished ferment, and the brine turned a little bit cloudy. The way that I knew this was ready is by taking a tiny bit of the brine with a clean spoon and tasting it. And if it tastes really salty, then you know it's not quite ready. But if it has like a sour flavor to it, then it may be ready. And you can leave it even longer than 1 week if you like it more sour. But I find just after 1 week, they're quite delicious. And Cece likes them too. One more thing, a frequently asked question is whether the microbes that grow in a ferment like this are probiotics. And the answer is no. No, because the definition of probiotics are live microorganisms, which when administered in adequate amounts confer a health benefit, which means that you need to know what types of microbes exactly and how many, because they need to be administered at an adequate dose. So you need to know, you need to characterize them in terms of their name and their quantity. So in a ferment like this, I don't know exactly which microbes are in unless I would test this. I don't know how many. And so if I wanted to guarantee you some sort of health benefit, I would have no way of reproducing this exact ferment. You know, every ferment like this is a bit different, created in a different place by different people, and just a beautiful wild mix of microbes. So that is not the same as a probiotic. But they are called live cultures, and I think there are several scientists in groups around the world working on finding out the health benefits of even these wild collections of microbes. So happy fermenting. Thanks for joining Cece and I today. And if you do make these fermented dilly carrots, we would love to hear about it. You can tweet me a picture. You can find me on Twitter @by Chris Campbell. That's BYKRIS Campbell @by Chris Campbell. Happy fermenting. Bye. Now you can turn.
Yeah, so we hope you are going to try and do that probably on the weekend when you have enough time to do that. And now we're running a little bit out of time. So let's keep talking about food. So as you know, oranges are, sorry, I need to move this. Carrots are orange, and that's because of a pigment that they have that is called beta carotene. And this pigment is used in the food industry to give color to different foods, just like cheese, for example. If you run into a piece of cheese that is more orange than usual, it probably has beta carotene. However, in the industry, beta carotene is not obtained from carrots. It is obtained from an algae, that is this one, called Dunaliella salina, that produces huge amounts of beta carotene. And when it blooms in lakes, actually the lakes turn pink, like the one that you can see here. So this is one pigment that we obtain from microorganisms and that is used in the food industry. We also have more red from the same bacteria, from Serratia morsensis. We obtain pigments that are used in dairy products, specifically in yogurts, probably strawberry yogurts, so they can turn and look a little bit more pink than they should be. We also have, this food that you can see here. I'm sorry if you're getting hungry, I hope not. Here we have fermented rice, surimi, salmon, and this is a dish called Peking duck. All these dishes are traditional from Asia, and the color that they have is sometimes enhanced or produced by a pigment that comes from a fungus called Monascus purpureus. And this fungus is the one that you're seeing on this petri dish, can produce pigments that go from yellow to orange and even purple. Like I said, all these dishes are common in southern China, Japan, and other regions of Asia. And finally, I want to tell you about another bacteria that is called Chromobacterium violaceum. It's the one that you can see on the petri dish, and it's obviously violet, and the compound that it produces is called violacin. This compound is nowadays used as an antibiotic, but it's currently being researched because it has a lot of potential in the food industry to give different shades of violet, but most of all in the cosmetic industry. So who knows, maybe the cosmetics that we are buying in the next few years will have a little bit of violacin in it. So that's another use that microbial pigments may have. And well, I think that's all. We're running out of time. You can find me on social media, Facebook, Instagram, and Twitter under the name of Pop Microbiology. So I hope you enjoy this section about pigments in food. And that would be it. Yeah.
Lovely. Thank you very much. Thank you very much indeed. Yeah, it's very inspiring. You've put together a fantastic set of workshops there with lots of different things I'd never thought about before, and yeah, giving us the patterns and everything was great. Do you both try everything like this? Do you kind of do all the different things that you showed, or do you specialize in one particular area?
Not me. I'm doing my PhD in bioinformatics, so I'm not really growing microorganisms in the lab, but I think it's really interesting. Yeah, I don't know, Elizabeth. I think she does something similar to me.
Yeah. So I'm a bioinformatician, but I've always loved auger art, and I used to work in a veterinary diagnostic lab, and I was there. I'd often use the microbes to draw different things for the holidays. So I did like Easter eggs or hearts for Valentine's Day. I just think it's a fun medium to express yourself creatively. And then my fiance and I also brew our own mead, which is something that we started doing once we started doing microbigals, is really getting into fermentation and seeing all the different yeasts and the different flavor profiles that they can create has been really a fantastic experience.
Great. Yeah. I think, do you feel like it's a nice way to kind of interact and engage with microbiology outside of the kind of day job, away from the computer screen doing the bioinformatics? It's nice to do something tangible, but also related to the subject that you enjoy.
Definitely. Yeah.
Yeah, it would be a good way to go.
Cool. Well, yeah, thank you for all these and all the resources you gave, like the videos and stuff. I'm definitely going to go into them and see if I can find something to try my hand at and try and explain about microbiology while I'm doing it. It's been very, very fun indeed. So thank you very, very much. And I'll let you go.
Thank
you.
Bye.
Bye.
Okay, we are getting very close to having a break, but we have one session to go. It's a pre-recorded session. But before we do that, I just wanted to check the time zones that we're on now, because a lot of the world actually is now on September 17th, which means a lot of the world is already in International Microorganism Day officially. We have got Berlin, Germany, lots of Europe. Actually, it's just gone past midnight in London, in the UK, and Portugal, and I think the whole of Africa now is in International Microorganism Day. So I guess we're just coming for America, North America, Central America, South America. We're coming for you. You're not here yet, but you will be, and we'll be there when you cross that date line. So yeah, let's move into the next session. Don't forget to check us out on social media. You can watch the live stream there if you're kind of browsing on Twitter or Facebook. And we also have it on YouTube on the websites. But right now, we're going to go into the final session, which is about the human microbiome, the loss of our oldest allies. Jalissa Reynoso Garcia has pre-recorded a video for us. She's a PhD student at the Department of Biology in the University of Puerto Rico. So without further ado, I will hand over to her.
Hello, everyone, and thank you for being here. My name is Yelisa Reynoso Garcia. I am a doctoral candidate in microbiology at the University of Puerto Rico Rio Piedras campus. And I will start with a question. Do you know that the vast majority of the cells in our bodies are cells of microorganisms that have lived with us for millions of years? This collection of microorganisms, known as the microbiome, do important things for us. The loss of members of the microbiome can have great consequences for our health. While some bacteria are related to disease, others support life. To help support the microbiome, eat a healthy diet and avoid the inappropriate use of antibiotics. This is a representation of global warming, and we can find in global warming has a large-scale change due to human activities. We're going to talk about a change on a microscopic scale. And I'm going to make the point that the scale of the problem may be equivalent to the scale as well. So what is the microbiome? Our body is home to trillions of microorganisms, including bacteria, fungi, and other microscopic living things that live above and inside us. We have more microbes in our bodies than we have human cells. Indeed, the microbiome is considered an additional organ, not only because it weighs 2 to 5 pounds, which is roughly the weight of our brain, but also because of its essential role in human health. Digesting our food, protecting us from bacteria that cause disease. Therefore, without the microbiome, it will be very difficult to survive. We do not get microorganisms in us randomly. They have a specific habitat and function in our body. That means that the microbiome in your gut is different from that of your skin and your mouth. The most diverse is the gut microbiome, and we will focus on that one. We all share microorganisms, but there are others that will be uniquely yours, such as a fingerprint. And you may be wondering, so what? The problem is that our ancestral relationship with our gut microbiome has been altered over time. One possible explanation for this phenomenon is the missing micro hypothesis, which postulates that urbanization and modern medical practices have decreased infection diseases such as tuberculosis, measles, and malaria. However, access to healthcare and increased life expectancy come at a cost. Increased immune and metabolic diseases, such as diabetes, asthma, and obesity. These diseases that increase with modern lifestyle are associated with changes in diet, inappropriate use of antibiotics, and increased sanitation. Populations with an ancestral lifestyle, such as hunter-gatherers and farmers, have a diet high in fiber and complex carbohydrates and maintain greater exposure to the environment. On the other hand, modern humans have adapted a Western diet with processed foods high in fat, sugars, and simple carbohydrates. Additionally, we spend much of our time indoors, contrary to our ancestors. These practices have contributed to the disappearance of microorganisms in our gut. Studies have revealed that the microbiome of extant hunter-gatherers and farmers around the world have greater diversity of gut microbes compared to urbanized populations. These populations that never had contact with Westerners and the farmers have differences in the composition of their microbiome. However, they are more similar to each other compared to the urban populations. This suggests that the microbiome of urban population differs from the microbiome with which we evolved millions of years ago through differences in composition and the number of bacteria present. The ancestral microbiome has also been studied in archaeological samples. Advancing technologies are providing us with the opportunity to compare the gut microbiome of modern and extinct populations by the study of ancient microbial DNA preserved in archaeological samples such as dental calculus and coprolites, which are mummified feces. Yes, the DNA, the genetic material that you inherited from your parents, can be preserved for thousands and even millions of years. Coprolites collected from a rock shelter in Mexico showed that the gut microbiome of extinct population was more similar to that of rural populations than that of the US residents. This is evidence that the modern lifestyle may alter the human gut microbiome. I know, coprolites are amazing, at least in a scientific perspective. Some modern practices, such as antibiotic treatment, have saved the life of many people who would otherwise have had a fatal disease. Antibiotics were discovered by Alexander Fleming. I imagine you must think that through a complex experiment, but no. He discovered it by accident. When a bacterial culture, which is a method to grow bacteria, was contaminated with fungus. And he realized that fungi prevent bacteria from growing. The antibiotic penicillin has that name since it comes from the fungus penicillium. And that was how a mistake changed medicine and science forever. So next time you made a mistake, don't feel bad because you can be changing the world soon. But let's get back to what we were talking about. Due to the effectiveness of antibiotics against infections, people began to use them more, and more. However, the use of antibiotics can disrupt our microbiome and cause the loss of some microorganisms, and as a result, alter the way our immune system attacks disease-causing bacteria. Increasing evidence suggests that antibiotics can have a detrimental effect on health, especially at an early age, where the microbiome is still unstable. Children who receive antibiotics in their first years of life are at increased risk of developing asthma, allergies, and obesity. Although antibiotics save life, the microbiome do not fully recover after an antibiotic intervention. So, how can you improve your gut microbiome? Overprescribing and inappropriate use of antibiotics should be controlled. It is estimated that 30 to 50% of antibiotics used in the hospital are unnecessary. Therefore, we need regulation and education on the importance of the proper use of antibiotics. Antibiotic treatment in the first years of life should be avoided due to the long-term risk it poses to children. Antibiotics kill many bad and good bacteria in the gut microbiome, possibly contributing to weight gain and antibiotic resistance. Thus, only take antibiotics when medically necessary. In the future, we need also shift our medical approach towards the use of antibiotics with higher specificity to avoid disturbing the rest of the microbiome and to use one that treats specific microorganisms. We need to restore the microbiome. If someone have to take Antibiotics, maybe we can replace the lost microbes with probiotics, which are found in food, or enhance depleted ones with prebiotics, which are fibers in food that stimulate the growing of microbes. And talking about diet, the Western diet also impacts the gut microbiome. The Western diet is high in fat, sugar, and simple carbohydrates, which has been associated with a reduction in the variety of microorganisms and an increase in disease. So this diet-induced loss of microorganisms limits the transfer from mother to babies, perpetuating the inheritance of an unhealthy microbiome. Furthermore, this reduction in microbial diversity cannot be recovered after several generations, leading to the extinction of microbial species in the gut microbiome. There are many ways to improve your gut microbiome. Eat a diverse range of foods. This can lead to a diverse microbiome, which is an indicator of good health. In particular, legumes, beans, and fruit contains lots of fiber. Eat high fiber foods. Fiber is a prebiotic that stimulates the growth of beneficial microorganisms and promotes the diversity of microorganisms. These microbes break down food fiber into other products essential for health. Eat probiotics. Probiotics are live microorganisms that can be consumed through supplements or foods such as yogurt and kefir. Limit your intake of artificial sweeteners. Some evidence has shown that artificial sweeteners increase blood sugar by stimulating the growth of unhealthy bacteria in the gut microbiome. Remember that our microbiome is a mirror of our lifestyle. Thank you.
Okay, thank you. Thank you very much for submitting the presentation. Thank you everyone for listening. What we're actually going to do is take a short break now. So we're going to come back on the hour in about 45 minutes. So go and take a break and check out our social media. I would like to share something with you now that we're trying out on Twitter. Because if you get the chance to take part in this, it would be very, very cool. So I was asking the question, can you describe your research or a subject that you're interested in emojis? And we've been getting a lot of responses actually. So yeah, here we've got Dr. Justine Debelius, and she's pretty nicely explained her research in emojis here. I guessed it wrong, unfortunately. Thought it was fecal microbiome, talking about community and geography, but this is actually community of bacteria, community mapping, finding targets and improving disease. Lots of people have tried this. So if you actually take a look, you can try guessing them, because I've been doing a terrible job, to be honest with you. So if you can do better than me, that'd be great. But also tell me what your research is, what your subject is in emojis, because well, people have been getting very creative, so that might be something interesting for you to try. But I will leave it there for now and say thank you very much for your attention, for your time, and I'll see you in just over 45 minutes for the next session, and I'll give you a little bit of a teaser of that session there. Bacteria and Antibiotics 101. So yeah, I'm looking forward to that. It's presented by people who are actually very close to me in Sweden. They're from the Uppsala Antibiotic Center, Uppsala University, and it's going to be a good one. So I'll see you there. Thank you very, very much indeed. Okay, hello again, everybody. We're back from the break and we're ready to get into some more International Microorganism Day talks just on the time zone website here. And I'm looking to see if anyone else is now on International Microorganism Day. What have we got here? So London, UK went into International Microorganism Day. We've got UTC, and we're waiting for Rio de Janeiro in Brazil. They're still 9 o'clock, so there's still a few hours to go. So we're really just trying to get across the ocean here. And while we're waiting for that, I'm going to introduce another talk. This is a video that has been prepared by some very, very talented science communicators and scientists in Uppsala, in Sweden. It's called Bacteria and Antibiotics 101, and the presenters are Eva Gorandia and Jenny Jagman, and it's been produced by Jimmy Tokmaji. So it's a pre-recorded video, but if you have any comments or questions, you can still write them, still send them to us on social media, any of the social media platforms, or in the comments on YouTube, and either I'll try and answer them, or we'll get back to you with a response from the makers of this video. So without further ado, here we go.
Hi guys, welcome to this part about bacteria and antibiotics. I guess if you have clicked on the title, it's because you are interested to learning a little bit more about bacteria and the ways that we have to treat bacterial infections, which are antibiotics. So in this video, we're gonna give you a run through about our bacterial world, like it says here, about where you can find bacteria, where bacteria are normally living, where do antibiotics actually come from, and also a little bit about the real antibiotics that we can get from the doctor when we actually need them.
I think a lot of people haven't always thought about bacteria in the sense of where they are, what they are, all of that. So Ava, can you see bacteria with your bare eyes?
I wish, because that would mean I have really good eyesight. No, we cannot see bacteria with our bare eyes, but bacteria are actually everywhere.
Yes, they're on all surfaces, pretty much. They're on us, in us, in the ground outside, in the water outside.
Microscopes allow us to see the bacteria, the very, 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 very tiny bacteria, as single organisms. And this is what they look like.
Yeah.
We have here some photos of bacteria that are actually growing in different parts of the body. The photos that you are looking at right now, they are called micrographs because they're photos taken with microscopes, and they come from 3 different parts of the body. And what we want you to do right now, we are gonna put each one of the photos, we're gonna leave it for 5 seconds, and we want you to think about where in the body these bacteria are actually growing. Let's play this game, okay?
Yeah.
So first photo. Jenny, do you know where these bacteria are coming from?
So this is actually from your mouth.
Yes.
Or a person's mouth, not yours specifically.
Yes, no, my mouth. But what you can see here is actually a quite diverse environment with bacteria. Some are elongated, some are a little bit more like filament, some are pretty round. Then we're gonna do the second picture. So here you're gonna see another micrograph and guess which part of the body these bacteria are from. Can you tell us, Jenny, where is this one coming from?
Well, I can give a little bit of a hint too. So if you remember seeing a piece of what looks like wood here, so things that we eat have fibers, right?
Yeah.
Kind of look a little woody. So this is actually in the intestine.
Yes.
There's a lot of bacteria in your intestine. Bacteria help us break down the food that we eat and help us get out all the nutrients from it. We would honestly not really be able to survive without the bacteria in our gut and in our intestine, our microbiome.
And as you can see here, the predominant bacteria, the bacteria that you find the most, is this sort of like shorty, elongated bacteria. These are the typical form that all the coliforms, similar to Escherichia coli, or the lactobacteria, that is the bacteria also found in the yogurt, for example. So that's the majority of the bacteria in the gut that helps us having a healthy digestion and a healthy body, are these kind of bacteria. And the last one, but not least, this one, we're gonna see another micrograph and try to guess where this is from, and maybe see if there's any difference with the other photos that you saw before. This one makes people laugh a lot when we tell them where it's coming from. So can you tell us where it's coming from?
Well, first off, we could think that it looks pretty different, right? There's almost only round bacteria here, pretty circular bacteria. So it's not really inside us in the same way that the other pictures were. But if you could think about a spot on your body where maybe there's a collection of some stuff.
Maybe a spot that doesn't get cleaned very thoroughly, very often.
As often as it should. It's actually your belly button. So there's a little collection of bacteria in your belly button. There's actually bacteria all over our skin, and they also help us.
Yes.
So yes, when you cut yourself, maybe it's not great, but the bacteria in our skin also kind of protect us from worse bacteria that could make us sick. Many of these wouldn't make us sick at all.
No, actually, the bacteria that is in our skin and in our flora, either inside intestines, it's essential to have a proper immune system. It is so important that now it is considered an essential part of the innate immune system, which is everything that protects us from infections and from external agents like viruses or bacteria that can make us sick. As we were saying, bacteria are very, 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 very tiny. We need to have these micrographs and these microscopes to see them. But is that the only way that you can see bacteria?
No. So there's actually a few examples where we can see bacteria with our bare eyes, right? Pretty cool.
Yes.
We have an example here where actually even from a plane, you can see bacteria because there's so many of them at once.
Yeah.
So you don't see the individual ones, but you see the total.
Yes.
So here we have a picture of a lake that's turned green from the amount of bacteria in them. We also show you a micrograph of the bacteria so that you can see how small they actually are. They're about the same size as the other bacteria we've shown.
So you have the difference between the individual single bacteria and then the community of the bacteria when there are many of them growing together. So knowing this, that you can actually see bacteria with your bare eyes if there are enough of them, now we can go ahead and we can test if there is bacteria in a lot of different places, because we can sample those places, we can put them and give them some food so they can grow, and then we can see if there is bacteria growing on them or not by seeing if there are bacterial colonies or not.
And a bacterial colony is really like a pile of bacteria growing on itself.
It's like a big family all living together in the same place because they all come from one bacteria that started dividing and creating a lot of sister daughters bacteria that we call it. So now that we know that we are actually able to see bacteria and to sample if there are bacteria in different places, I think it will be pretty fun if we check where can we find bacteria. And you actually took some samples, right, from some places.
I will see what I got.
Yes.
We already got some samples and we're going to get some more in our lab itself.
We go to the lab and we take some other samples and then we see you back for the results as well.
Yeah. This is a real life used lab. A lot of research going on here in the background, but we're going to show you guys some more basic things about how bacteria work, where we can find them, what can kill them and that sort of thing. But just so you know, there can be people passing by and things going on in the background and noises, and that's exactly how this place is. If we want to see the bacteria in the world around us, then we have to be able to grow them because you can't see an individual bacteria, right? We have to see kind of the pile of bacteria. So we have a few samples here from some fun places. We have a soil sample and we have a cat poop sample, my own cats. They were a bit skeptical as to why I was stealing their poop. So what we do is plate these samples on agar plates. So these are kind of squishy. They're kind of like jelly. So I'm going to go ahead and use these glass beads to spread the samples on the plates. So we're going to start with the soil. And then we're going to go ahead and plate the soil sample. And same thing with the cat poop. We give it a bit of a mix. All right, then we shake them. So we shake the plates to spread the bacteria across them. So then we just have to throw these beads away. But regular things around us that we touch every day, like keys, for example. Do we think there are bacteria there? Could be fun to find out if I can get my keys off my lanyard. So I'm going to try this one and just put it down on the plate. Now the tricky part is getting it back up again. Oh, first I'll push down to make sure it's there. Okay. What else do we want to try? We can try rings. So that can also be something that carries bacteria. Both of my rings. We'll see how good I am at washing my hands. People often forget to move rings around when they wash their hands. We can try a clean fork. See how clean it really is. So I'm going to go ahead and roll the fork on the plate. See how good people in this lab are at doing the dishes. And there's also a lot of bacteria in our hair. So I'm actually going to go ahead and test my hair tie as well. Try to get the hair off though first. And this isn't exactly something you clean that often, huh? So I'm going to try to twist it up so it doesn't go everywhere and ruin everything. Ooh, let's see if I can do this well. Okay. Here we go. So we looked at a few pictures of bacteria in our bodies, and we kind of want to try also to see, can we visualize this in plates in the same way? So what we're going to do is sample the bacteria in us. Ava, do you want to help me out?
Sure.
So we're going to start with tongues.
Oh.
I thought.
Okay.
Lick the plate. So these are clean plates. We know that there's no bacteria on them. I'm not condoning people lick stuff randomly.
No.
But let's go for it.
All right.
Ready?
Yeah.
Nice and salty. Okay. So another thing I thought we would test is the ear. There's also a lot of hair around and everything. We can see if we see some fun patterns maybe. So do you want to go first or should I?
I can do yours.
Okay. Now you can
Jenny
get a good
here.
grip on it. See if you see my earring in bacteria. Chili. And wet. All right, let's see yours. There we go. Oh, I hope I got enough. We'll see. I'm not sure we want to see the results of that. But we're supposed to have bacteria on it. So it's not, we're expecting there to be bacteria on it.
Yeah.
Another way we can test things is by swabbing a wet cotton swab on something and then rubbing it on the plate and seeing if things grow. So one of the things we're going to test is my phone. Again, phones are disgusting. So I'm not really looking forward to seeing this, but we'll see how it goes. I have my phone here. So I have to wet the tube first, or wet the cotton swab, I mean. And I'm gonna just go ahead, swab all over the screen. And for good measure, I'm gonna go ahead and swab this side too, which I'm sure I'm worse about cleaning. And then I'm gonna go ahead and spread this on the plate. Okay. So we're going to do the same thing to some other surfaces around here that theoretically maybe should be cleaned, but are touched very often. So we'll see how clean this lab is. We're going to incubate these plates in these big incubators that look kind of like giant, I don't know, cabinets. Yep. Go ahead and put them here. Nice and toasty. So now it's been a few days and we've been able to incubate our plates. We started incubating them at 37 degrees, right? But some of these things we actually realized after a while actually grow better at room temperature. So if you think about it, I mean, 37 degrees is body temperature. So things in the body grow better at 37, but things on our skin or in the environment maybe grow better at 20 degrees. For most of these plates, we did starting at 37 for one night and then one night at room temperature.
What did you see, Jenny?
So we can start with our cat poop plate. Here we see, interestingly enough, my cats seem to only have one species of bacteria that they carry, but there's a lot of it, which is maybe not so surprising. We carry a lot of bacteria in our guts, and they generally, as we said, are good for us, right?
We saw in that micrograph, the majority of the bacteria that we have in our guts tend to be like coliforms or lactobacteria that have similar shapes in the bacteria themselves, and also perhaps they make similar colony looking. So that's why it looks like it's almost all the same bacteria. It might be there are different species, but of course, just looking at the colonies, we cannot really tell.
Yeah, but we see a very different result when we look at the soil sample that we have. So here we see lots of different kinds of bacteria, and they all have kind of different shapes, the colonies too. So more different colors, different shapes, different styles.
Different sizes.
Different sizes, yeah. Some things grew much better than others.
And this is because in the soil, generally, there is a lot of different things, especially if you have a like soil from a household where it's going to be more in contact with humans. It might also
and
have
cats.
And cats, of course. It might have some bacteria that are related to the human flora. Then, of course, you are going to have not only bacteria, you're going to have other microorganisms, like could be some fungi, some molds. And actually, we can see here in the plate really well how we have a lot of different colonies. And I suspect that those bigger colonies that we have over here, those might be perhaps the fungi type of
they kind of have fingers coming from the colonies,
which could be like the hyphae from the fungi in the soil itself.
But it's hard to know what these are, so that's important to keep in mind when you're growing bacteria from an unknown sample, from especially soil, especially feces. You got to be very careful with it because you're growing large amounts of unknown bacteria. You have to treat them as worst case dangerous bacteria.
That's why we are not opening this plates right now.
These we are extra careful with. Some other things that are things that us humans maybe touch more often. So we can start with our the plate that we did random stuff on, kind of. So here we have, if you remember, we put my rings.
Yeah, we put your rings.
My keys.
And your rings were?
My rings were up here.
In this area here.
And then the keys were here. Unfortunately, the plate got dropped later, so there's a crack in it, but that's not from anything we put on it. This is the fork. You can kind of maybe see the
shape of the
shape of the
three,
fork.
1, 2, 3.
The prongs. That was a clean fork, remember?
Supposedly.
And then you see the coil for my hair tie here.
This is the most striking one, and the one that you can see the shape of the thing
Exactly.
we sampled the most.
Yeah.
You can see perfectly that everything that tie touched, there is a colony growing.
Yeah, and some different colors here. It's a lot of diversity in how these colonies can look. So then we also have a plate with my daughter's hands and feet. This is a slightly concerning plate as a parent, but remember, this is actually totally normal.
Yeah.
So the top of the plate here, you see my daughter's hand. So she kind of dug her hand into the agar of the plate. So the agar is the jelly stuff.
The food for the
bacteria, yeah. So you can see that she kind of dug her hand all the way through there. And there was bacteria in pretty much every single finger. You can kind of see the forms of her fingers there. And down here at the bottom, you can see kind of her foot. So this is the pad of her foot with her toes, and this is the heel.
I really like that. You can almost see the shape of the toe. It's so cute.
It is quite nice.
Yes.
Most people take, like plasters of their kids' feet, and I take
bacterial
samples. But yeah, so
that
not
means,
totally newly washed kid, but a relatively clean kid.
I mean, kids normally have their hands in their mouth all the time,
Yeah.
and mouth is a place where you have a lot of different bacteria and a lot
But I
of
also
it,
wouldn't
so-
expect anything different from us
No.
humans, us adults. It's just, in this case, her feet and hands fit better on a plate than ours do.
Exactly. Yeah.
But we did take some samples from us.
Yes,
remember?
we did try to see if we have a lot of bacteria on us or not.
So
And
let's
what
start
kind?
Yeah, let's start with our ears.
Yes.
We actually have quite different samples here.
Mhm.
Let's start with Ava's. So Ava actually didn't have a lot of bacteria in her ears, or maybe I also didn't push hard enough. So a lot of this comes down to sampling technique also, if everybody does it the same way.
Mhm.
But you see that there's a little bit there as well.
Some yellow bacteria, some white bacteria, which are typical skin bacteria that we
Yeah.
can find.
And also, I mean, if you get hair touching as well, it's usually the same kind of bacteria in
Mhm.
hair. Mine has a little bit of a crack in it as well, but it's quite a bit more and a little bit more diverse.
Yeah.
You do see actually a little bit of these red orange colonies here.
Yeah,
as well.
this is something that
Gonna
follows
come
you.
back a little bit, yeah. But a little bit more bacteria on mine. Then we also did our tongues, and this is pretty interesting. I don't even know if you're going to be able to see too much of Ava's, but Ava had an incredibly clean tongue.
There is a little bit of a background growth,
Mm-hmm.
very, very small colonies. I would suspect that those are very slow growing because you
Yeah.
had them 2 nights incubating. And then there are some bigger colonies, normal size colonies for the amount of incubation,
Yeah.
which, yeah, it means like
Ava's got a clean tongue. You
have
clean your
so
teeth.
much bacteria in my mouth, even though it's completely normal to have a lot of different bacteria, especially in the teeth and everything.
Yeah. So I have a bit more. You could see mine as well here. You see this background again, the kind of slow growing bacteria growing in the background, but I do have more of the bigger bacteria there as well. But we did take some swab samples too, which I find really interesting. So usually we don't really do this when we do these samples because it's not so interesting to show somebody this is what we swabbed. It's nicer to see like the form.
But now you just saw where we actually
Yeah.
took the samples from, and
We get
now
to
we have
show
the
all of it.
results here.
So we can start with, this was actually my phone. So you see these kind of the red and the yellow bacteria coming back. And again, here we see quite a big diversity.
Yes.
And I have to say, usually I sanitize my phone because I work in a lab. So I can get bacteria on my phone. So I sanitize it regularly. Here I intentionally didn't sanitize it for only, I think, 3 days.
Mhm.
So this was 3 days of stuff from a phone.
Mhm.
Phones are pretty gross
Yeah.
when you think about it. This was our lab's coffee machine.
And this is actually super interesting.
Yeah.
Because
We have a clear mold.
obviously the coffee machine gets cleaned by itself with this automatic system, but the surrounding areas not so much, and it's an area
Yeah.
that it's generally very humid all the time. And this is the only sample that we actually got this clear mold growing
Yeah,
in
a
it.
very obvious mold.
So this is the typical mold that you can find, in bread probably.
I was gonna say this is probably what most people are used to seeing.
Yes.
Yeah.
Yes. So this very distinctive from the specific environment we took the sample, and also some sort of bacteria, but not so many. So it wasn't
No,
that
not as bad as I thought it could
yeah,
be.
exactly.
However, our lab computer's keyboard, the computer keyboard, also, I mean, a lot of skin bacteria, what we saw before.
Yeah, the typical yellow bacteria that you might get in this skin or like skin samples or things that skin has touched, they tend to be Staphylococcus aureus bacteria. That's
or
where
other
the names come from,
Staphylococcus.
or other Staphylococcus. What I think is interesting is if you get your plate from your phone, and put it next to it, because of course the phone plate is sampling the skin flora. If you see, this would be the flora that is associated with Jenny in particular, because it's her phone, but this one is more a representative of the whole lab, because everybody comes to this computer and touch the keyboard.
So
you
it's all
can
my
see
colleagues
that it
as well.
depends from person to person. The diversity and the flora in the skin can vary a lot, so much that you can even see it just with this kind of easy sampling in plates.
My phone sample actually looks closer to my ear sample, more than the keyboard, even though I don't touch my ears that often.
But if it's your skin, and that's
exactly
your
it's
own
skin
flora,
either
and
place.
it's something that is really important inherently to your own health and to your own body.
Yeah, it's important to say that most of these bacteria, I mean, obviously we walk around with bacteria on us all the time. We usually don't get sick. I mean, you wash your hands before cooking, especially if you're cooking certain things. And especially right now, in these times, we wash our hands maybe a lot more often. But I mean, this is normal and this is natural and not unhealthy.
Yeah.
However, this is the place that you want to have a bit clean. This was the toilet seat that we sampled. And it is quite clean.
Pretty clean, yes.
Which is not that surprising when you think about it. I mean, you clean your toilet more often than you maybe clean your phone.
On your keyboard, obviously, as I see. So,
so that's comforting.
Yeah, it is indeed.
And a door handle is also pretty clean. Not too much going there. That's also kind of, I mean, it's something that you touch, but it's
something that gets clean often. Yeah.
It's
It's part of the routinary cleaning, you know, like when you clean surfaces, you always clean doorknobs, but you don't clean perhaps phones so often.
No. That's what we've seen on these environmental plates that we've taken. We see a bit of diversity, and like we said, some expected, some unexpected things, and I hope you guys learned a lot from that.
I hope you like seeing these different colonies on the plates. I think they're pretty exciting, and they teach us a lot. We can actually understand a lot of the things that are around us
that we can't really see usually. So now that we've seen where bacteria can be, and we've seen that even on clean objects that we know are safe, they can be pretty much everywhere, right? And a lot of them, especially on us, in us, everything like that. So we know that bacteria around us aren't necessarily bad, right? We're not sick all the time.
No.
Things are pretty good.
And the majority of the bacteria are actually either harmless or beneficial.
Exactly, yeah. But sometimes you do get sick from bacteria. So a lot of people may have had a strep throat sometime in their life, or other infections. Stomach bugs can be caused by bacteria.
Some urinary tract infections as well. Pretty common.
So we do occasionally need to kill bacteria. And it can be good to know how to.
Yes.
So if you're trying to kill bacteria, and you've never, we didn't have antibiotics before, you didn't know where anything comes from, you kind of just trying random things. What's something you might try to kill bacteria?
Well, something I might try, something that is strong, I would say, right?
And oftentimes these old remedies that we have that are passed down from in family, they're often associated with food, right?
Yeah, some natural
products, food, and that sort of thing. So maybe there's some food that kills bacteria itself.
So I think we should be fun to go ahead and maybe test some strong foods, like it could be garlic, like it could be chili peppers, that ginger, for example. I think people.
Yeah, I think a lot of people have heard of ginger to treat a sore throat, maybe.
Yes. So I think it would be fun to go back to the lab and then put these foods into plates with bacteria and then see if they are able to kill bacteria or not.
Sounds good.
Let's do it.
So what I'm going to do is first spread some bacteria on some plates. So we're going to use cotton swabs that are sterile. I have some bacteria that I need to mix. Then I just dip the swab in the mix and spread it on the plate. So this way, what we're going to see after the bacteria have grown is bacteria covering every part of the plate. Which means when we put something on the plate, on top of the bacteria, we can see where they don't grow, see if they die. Take a fresh one of these. So this is just regular lab bacteria. They're very, very safe. I usually tell people that you can theoretically drink a glass of this and nothing will happen to you bad. So then I have some foods that we want to try. We're going to start with ginger. So I'm going to try to scoop up some of this ginger, get some of the nice juicy gingerness of it to put on the plate. This is easier said than done. Try to spread some of this stuff out. I have a bunch of different kinds of chili, so I'm excited to see what happens with this one. There we go. And same thing here. We want to get a lot of the chili juice to see that might be where the real power lies. Like that. And then the habanero. Smells pretty strong in here now, huh? Okay. So we've got a nice colorful chili plate. Historically, one of the places where we found natural antibiotics is actually in molds. So think, moldy bread, moldy vegetables, moldy fruits. We're not sure about this one. Never tried this one personally, but I found a moldy kiwi. So I'm going to see if this one works. Now this doesn't smell quite as good. I think one's enough. We have some garlic. Smells really strong. Maybe it'll work. We'll see. In this case, I want to get some of the nice garlic juice up as well. Oh, that's a strong smell. Okay. And we all know lemon is sour. So the idea might be that this might also kill bacteria. I have a slice of lemon. I'm just going to put that straight on the plate, and we're going to see if this has any effect on the bacteria. Press out a little bit of the juice from it. So we see if it works. There we go.
After having incubated the plates that contain simple lab bacteria with some foods that we want to check if they actually can kill bacteria or not, let's start with one that, of course, a lot of people think that might kill bacteria, and because it's quite strong, because it repulses a lot of people also, like how strong it is. And bacteria seem to not like it at all.
No.
And this is the garlic sample. As you guys can see here, there is a pretty intense zone of inhibition. So the way that we are checking these plates is everything that you see cloudy like this is the bacteria that we put in the sample. It was, of course, transparent when we were actually sampling it because it was really small, but after one night growing, it becomes like this. In the middle, we put the food that we want to test. And what we see is that there is no bacteria growing at all for really big area around the food in itself. This means that whatever substance was in the garlic diffused through the agar, and then the bacteria was like, nope, when this thing is here, I don't want to grow.
Not interested.
Not interested in growing, or even cleaning it actively. It depends. We cannot really say which one of the two options it is. But as you can see, garlic, it's pretty strong antibacterial. And this we have known for a long time, I would say, in human history, that garlic somehow can do something towards microbes. The thing is that the compound that does this to the bacteria, it gets completely inactivated when it gets to the stomach.
Yeah,
so of course,
your stomach acid kills it.
Eating a ton of garlic is not going to help you get rid of an infection in some part of your body. In principle, if you will cover all surfaces with garlic juice, bathe in garlic, you could potentially, get rid of a lot of bacteria, but that's not really the point. We just wanted to mention that.
You got to question if it's worth it at that point. Do you want to smell like garlic all the time very strongly?
What we can see is that, there are natural things in food products or in some natural that can actually kill bacteria. Another plate that is perhaps a little bit surprising, because I would expect that the strong chili peppers might actually kill bacteria.
You'd think. I mean, you think of the spicy everything, like it should be killing stuff.
But what we can see here is actually not so much effect when it comes to the inhibition. At least, of course, not as strong as the garlic, definitely. No. some sort of very, very, very close to the green chili, but no so much or almost nothing into the red chili and the yellow chili.
And most of what you see, it's not necessarily even that the bacteria are dying or not being able to grow at all. It's that you see that they're growing slower. So we don't see as much bacteria around the chili while we still.
It smells tasty.
Smells so strong, yeah. Yeah, this one actually did smell quite nice. But we do see that the bacteria aren't growing as fast around the chili. So they are affected.
Yeah.
But not killed. They're not
Not
completely stopped from growing.
One that is actually pretty interesting. And
Yeah, this is new for us.
We're trying, we had never really tasted the kiwi that was a little bit moldy that you sampled. And we are a little bit surprised by the results because we do see an effect, quite a strong effect around it. Of course, not as much clear as the garlic, but still there is an effect around. But the area where there is more mold growing, which will be up here, as you can see.
You can even see the mold still growing on the kiwi. So the mold has continued growing on the kiwi.
In that part, there doesn't seem to be a stronger effect of the killing or the inhibition. So what we're thinking is happening around here is that the acidity of the kiwi is being spread through the agar, and is this acidic condition, this acidic environment that is not allowing the bacteria to grow. And we have a control sample for this acidity, which is we also plated a lemon. And as you can see, the lemon effect is really strong, but we do know that lemon doesn't have any component that is actively antibacterial.
Yeah, it's the acidity of the lemon.
We know that the juice is very acidic. The acidity kind of spreads through the agar, and then the bacteria don't like growing. This particular bacteria don't like growing in this acidic condition. There are some bacteria that love acidity, but they are actually not this kind of bacteria.
And I mean, this comes kind of naturally. So if you think about it, if you cut your finger and you squeeze a lemon, it's so painful, right?
It is,
yeah. So it makes a bit of sense that this would be.
Yeah. So you can see very, very clear. And if we compare perhaps the kiwi and the lemon, you see that it looks a little bit similar.
Yeah.
So the kiwi effect might not be the direct mold that we were trying
it's hard to know if
it's,
and it might also be that the mold is preventing the growth a little bit, but not killing it. And the killing, or like the stronger effect is the acidity. It's hard to tell.
Yes. And we are going to end up with a little bit of a controversial food item that we test here in this sampling, and it's the ginger. And this comes with a disclaimer. We know that a lot of people think that ginger helps somehow when you are feeling down with a cold, which might or not be bacterial or viral related.
Usually viral. And a lot of people make like ginger teas and they do feel better. And we don't want to make you think that you are not right and that you don't feel better when you're taking these teas, because they certainly help. But when it comes to testing if they can kill bacteria, what we see is that there is no effect whatsoever. So they cannot kill bacteria. Ginger per se cannot kill bacteria, but it can certainly make you feel better. It can be anti-inflammatory. It might, things don't swell up as much. The effects from the bacteria might not feel as strong.
It's also an immunobooster. So it is been shown to help the immune system to get rid of whatever infection is happening.
But the ginger itself is not killing any bacteria.
No. And this you can see very clearly in this sample, especially if we take one of the things that can kill bacteria.
Okay, let's go back to garlic and compare the two.
Yeah, so we can compare.
That garlic doesn't kill bacteria in you either, but
outside, which is the sample, the conditions that we're testing it, you can see that there is no effect from the ginger. Although ginger is very tasty and is present in a lot of tasty food.
As I say, all these things combined minus the moldy chili makes a pretty delicious dinner. Also minus the bacteria.
It's really good.
Yeah.
So as you can see, there's a way that we can test if some foods might have antibacterial properties and kill bacteria in the lab conditions and in the situation that we're testing them in. And we got some surprising results, some things that maybe
they're very easy to see too.
Yes,
very
exactly.
Visual.
And it's also kind of fun to test.
Yeah,
good.
It smells nice.
We've seen what food works, what food doesn't work. And what I want you to know is that actually antibiotics or something that could kill bacteria was firstly found in also a natural product. This is probably you have heard of Alexander Fleming. Alexander Fleming was a not so clean lab technician back in the days.
Most labs weren't back in the day.
Yes. So he had some plates of the kind that you have seen in the video thus far. And he had some plates, he went on to holidays. And when he came back, he saw that some of the plates had grown so old that they had mold growing in them. The same mold that you can find if you leave your oranges for way too long at home, or perhaps lemons, you forget a lemon in the corner of the fridge.
Or in his case, it was actually moldy bread.
Or moldy bread as well, yeah. There's different kind of molds they grow on foods. And what he noticed, he was smart enough to notice that around the mold growing in these plates, the bacteria that was supposed to be there, because he was working with bacteria, it was completely gone. And he was like, maybe this has something that can actually kill bacteria. And this antibiotic is the one that we know as penicillin, because it actually comes from the mold Penicillium.
And actually, I mean, in Sweden, a lot of people use the phrase penicillin and antibiotic interchangeably. They call all antibiotics penicillin, because this is the first and often a still commonly used one, not exactly the same, but a derivative of that one is still commonly used.
And talking about derivatives, we have seen that some food actually have compounds that can kill bacteria. But if we get sick and we go to the doctor and the doctor really recommends that we have to take antibiotics, it's not enough with just eating a lot of this food that might have some antibiotic properties.
You can't bathe in garlic, can you? Or I wouldn't suggest it. Something might kill bacteria in a dish, but is it ever going to work in us? It has to be functional. It has to get to the right spot. It gets pretty complicated pretty fast. So what we actually give patients who are sick is what's called antibiotics.
Yes.
That are often maybe from a natural source, but they're produced not naturally. You don't give them the food itself.
Because you need to have the right concentration, and you also need to make the antibiotic be able to get to the place of the infection. So for that, a lot of scientists for many years have done hard work to find the ways that these drugs can work as a medicine.
Changing little details at a time, usually, until we find something that works as good as we can make it work.
But what we can actually do in the lab is to test if a bacteria can be killed or cannot be killed by a certain antibiotic. And this is the test that is normally done in the labs, in the clinics, and in the hospitals when a patient has a bacterial infection and they want to test if they can use an antibiotic or they cannot use it. They use the same technique that we are going to go back to the lab and try to show you how we can test if we can use an antibiotic or not against a specific bacteria. Do you want to go to the lab?
Sounds good.
Let's go.
So we're going to test if certain antibiotics can kill the bacteria that we're looking at. In order to do that, we have these little paper discs that are kind of drenched in antibiotics. So when we put the little paper pieces on the plates with the bacteria on them, the antibiotics spread out into the plate. And hopefully they can kill the bacteria that we're looking at, because that's what we want. We want to be able to kill bacteria when they're causing an infection or causing us harm. So first I need to spread some bacteria on the plates. I have two different kinds of bacteria that I'm going to spread. And I'm going to do this the same way I did before. So these are really two very similar bacteria strains, but one of them has gained some genes. So we can see if those genes impact whether or not the antibiotics are able to work against the bacteria. So then I'm going to try to put these discs on the plate. Sometimes it gets a little bit messy, so we'll see how nicely this looks, but I have three different kinds of antibiotic discs here, and I want to get one of each on the plates. I use this little needle to help me and just kind of poke it on. One of those. So then I've got one of each kind of antibiotic on this plate. Do the same for the other plate. Okay. So then after the bacteria have grown, or not grown close to the antibiotic disc, we should be able to see how well that antibiotic works against the bacteria.
If you remember, what we did was to plate different bacteria that we know are more sensitive to some antibiotics than others. And we put also different antibiotics, so we can test different antibiotics at the same time. So here, what you can see is that this particular bacteria that we put in this plate has three different antibiotics, and there is a big zone of inhibition around two of those antibiotics, but there is no inhibition at all around this third antibiotic. That means the bacteria here doesn't really mind wherever antibiotic was in this disc, whereas antibiotics that are in these two discs are effective. They work against this particular bacteria. Here, a lot of inhibition, here, a lot of inhibition. We don't really make differences, or we don't make assessment in how big these circles are so far. It's just that we see they work. This bacteria is sensitive to these particular two antibiotics. On the other side, when we have this other plate, what we can see is that this one is sensitive to only one of the antibiotics, whereas it's resistant to the other two antibiotics. If we put them next to each other, we can see is that both bacteria are sensitive to this particular antibiotic here, but this bacteria has become resistant to this antibiotic, because here you have the zone of inhibition. Here, this antibiotic is working, but here it's completely overgrown with bacteria around it. So it has become resistant to this particular antibiotic.
And it's worth keeping in mind, these are actually very, very, very similar bacteria. It's the same species, or the same strain even, that's just gotten a few extra genes through something called a plasmid. So it's by that way become resistant to the antibiotic.
Yeah, so we can actually see these two bacteria are related. They're very similar, but something has happened that now it makes it able to grow with the presence of this particular antibiotic, where before it would be completely killed. And this is what is called the acquisition of antibiotic resistance, in particular through maybe horizontal gene transfer, which is, the transfer of these particular plasmids, which are extra material
outside of
the normal DNA of the bacteria. They acquire something, and then they have like a superpower, like a little bit like this one, gets like a super cape. It acquired a resistance to a specific antibiotic.
Yeah, which is what happened in this case. It's the addition of new genes. So now we've seen how this ability of antibiotics to kill bacteria can be seen in the lab. We use these little discs, and we can measure the diameter of the zone that's clear, the zone of inhibition, as we call it. But the reason why these antibiotics maybe don't work against the bacteria can differ a lot. So some bacteria, just by how they are, they're the way they are naturally, the antibiotics won't work against that specific kind of bacteria. So like if we look at this bacteria behind Ava here, it has a pretty thick wall here, right? And depending on how this wall is built, maybe the antibiotics just can't pass it. In this case, it doesn't mean that there's anything that's changed about the bacteria themselves. It's just that antibiotic doesn't work against that bacteria, which is why we often need to know what bacteria is causing what infection. It helps the doctor a lot to know how to treat that. But other times, we maybe have antibiotics that we think should work against this bacteria, but they don't. This bacteria has become resistant instead. And if you see in here, this purple stuff, this is this bacterium's genetic material. And bacteria can actually develop the ability to resist antibiotics. They maybe break them down sometimes. So something in here is coding for something that breaks down antibiotics, or maybe builds in a little pump that pumps out the antibiotics from the cell.
So the antibiotic comes in, but before it can kill the bacteria, it's pumped out again.
Yeah. Or if the antibiotics are targeting something in the cell, maybe a mutation occurs and that target in the cell is changed and it no longer really works against the bacteria. So this is a constant thing. Bacteria are always changing. They're always evolving. There's natural mutations occurring, that sort of thing. It's nothing that's really surprising to us. We kind of expect it to happen.
Yes.
The problem is that it's happening so much now.
And it's happening really fast because we're using a lot of antibiotics, and sometimes we're using them for things that are not supposed to be used for.
Yeah.
Perhaps you have heard about the use of antibiotic in animal production, because it's easier to give antibiotics than having to keep them that possibilities of getting infections. So a lot of people used to just give them antibiotics to prevent the infections in the animal husbandry. Also,
sometimes a viral infection looks like something that might be caused by bacteria. It's if the doctor doesn't know the difference between these, it's hard to tell. It's not that the doctors aren't doing their best. It's that it's hard to tell, and sometimes you need to treat somebody pretty fast. And we have a few examples here I kind of want to show real quick.
Yes.
Not all viruses look like this, but we have two examples of viruses that look about like this. Well, if we compare to some bacteria examples we have, or the bacteria that we have here, they look a bit different. But it's also worth bringing up, these are much smaller.
Much smaller. And actually, I think it might be even up to scale. This one here is a virus that actually infects bacteria, not human cells or animal cells. And this is actually kind of like maybe a bit up to scale. Maybe this one should be even
As I say, I think it should be even smaller.
Because this one will come here and it will try to the bacterial cell the same way that another virus goes into our cells if it's a human virus. So we want you to really see that there is a lot of difference in how they look, but also
how they work and how they
sizes of it as well.
Yeah, how they do things.
It's very important that we think about these things, that antibiotics are not just expendable medicines that we can use at any time, in any moment, and for anything. We should use them only when a bacterial infection is suspected and confirmed. And if your doctor tells you that your bacterial infection is not strong enough or bad enough or the kind that will go away by itself, that antibiotics are not needed, that we don't really think we should have them because they exist, because we have to say them.
Yeah. And on that note, you mentioned before that antibiotics have actually changed how medicine looks today. So Ava, do you know some medical advances that have only been possible due to antibiotics?
Yes, it's very important to think that, for example, things that we think are common and that we cannot even imagine medicine without them. For example, surgeries that extend the quality of life of our elderly, like it could be hip replacements. Those surgeries are not life or death surgery. They are just increasing the quality of the life, but they are risky because any type of surgery, anything that is opening a body and changing parts of the body has the risk of infections. So we need to have antibiotics that work in case these patients get infections. The same with this, with patients that are on particular risk of infections, like could be immunodepressed patients, like patients that have gone through transplants, or patients that are undergoing chemotherapy because their own immune system is not working properly. So they are more at risk of getting an infection from normal bacteria that normally don't get any harm to a healthy individual. Those are very important to have antibiotics as well.
Absolutely. And there's a lot of common medications nowadays that actually decrease our immune system, because oftentimes disease by an overactive immune system. It's not all good. So it's important that we keep these things in check, that we use antibiotics necessary.
For example, are also very rely a lot in antibiotics, and some of the bigger measures that they were talking about in case resistance gets really, really bad is to stop doing this kind of surgeries, because a planned C-section might not be a life or death situation. So they're only going to do it if they really, really need to, because they don't want to put the patients at risk of an infection that cannot be cured. But the whole thing is not as grim as we're putting it here, because we are actually doing something about it. And because we're here talking to you about bacteria and antibiotics and antibiotic resistance, we think that increasing the overall this topic is going to be the way to reduce the chances that resistance gets so bad that we cannot do these surgeries anymore, and that we are not going to be able to treat a regular infection like we are today.
So it is important to think about, okay, what can we do to prevent the use of antibiotics? As an individual, you can think about where your food comes from and how it's made. That plays a pretty big role. You can think about preventing infections. So if we help many parts of the world get clean water and all these other things, proper sanitation, less infections equals less antibiotics used. Good for everybody.
Proper diagnostic. We have technologies that are advanced enough to give us if an bacterial or viral, and within bacteria collectively and better. Yeah, so as you can see here, there are a lot of aspects to the issue of antibiotic resistance, but what you can do as an individual in your day-to-day basis is try to prevent the spread of bacteria as much as possible. And also, if your doctor is telling you you are not needed antibiotic treatment, trust your doctor, because that will be the best, both for you and for the common good of the antibiotics for everyone. And please, if the doctor says that you have to take antibiotics, I know that as soon as you started taking them, you're going to feel much, much better, but you have to follow the instructions and finish the whole course of antibiotics in order to also help out the spread of resistance or the accumulation of resistance in a sense.
So Eva, if people have been watching this video with us for a while,
yes,
what are some central things we want them to remember?
Yes, so if you those of a little bit watching this video, just pay attention now. We wanted you to know that bacteria are everywhere. The bacteria are majority harmless. A lot of them are actually beneficial for our health and our immune system. That they're really, really, really small and you cannot see them with your bare eye individually. You need a microscope for that, but you can actually see them with your bare eyes if you can grow the bacteria and get bacterial colonies. And then you can go out there and test where bacteria might be found. We also learned that antibiotics, which are medicines that can kill bacteria, originally come from natural products, and still some food might have some antibacterial properties, but we have to have proper antibiotics as medicines in order to treat bacterial infections. And we are able to also test in the lab if these medicines are working or not working. When antibiotics stop working, what we have is a case of antibiotic resistance. And this makes the bacteria survive an antibiotic treatment. This is quite dangerous because that will mean that we cannot treat bacterial infections in people that get really sick. And it also might mean that the medicine as we know it today with transplants, C-sections, cancer therapy, hip replacements, for example, might stop looking like we know it today. And therefore we need to make sure that we only use antibiotics when we really, really need it. I know we said this a lot, but this is very important. Just use antibiotics when they're needed.
Exactly. Good finish there.
Yes. Thank you so much for being with us. We hope you enjoy and that you learn bacteria as much as we do, right?
Yeah.
Okay. So thank you very much to Eva and Jenny for that pre-recorded video and such a nice example of how to look at antimicrobial resistance and how to demonstrate it and how to demonstrate how natural products can actually produce, can kill microorganisms and how antibiotic resistance has become so prevalent and what we can do about it. So let's just have a look at the time here. We've got a couple of minutes before we go into the next presentation. And the next presentation is going to be about culture collections in global sustainability and bioeconomy. And it's going to be given by Nelson Lima from the University of Minho in Portugal, but it's also a pre-recorded talk. But let's just have a look at the rest of the program for, it's now getting into the early morning here, but to early morning and the rest of tomorrow. If I can just share my screen briefly so we can have a look and see, so you can maybe pick things out that you would like to take a look at. So we've just seen Bacteria and Antibiotics 101. Got culture collections in the global sustainability and bioeconomy. Then I'm going to hand over hosting to Devanita Ghosh, who's going to talk about and chair the session on iron arsenic microbiology. We're going to have a little break, and they've got the super awesome science show host Jason Tetro with a panel of guests to talk about some really, really cool microbiological studies that have just been released, a range of stuff there. Then we're going to go to the vineyard. We're going to go to the Yangara vineyard in Australia, and we're going to get a tour of the vineyard and the winery as well, which is very, very interesting. It's going to be a live tour, and we tried that before with the brewery, and it seemed to go very well. So I'm very much looking forward to seeing the vineyard tour. Then I'm going to hand back to Joe. So we're going to go for the breakfast show in the morning, 7 o'clock Central European time. You can see the different time zones here. We've got America, I think it's Eastern Standard Time. And then what are the other time zones we have here? Eastern Standard, Central European, IST and JST. So 7 o'clock in the morning. And then we have traditional Japanese beverages, AMR, outreach soapbox, and then when we close, we're already over halfway through. It's gone extremely fast. So please pick out the ones you would like to go to, attend them live. If you'd like to ask questions, there'll of course be opportunities to see all the talks later on our YouTube channel. And please engage with us on social media. Watch there, comment, ask questions. We're really looking forward to hearing from you. And so without further ado, I'm going to go straight into the next talk now, which is the Culture Collections in a Global Sustainable and Bioeconomy by Nelson Lima.
Microorganisms is vital to everything that happens in the biological ecosystems of our planet. Microorganisms play key roles in human health and the environment, such as digestion of food and nutrient recycling. For centuries, this invisible world has been documented. Today, microorganisms are preserved and studied in what we call biorepositories, known as microbial biological resource centers that serve as libraries of microorganisms. Biorepositories all around the world help us exploit millions of microorganisms to understand how they function and use this knowledge to prevent or treat diseases and develop new technologies. That is why MIRI, Microbial Resource Research Infrastructure, was created in 2012 as a pan-European center to provide services that support research, innovation, and development in the field of life sciences and biotechnology. MIRI aims to ensure study and access to microorganisms is defragmented. Researchers can effect. To implement MIRI as a fully functional and sustainable research infrastructure, the European Commission funded Horizon 2020 project, IS-MIRI 21, Implementation and Sustainability of Microbial Resource Research Infrastructure for the 21st Century, was launched in 2020. Coordinated by the University of Minho, with 14 partners and 8 third parties from 10 European countries, IS-MIRI 21 intends to pave MIRI's sustainability by establishing it as an ERIC, a European Research Infrastructure Consortium, and instituting a set of services for MIRI's users to facilitate transnational access, or TN, to over 400,000 isolates of living organisms and their related information to the other. A central coordinated to manage transnational collaboration. A single entry point portal called Collaborative Work Environment to promote MIRI's expertise and resources. A higher education and training program for capacity building initiatives. An expert cluster platform to promote interaction among innovation actors and researchers. A larger international membership to enhance its accessibility. A robust financial sustainability plan to secure its longevity. Let me show you one of the ways IS-MIRI 21 will make a difference. Scientists from around the world can transnationally access thousands of microorganisms, microbial data, and higher educators share innovation through one single platform, CWE. IS-MIRI 21's outcomes ensure MIRI's mission is aligned with relevant European strategic agendas to advance research and innovation in biosciences, as well as increase recognition and appreciation of microbial life among its stakeholders. Through the collaboration of its various stakeholders, IS-MIRI 21 contributes to the enhancement of cross-national partnerships and the creation of a competitive and resilient bioeconomy in Europe and beyond.
The genome of all the microbes living inside and outside an organism is defined microbiome. The human microbiome is also known as second genome. Every anatomical site, including skin, gut, oral mucosa, glands, etc., has its own microbiome. We can say that human beings are colonized by microorganisms, since, on the whole, 57% of the cells are microbial. Beside the human microbiome is the soil microbiome. Consider that 1 gram of soil can contain up to 100 million microorganisms. And the plant microbiome that is located in the aerial parts of a plant, in the rhizosphere and roots. In all these cases, a good microbiome is beneficial to its own host. Microorganisms inhabit the oceans too. Herein, they're present either as single cells freely floating or as communities associated to vegetable substrates like algae or to organisms like corals and fishes. These communities make up the so-called marine microbiome that is still largely unexplored and that can also reside in the sand, in the sediments, and in the water columns. Environmental and human microbiome are strictly connected. A person with a bad diet will have a fragile gut microbiome and consequently a fragile health. Likewise, a person eating fruits and vegetables derived from plants and soils with a poor microbiome will not nourish his/her microbiome and will develop a fragile health. On the contrary, a person eating healthy food, rich in fruits and vegetables derived from plants and soils with a healthy microbiome, will strengthen his/her microbiome, developing a good health. Have a look at what happened to corals with bad microbiomes. They are all dead. The same happens in soil with a bad microbiome. Nothing grows. A new microbiome has been recently discovered, the plastisphere, or the microbial community capable of colonizing and growing on plastic. Plastic waste is literally suffocating our world. And as you can see in these pictures, also the oceans are dangerously full of it. The fact that microorganisms can grow and reproduce on plastics rudely abandoned in the environment means that they get nutrients from it. That is significant because it means a degradation of the plastic material is happening. Somebody will want to know who are the main actors that together build up a microbiome. Well, all the microbial categories are involved. Bacteria, microalgae, viruses, unicellular organisms or protozoa, filamentous fungi, and yeast. Now, a straightforward question will be, are all these microbes good or bad for us? We are used to consider them bad since we know them only for the diseases they cause. But this is not the whole truth. There are awful and malignant microbes, but also marvelous and fantastic ones. Let's get to know some of them. Among the worst bacteria, we can find the causal agent of flesh-eating disease, staph infection, meningitis, pneumonia, tuberculosis, cholera, and dysentery. There are diseases caused also by fungi and yeast. Fungi can affect the lungs, the brain, the nails, the oral cavity, and the guts. Bacteria causes diseases also in plants. They can damage strawberries and tomato plants and olive trees. Likewise, fungi are important plant pathogens. For example, they can affect grapes, cause race blast on rice, or devastating wilt diseases in many plant species. In addition, some bad fungi can produce mycotoxins, extremely dangerous compounds that can cause illness or death to humans and animals following exposure via inhalation or consumption of contaminated food. Despite what we've just seen, the number of pathogenic microorganisms is very low. In fact, more than 99% of the known bacteria, viruses, protozoa, fungi, and yeasts do not cause any disease and can rather be useful instead. Many microorganisms are used in food industries, in pharmaceutical industries, and in industrial biotechnology. Therefore, microorganisms, together with the compounds they produce, find numerous fields of application, ranging from agriculture to the production of biomaterials. Let's have a look now to some examples of beneficial bugs. For a start, we can have a look at acidic acid bacteria. Acidic acid bacteria can convert a number of carbon sources into biomolecules useful in food and beverages, chemical, medical, and pharmaceutical fields. They are commonly involved in vinegar, kefir, and kombucha production, and can be also found in other fermented beverages like wine, beer, and cider. Acidic acid bacteria can transform alcohols and sugars to produce a number of compounds that can be used in pharmaceutical industry. Lactic acid bacteria are mainly used in the production of fermented dairy products like yogurts, meat, and vegetable foods, but also sourdough. They're also used for bread fermentation to improve its properties and shelf life, and to delay bread staling. And here we are with filamentous fungi that we normally eat. Penicillium roqueforti is used for the production of a number of blue-veined cheeses, like Stilton, Roquefort, and Gorgonzola. Thanks to the production of enzymes, this fungus is involved in cheese ripening and flavor production. Likewise, Penicillium camemberti is the main ripening fungus found on the surface of soft cheeses such as Camembert and Brie. In 1928, Alexander Fleming realized that a mold later identified as Penicillium chrysogenum was capable of killing a wide range of harmful bacteria. His assistants, Stuart Craddock and Friedrich Riley, managed to isolate and purify the molecule responsible for this activity, penicillin, and so the era of antibiotics began. Fungi, as well as bacteria, can be used as agents of biological control to overcome the use of harmful pesticides. The secretion of metabolites with biological activity and the competition for space and nutrients with other pathogens leads to a reduction of pests and consequently to an healthy. What about yeasts? Thanks to the ability to ferment sugars and produce ethanol and carbon dioxide, yeast predominates in most of the traditional fermented foods and beverages that are present in human diet. No leavening of bread or pizza would take place without yeasts of the species Saccharomyces cerevisiae. And no beer or wine would be obtained if yeasts didn't use grape sugars and barley maltose to produce ethanol and a large part of the flavors typical of these beverages. We have now realized what single microorganisms can do. Together, in a community, in a microbiome, they can be stronger, more powerful, and useful. It is a united we stand, divided we fall situation. This figure tells us that every aspect of our lives is part of a unique whole. Therefore, our health is strictly connected to the environment health and to the health of all the animals that share the planet with us. It is thus extremely important to find a way to recover what has been damaged or destroyed, and the microbial communities will help us in this. A good microbiome is capable of remediating and restoring polluted sites. Bioremediation is an interesting approach that has a lot of potential and that has been already applied for the treatment of contaminated sites. Selected bacteria and fungi can together use and transform dangerous compounds like petroleum, polycyclic aromatic hydrocarbons, industrial solvents, pesticides, plastics, and metals. In light of a sustainable agriculture, fungi and bacteria can be used as fertilizers and, as we have already seen, as biological control agents and bioremediators. Microorganisms can also be exploited to produce biofuels. Microalgae are good candidates for production of biodiesel that so far rely on vegetable oil. Also, bioethanol is a good alternative and is produced through microbial fermentation of sugars and starches derived from agricultural and forestry residues. Microorganisms are also an alternative source of nutrients. Microalgae are rich in proteins, carbohydrates, fatty acids, minerals, and vitamins. Marmite, a spreadable cream so popular in the UK, is made of yeast extract, also rich in proteins and group B vitamins, while corn is rich of mycoproteins derived from the filamentous fungus Fusarium venenatum. Not only microorganisms like fungi are used to create biomaterials and eco-leather to make up shoes and clothes, but altogether they are an extraordinary source of bioactive molecule and useful enzymes. And finally, this is the BIQ house located in Hamburg, the first experimental building powered entirely by microalgae. Bioreactors are based on microalgae that, through the photosynthesis, produce all the biomass and energy necessary to power the building. The direct exposure to the sun increases the growth of microalgae, increasing in turn the quantity of energy produced. For all these reasons, it is necessary to conserve microbial diversity. And please do remember, to take care of it means that you care about yourself. Thank you.
All right, thank you very much to Nelson there for that very, very interesting pre-recorded talk. What I'd like to do now actually is we're waiting for another session to start. And it's going to take about 10 minutes, I guess. And while we're waiting, I thought it'd be really nice to go through the International Microorganism Day website, because this is really the best place to engage with us. And it's had a lot of work done on it over the past year. We've been contributing a lot. We've been upgrading and improving things. So I thought I'd just walk you through it a little bit. I mean, as you come to the first page, you see that we have the live stream here. That's obviously going to be important now for the next few days. So if you're ever wondering where to watch the live stream or catch up on what's going on, you just go to this link and you press play, and you'll be up to date with the latest in this marathon live stream. We've also got the program here as well. On the homepage, we've got links to our blogs, both our English blog, and we also have a Portuguese blog. International Microorganism Day was started in Portugal in 2017. And then we also have some of the featured blog posts and some more information about us and social media as well. So one of the things that we have highlighted a couple of times through the sessions already are the art. We've produced art for the blog, for the Women in Microbiology, but we've also produced art, or should I say you have produced art in the art contest. And you can see all of that in our gallery. All of it's online, and you're kind of free to use it, share it with attribution, and yeah, do whatever you want with them, basically. So we can maybe take a quick look here at the microbial art contest. So here you can see entries from 2021, 2022, yeah, they're currently being counted, of course, because we've only just announced the winner, but they will be there. And here are the entries from 2020 in all the different categories. And if we go back, you can see the pictures from past events on this page, but you can also see, as we mentioned earlier, the Women in Microbiology illustrations. And so these are in the gallery because these are all individual illustrations. You can use them on social media, download them, share them, do whatever you like with them. And they belong to a blog. And this is the other section that's had some work. In fact, I go to the blog last because there's one more thing before I want to go through the blog, and that's the educational resources. Last year, we introduced a Winogradsky column and a sourdough bread educational resource. There's a student's infographic and a teacher's pack. And this year, we've added 2 more. So we've added making yogurt and making agar plates. So if you want to try them out, if you would like to do any of those, then just download them. They're freely available for you to go ahead and do that. And finally here we have the blog. So the blog, we've actually been trying to recruit bloggers and trying to get more people writing, which has been going well. You can see we've got contributed articles if you're interested in contributing, you're more than welcome to send us an idea or a pitch or an article. If you're interested in learning about blogging or the format of the blog or skills that you need to start blogging, then we've also got a blogging bootcamp here, which I just scrolled past. And if you go to the post on the blogging bootcamp, you'll get all the information you need to actually join a free mini course where you can actually learn the fundamentals of blogging, and you'll learn basically how to produce a blog post for International Microorganism Day. So to do that, you can just go there and register your interest, and you'll be enrolled in the course. And then you can actually, if you think you would like to write it, think it's something you'd be interested in doing, but you're not sure how, then that could be a very good way to start. You just do the course, and then you kind of are taken through all the steps you need to write an article. So let's have a look. We've got some people in the waiting room now who I'm going to bring in. We're a little bit early, but that's okay. Let's see who we've got. We've got Devanita and we've got Sugata. Hello, Devanita. Hello, Sugata. How are you? Hello, hello.
Hi. Hi, Professor.
Welcome to the live stream. Yeah, I hope you're doing well. I'm going to hand over to you, Devanita, to host this next session. I'll be here in the background for any technical help if you want it, but essentially now the floor is yours. So thank you very much, and I will take a back seat now and hand over to you.
Yeah. Thank you, Joe. So this session is on the iron and arsenic microbiology, and since we know arsenic is quite a problem in many parts of the world, and iron plays a major role in the mobilization and especially the microbial action. So we'll hear about that from our two eminent speakers. They are global leaders in the field. And our first speaker, Professor Saugata Datta. So right now we have him online. So currently he is a distinguished professor, Hammond distinguished professor at University of Texas, San Antonio. And earlier he was in Kansas State University. Professor Datta has been working in this field for a very long time, along with the issues of water resources, water availability, and understanding the cycling of different metals and organic compounds. So today, with this, I would welcome Professor Datta.
Sure. Thanks, Devanita. This is a real pleasure. Can you hear me properly?
Yeah, we can.
Okay, great. Yeah, it's a real pleasure to be here, and thanks for the invitation to be a part of this speaker series of this eminent, FEMS. The idea that you chose is just excellent. So I'm very happy to introduce our work a little bit to all of you, and also initiate some discussions maybe towards the end if time permits and stuff. So let me try to see if I can share my screen. Okay.
Yeah, we can see that.
Okay, can you share? Sorry, can you see my screen properly?
Yeah, yeah.
Okay, great. So good evening, good morning, good afternoon, wherever you are. I know we are at 2 different continents. Here it is, what, 8:30 in the evening. I had my real long day. And then I taught from 4 to 5:15, and then I prepared my talk for this evening that Devonita had invited us for. So I'll be talking about the role of natural organic matter, or NOM, in microbially mediated mobilization, and keeping the theme of iron and arsenic in mostly these reducing aquifers that we have seen all across or all over the world, probably. This is not just the couple of case studies and ideas that I will place in front of you is from the Bengal Delta, but this is not just the only ones that we know of. So hopefully I will draw some attention from you on that. Okay, so the outline of my presentation is such that it is divided into microbial presence on Earth. Let me just switch off my phone, sorry. It's microbial processes on Earth, microbial iron cycling, how natural organic matter comes into play, and then microbially mediated iron cycling and its impact on human health. I think this part will draw some attention, and then a few case studies from our group that actively we are working on. Okay, so we know that microbes are present everywhere, and this is something that this audience is fairly knowing about, on the ocean or on the subsurface or in the soils. We have not gone into the ocean from our research group, but definitely we have dabbled into subsurface and soils. And these are considered as the major habitats of the microbial life, right? And relatively, a smaller population of microbes actually can live in minor habitats, as for example, that's where we work mostly on groundwaters, wastewaters, even in animals or plants. So this is something that we already are knowing this to go forward with our idea of knowing the microbial cycling of iron and arsenic, for example. But here I just present the global iron cycle. Again, this is iron being a very important redox sensitive element in the environment, which microbes actually cycle through both assimilatory and dissimilatory metabolisms, right? Both of them act together. Most of the cases, the autotrophic microbes obtain their energy by oxidizing the reduced form of iron and sulfur, whereas the heterotrophic microbes, they obtain their energy by consuming the carbon in the environment, right? So in turn, they also oxidize the iron and sulfur. So cycling of iron is very important in geochemistry, especially with this soil sediments, groundwater geochemistry, and is incomplete without really studying the roles of microbes. So knowing that microbially iron is cycled, microbes love the redox process, right? So as there is always a transfer of electron, and they obtain the energy for their survival from this electron transfer. Now, the chart to the right actually indicates that various mechanisms by which iron is cycled in the natural environment through the microbial oxidation or reduction reactions. So this is something that is important to keep in mind when we have that process of exchange of one electron that provides the energy to the microbes. Okay, natural organic matter. So the natural organic matter, as we all know, can be really divided into various fractions, right? As shown in the figure to the left. And this is based on the size of the molecules, the elemental groups, or the functional groups, mostly composition. For example, the biggest circle really is the total organic matter, right? I mean, total organic matter. And then we have the larger cycle of the DOM. Now, for example, a lot of the dissolved organic matter contains the dissolved organic carbon, right? The nitrogen, the phosphorus, which is based on elemental composition. Now, the DOM is also classified as either humic or non-humic substances, which also play an important role in the mobility of these metals or metalloids. In this case, one is iron for sure. Okay, so this picture actually shows the typical sources of, so we say that, release of organic matter from natural water. So as you can see, there are terrestrial sources such as, higher plants, aquatic sources, such as the microbes or the algae. And then figure to the right, to a little bit bottom, shows the various processes and transformations that this dissolved organic matter undergoes before we characterize it in a sample. Now, these processes could be either photochemical and chemical transformation, as well as it can be microbial processing. Now, while characterizing this dissolved organic matter in water samples, it is actually important for us, these results in the context of field results also, to compare them with. And that's what we do in most of our cases. So the figure this here, I'm hoping that this is very familiar to the audience here. It's kind of the size-based classification of the organic matter. Nominal size is that 0.45 micron is used to distinguish between particulate or the dissolved organic matter. But some studies have shown that the 0.7 micron or the 0.2 or the 0.1 micron filter sizes as well. So therefore, the distinction between the particulates and the dissolved organic matter is often, I would always consider, as operationally defined, right? And so you can see that the molecular weight of the various dissolved organic matter components are here, right? So bacteria and clay, for example, or clay humic metal complexes are sometimes particulate, and sometimes they are dissolved. Now, this classification can help in the DOM characterization that we do most of the times. Okay, so one of the research areas that my group are currently doing is working on microbially mediated elemental cycling and its influence on human health, or impact on human health. As we know that water quality is directly linked with human health, many microbial processes after the quality of the life by dissolving elements that are harmful to humans. And one of them we know of is arsenic, and I think you will hear a little bit about it after my talk also. So arsenic is a naturally occurring metalloid, and it's a carcinogenic too. It's considered carcinogenic, a class 1. And it occurs in soils, sediments, and they are mostly of geogenic origin. It enters the water systems by the sediment-water interactions and the microbial processes. On the top right, we show the Bengal Basin. That is, most of us have worked there. Major rivers of the Ganges, Meghna, and Brahmaputra is known to be arsenic affected. And the microbial processes here play an important role in mobilizing arsenic, which my group is mostly studying. But I would like to share some of the findings from there. Okay, so this is a very quick overview of some of the work that we have been doing in our group, mainly to understand the hydrologic, the geochemical, and the microbial controls of the mobilization of arsenic in groundwater. And we actually use all three, field, laboratory, and modeling-based approach to conduct these studies. Okay, so this schematic actually represents the biogeochemical processes that happens in a shallow reducing aquifer in the Bengal Basin, right? And in this case, the labile organic matter from the aquifer sediments serves as a food, as an electron donor essentially, for iron-reducing bacteria. Now, older or recalcitrant organic matter in sediments is relatively less labile for microbes to use as their food. Now, therefore, it was hypothesized that such humic-like organic matter can accelerate the acetotrophic iron reduction process via this electron shuttling. Aqueous complexation is there also, and then competitive sorption. That's also, other elemental ions like phosphates quite very easily demonstrate. So the microbial processes that are in the groundwater that follow a thermodynamic ladder, in this case, that's what is shown on the top figure over here. And these heterotrophic microbes can use the organic carbon as a source of energy, just like we eat and breathe, right? I mean, these microbes eat organic carbon and breathe not only the oxygen, but a variety of electron acceptors. For example, the oxygen or the nitrates or the manganese, iron, sulfur, and hydrogen, and in a sequence, like as it is shown here. So for example, oxygen is most favored electron acceptor for the microbes, whereas hydrogen is the least, right? And others are kind of falling in between. Okay, so having said that, we can see from the figure that as the oxygen is depleted in the system, then the system also becomes anaerobic, right? And in the Bengal Basin aquifers, we have seen that the sediments that are rich in iron 3 minerals, arsenic also often absorbed on these minerals. And that's where the arsenic relation comes in. When the microbes reduce the iron minerals and dissolve them in the groundwater, arsenic is also followed, meaning they are mobilized into the groundwater. So the supply of organic carbon into the aquifer actually dictates how much arsenic will be mobilized or solubilized as it supports the microbial life, right? So this schematic actually here at the bottom shows that various mechanisms rather by which microbes can mobilize arsenic in the groundwater that can use organic carbon as a food. Okay, so having said that, we have also looked at the microbial communities that are present in high arsenic aquifers in Bangladesh. This is a work from Bangladesh, strictly in MATLAB. In the picture, you can see that the sediments were collected from the aquifer, and then they were analyzed with evidence of the microbial presence communities. And we do observe that some of the bacteria groups are very well known to reduce iron 3. Laboratory experiments were also done with various strains of the iron reducing bacteria in presence of different types of organic matter, such as either like the acetates or recalcitrant organics like fulvic acids, to inform how the microbes can utilize the available organic matter. Okay. So over the last decade, several studies have been done that our group have done regarding the groundwater samples from arsenic contaminated aquifers from all the way from West Bengal, India to Bangladesh, and found that in general, the arsenic concentration correlates with the dissolved organic carbon concentration. However, there is still a heterogeneity. That means that some high DOC samples contain very low arsenic and vice versa. The question that comes or remains that, how can we explain this type of heterogeneity using the bulk DOC concentrations in samples? We hypothesize that to do this study, this is due to the reactivity of the recalcitrant the dissolved organic matter in the aquifer. So we'll try to prove that in our subsequent few slides. So there are several techniques that is used. Again, this is quickly showing that we use the absorbance and the 3D fluorescence spectroscopy to characterize the fluorescent organic matter in the water samples, and various functional groups we look at, but definitely the different field samples are analyzed with these. So here are the results from the field, right? So we found that iron bearing high arsenic groundwater, or iron bearing high arsenic contaminated, much higher proportion of terrestrially derived humic-like organic matter, which is likely to be involved in this electron shuttling and other processes that accelerate the microbial iron and arsenic mobilization. Okay, so having said that, the microbes sometimes use some smart ideas to obtain the energy, right, from either recalcitrant organic matter like humic or fulvic acids. And these humic substances are not very good food for the microbes to start with, and they are considered non-labile. So these humic substances can form the aromatic rings and can be used by the microbes as an electron shuttle. So this is what is shown in the schematic over here, and we can delve with that a little bit more later if wanted. But let's go to the crux of the matter. So, however, there was no this is a experimental evidence to show that there was no direct evidence that the Bengal Basin aquifers contain the humic material that is actually involved in the electron shuttling process. So in this part of the study, we actually performed a laboratory experiment using humic substances to isolate from the high arsenic groundwater in Bangladesh and use a geobacter as iron reducing bacterium to calculate the electron accepting capacity of the Bangladesh groundwater humic substances. Okay, so these graphs actually are shown to summarize the results from our work from a part of the Bengal Basin other than Bangladesh. It's also from West Bengal on the arsenic mobilization. And we observed that relatively younger Holocene aquifers contain the higher concentrations of dissolved arsenic, as shown in the red symbols over there on the map, on the plots. And also you can see these concentrations of iron, manganese, and dissolved organic carbon that are also elevated in some samples with high arsenic. Now, this is because iron, arsenic, and manganese are all released during this microbially mediated reductive dissolution process, which is fueled by the dissolved organic carbon. So the concentrations of some of the other co-occurring elements, like what I show on the plots to the right, actually is of concern, or some are not of concern right now, but should be monitored in the future. The graph that I show on the left bottom over here, that actually shows the close relationship between the dissolved arsenic and the dissolved organic carbon at multiple sites in Bangladesh. Also mostly in Bengal Basin altogether that we have studied so far. Okay, so let's go into the another ongoing study that we are doing. It's about the dynamic iron curtain, or also we call the natural reactive barrier, right? Now, these dynamic iron curtains are usually at the boundaries between the aquifer and the river, right? So you have the oxidizing river water and the reducing groundwater. And what I show here are a dry season, monsoon season, variation of this barrier, so to say, the expansion of the barrier, and then contraction of the barrier also. And then at the same time, we show that how these river waters are either in the form of groundwater discharging from the river to the actual groundwater rather, and upwelling the groundwater, or the other way around. So this type of natural reactive barrier is actually formed of mostly the high iron content, and so iron oxyhydroxides, goethite, even limonite, or sometimes iron oxyhydroxides are the main component out here. It's very difficult to analyze it. We had a paper a few years back talking about from the Meghna River, and we are currently doing a work funded by the NSF to look at that more critically, this presence of this natural reactive barrier. Okay, so what we did was we collected the pristine sediment cores from the Meghna River and also from the adjacent shallow aquifer with high arsenic groundwater. The locations of the cores are shown on the map on the top, and also in reference to the Meghna River, right? And the depth to the water table is given along the different sections of the cores that we are showing. So what did we do with them? Basically, these collected sediments were performed as a leaching experiment to mimic the mixing of the fresh river water into the aquifer. And these extracts of the sediments in ultra pure water after the experiments were done were analyzed for variety of inorganic and organic parameters. And I will try to kind of illustrate that quickly. So the results actually indicate that the sedimentary iron and the arsenic and two other redox sensitive elements like cobalt and vanadium in these river banks. Now you can see that about a 40-meter depth interval, or rather 40-meter distance from the river, we observed that these enrichment of the iron, which could be seasonal natural reactive barrier. So also we note that the enrichment of the arsenic and other redox sensitive elements slightly varied from the location of the iron. So not that all the places where the iron was high, arsenic was also high. So which may be attributed to the differential oxidation of iron and arsenic upon mixing of the fresh river water, right? That's more oxidized river water. Okay, so this is also another way of looking at it. It's more like of a colored diagrams to understand that where from the river that we see this type of iron enrichment, and as such what the iron enrichment is correlated with, as I was mentioning about that vanadium, cobalt, and also mostly iron and arsenic. So we also looked at the in this case, these are looked at the loss on ignition, and we show that there is a proxy, or loss on ignition is used as a proxy for the total organic matter content of the sediment samples that are found with some silts and clays contain the highest amounts of the LOI. Now, while the riverbank and aquifer sediments contain much lower in this case. Now, interestingly, we observed that the leaching experiment, that the riverbank aquifer sediments, although they contain the similar bulk organic matter, their water leachability was drastically different. For example, the aquifer sands leached much more organic carbon than the river sands. Now, we also should note that even though the silts and the clays, like in this case, the silts and the clays to the right-hand side, you see that as the silts and the clays are plotted over here, where the water leachable organic carbon content, even though the silts and the contain much higher organic matter content, they did not leach that much of organic matter during the experiment. So we did the fluorescent signatures of the organic matter that leached during this experiment that actually varied significantly between the riverbank sands, the silt, and the clays, right? So the silts and the clays in this case contained humic-like and terrestrially derived organic matter, whereas the riverbank sands, they contained the fresh, relatively labile organic matter, and fresh aquifer sands contain a mix of the two. Okay, we shouldn't say fresh, but at least to us, when we collected them, when we analyzed them, they were pretty fresh. Okay, so this is just a cartoon to show that these results actually suggest that the arsenic enrichment was present in the Meghna riverbank. Now, the cycling of the arsenic and iron in the riverbank appears that to be different in this case than the typical like shallow aquifers in the region, right? And this is an ongoing study. We are currently pursuing more additional analysis, including sequential extraction of these sediments. Some synchrotron analysis is going on, and also quantifying this natural organic matter and this iron arsenic complexes in the sample as much as we can. So this is where we would like to have a summary of our findings so far. Surely we see that the microbes do play a vital role in the cycling of the redox sensitive elements, in this case iron and arsenic, which in this case arsenic at least is an important human health factor, and to some extent iron too. And then we did confirm that the microbial cycling of the iron is influenced by the presence and characteristics of the natural organic matter, right? And the natural organic matter that reacts with the arsenic and iron in several ways, as I said, aqueous complexation or competitive sorption, or even with the microbes via the electron shuttling. So in the riverbanks of mostly the seasonally fluctuating rivers, like I showed in case of Meghna, partly Ganges is like that too. And these are all meandering rivers. The presence of the iron cartons are quite a bit, I would say, interesting to point out. And they actually show a strong influence on the hydrology that controls the iron and arsenic cycling. All these caps that you saw as the barriers, these are enriched in iron minerals. And we see the mostly the arsenic getting absorbed on this iron-rich barrier, if you may call, in between this aquifer sediments and the river. So the microbial cycling of iron perhaps influence the mobility of other oxyanion forming trace elements in addition to arsenic, like we showed for vanadium and cobalt, but definitely others need to be paid more attention also. Therefore, it is of prime importance in a human health perspective, again, not just for iron, but for the other elements that we just talked about. So with this, I will be happy to take any questions or any discussions to follow. Thank you.
Thank you, Professor Dutta. That was a very interesting talk, and definitely very interesting for me. I work on a similar line. I have pretty much a lot of questions, but I would also like to ask the host if we have any live questions from the audience.
We don't have any live questions yet. Sorry, we don't have any live questions yet, but I'll jump in if I get any. I'll either write them to you in the chat or I'll open the microphone up, but go ahead with your questions.
Okay, thank you. Thank you, Ben. So I have one question, to start with the humics in the deep aquifer. So because in one of your slides I was looking at, and you said that there is an increase, and then there was a shoot up after 30 meters or so. And that seemed pretty interesting, like what is happening in the deep aquifers, because then we can think of when we talk about the safe and the unsafe aquifers, and where with some places where the microbes are in action and some places where they are not, and that's why some of the aquifers, the Pleistocene are safer. So why are you seeing these sudden increase in the deeper aquifers, or are these humics, or?
Yeah, probably you are definitely. I would say that it's on the right track of thinking that probably the deeper aquifers may have a higher quantity of the fresher humics. Now, how do they retain back at that depth? After a reasonable time frame, like for example. But if you can really imagine that these systems are so much fluctuating with time, meaning that they are so dynamic environments that probably much more of this upheaval of the sediment, especially with these riverbanks and the aquifers together, with the course of changes of the rivers, and these are, as you know, probably many of these stalwarts in this direction have pointed out, that these are the paleo-oxbow lakes or the paleo channels, right? So definitely these are more exacerbated, more of the reacted sediments that way. So when we talk about these more reactive sediments and they are going into the deeper aquifers, I think that characteristics do change. And probably the addition of fresh organics also, like in this kind of terrains, are quite often very possible. And now whether they percolate down to those depths is a big question. I do not know. But I do know, we do see that there are a lot of impermeable caps, for example, like the clays in between. Now, if these clays are also playing the same or similar action like the organics is another thing to understand. So one of the parts that we did not pay attention to yet, but we love to, is this clay, NOM, and then this arsenic or iron kind of the complexation to understand. The organic carbon that is actually more hosted in the clays rather than organic carbon itself within the other mineralized parts of the sediments. So yeah, with that in mind, I think that's I think a lot more work can be done on this direction. Very good question.
Yeah. So this was a very interesting talk, and something that really caught my mind was the dynamic iron curtain. So yeah, this is really interesting what you're seeing in the riverbeds, and I would definitely like to explore more. I mean, right after just this talk, I think I would start reading on that. So do you see methanogenesis in
Yeah, not to that extent of a complete methanogenesis. No, it's not a complete, but we do see signs of methanogenesis do start to happen in some of these sediments. One of the things that we did do is we haven't been able to analyze them yet because of all the pandemic upheaval. Basically to date some of these sediments and then see that if which dates of sediments, and these are very freshly collected, right? So probably with the recent river upheaval or the floods that happened in the couple of years back, the few centimeters of the sediment has been trapped in that within that time frame, right? So I would say that in our geologic time span and the sediments, I mean, these are fairly recent sediments, right? But the proportion of the organic carbon is different. So we kind of diversified that to see that how much organic carbon or type of organic carbon. And then now we are going to date these sediments to see that, if these are really, and probably the C13 also will help us to understand, and also C14, that whether there is any correlation of this freshness of the sediment, right? And whether they are, whether really this reactivation of these sediments can lead to so-called making the sediments old enough to either retain or make the organics inactive. Now, I do not completely believe that it's a case, but I have seen in papers that they are analyzing. I mean, they are postulating for, like our natural reactive barrier work. I mean, there are multiple of work that has been done in other countries and other riverbanks, and they are seeing the same thing. The only difference probably is the proximity to the river. Sometimes, like we did pretty much a transect from the river to the aquifer, like a 80 meter, almost an 80 to 85 meter transect we did from the actual riverbank to the aquifer, right? So we do see a very prominent sediment characteristic, and along with that organic matter characteristic varying. When we had to sample the river, the river bottom sediments, I mean, the organic carbon basically is, I mean, it's full of organic carbon, let's put it that way, right? And these are heavily populated areas, so all things to be taken considered, right? But these are still on the so-called the river banks. We studied only the Meghna in this study, but we did also study with my Columbia colleagues up in the Brahmaputra, not on the very source area, but more towards the delta. And we did see much less of this iron barrier being forming. Meghna is critically important in this. I have not seen much studies on the Bhagirathi or the Ganges that way. But surely if we do study in kind of pristine areas along those rivers, we will find the same. That's our kind of aim of the future studies that we will try to take on.
Right, great. Thank you. Thank you so much, Professor Datta. We have our next speaker in line, but this was a very interesting talk, and thank you for delivering this.
Can I stay back for the next talk?
Yeah, sure. Of course.
Okay. Thank you very much.
Hello, Professor Sir. Good morning. You are muted. I think you have to unmute yourself.
Yes. Can you hear me now?
Yeah, we can hear you.
Okay, so let me share my screen and please, shall I go ahead?
Yeah, I will. So I will introduce our next speaker to the audience. Our next speaker, Professor Pinaki Sar, and he's from Indian Institute of Technology, Kharagpur. And he is in Kharagpur since 2004. And before that, he was the Boy Scout Fellow at Rice University. And then he also worked in some other institutes in India. He's also one of the leading arsenic iron microbiologist globally. And he has been working in this field for a pretty long time. So I have been following his work for, I don't know, since I started my PhD. So with that, I would like to welcome you, Professor Sar, on the International Microorganisms Day at FEMS. So,
thank you, Dr. Ghosh, for your nice introduction. And it is indeed a matter of great privilege to talk about the arsenic microbiology in this International Day of Microbiology. So I'm going to share my screen. So please let me know that if you are able to see my screen. Okay. Are you able to see my screen?
not yet.
Okay, so because we are not using this particular Zoom platform for our regular classes, so you have to just train me possibly. So should I go to the desktop or exactly where I have to go? So Microsoft PowerPoint.
So once you have opened the PowerPoint, and then you have to click the share screen on the bottom of this Zoom screen.
Mm-hmm.
And there's a green symbol on the
green thing is coming, but
yeah. And okay, once again, Ben, Professor Sar has access to share his screen.
Yes,
Yes, he does. So at the bottom of the window, yeah, just click on share screen and it should give you a choice of options which window you actually want to share, and then you should choose the PowerPoint.
Well, so when I go for this share screen, a couple of options appears: desktop one, whiteboard, iPhone, Google Chrome, Microsoft PowerPoint.
Choose the PowerPoint one.
Okay, PowerPoint, and then share. Then another dialog box is coming: allow Zoom to share your screen, cancel and open system preference. Okay, so maybe security and privacy to grant access. So I don't know. I'm sorry. How to go about this security? Ben, can you please quickly help me?
So you'll have to allow access. So when it gives you any options to do that, you need to allow access for Zoom to share the screen. So if you accept that, it should let you.
Okay. So how do I give this access actually?
Is there an allow button somewhere?
No.
Maybe you can share your desktop and open the.
Files and folders. So, is it visible now by any chance? I don't know.
No, not yet.
It's not coming.
Can you share your desktop and then open your PowerPoint on the desktop and minimize the screen?
Yes, it is already open. It is already open.
Okay.
It is already open. I'm just looking at the security. I think my Mac machine is not allowing me because it has some security issues, file and folder. So I'm really sorry about this. So let me just quickly.
No problem. If you want, you can send it to me and.
Okay. Shall I do that?
Yeah.
Yeah. So I don't know.
So we're going into a break after this session, so we can go over by 5 minutes or something like that, so we can get the time back. That's no problem.
Okay.
The file is very big, so I have to go by Google Drive. So I think then, Devnita, you can go, as Ben suggested, go for a quick break, and then I think by this time we'll be all set.
Okay.
No,
okay.
sorry, I was saying instead of going for a break now, we do after, yeah, because it will only be a short break now, so there's not much point sending people away and then coming back, but we can overrun a little bit. But what I suggest we do, we kind of do the transfer of the file now. And as soon as you've done the transfer, Dr. Pinakasar, maybe you can give a little bit of your introduction while Devonita is downloading the file and kind of setting it up, so we can just hear a little bit about the intro of your research, and then when you're showing the figures and the slides and stuff, then the presentation will be ready to go. Does that sound okay?
Yeah, I'm okay with that. So I'm just sending through, uploading in the drive, and maybe in a few minutes, Devnita will get it.
Brilliant.
I'm really sorry for this mess up,
problem.
but
We're seeing this all the time these days.
Yeah. So it's taking time. The file is little big.
No problem.
Definitely for a while, I'll just keeping myself mute and I'll try if I can do something.
So, Devanita, maybe you can tell us a little bit about your work and your research in the meantime. I mean, yeah, you're the host, but we don't get very much opportunity to hear from the host always. So that would be nice.
Right. Yeah, definitely. I will take this opportunity to talk about something I do. So I work very closely on what Professor Datta is working on. Working on the organics and how the organics are affecting the iron arsenic microbiology. So it's the biogeochemistry of, it's a very regional study, but my current work is focusing more on the arsenic iron microbial role, and then how iron specifically affects the co-precipitation of arsenic. Now I'm using the ideas into a mitigation method, say filters to clean up water. But earlier I have worked on the role of organics, say larger molecules, alkenes, alkanols, and then these are not very labile or very dynamic, but what Professor Datta is looking at, the smaller molecules, humics, these are more dynamic and then easily they break down and more actively participate in the microbial metabolism. Whereas the larger molecules, they are more recalcitrant, they stay in an environment for a pretty long time, and then they are also not degraded by the common microbes. So they kind of tend to impose a natural selection in the environment, and then the microbes who are able to grow and utilize these kind of the recalcitrant molecules, they kind of tend to get naturally selected, and they are more abundant, and then they tend to control the environment and the fluxes of different elements. So that's the idea that I am working on, and for pretty much a longer time, say 10 years. So right now in Delft, the work is more on the filtration based on the iron arsenic participation and co-precipitation. So so Ben, you have been hosting this since yesterday. I am like joining in and out on YouTube for like, I don't know. And I was like thinking that these two guys, Ben and Joe, must be very tired at the time because Joe, I know he's in Netherlands too. So
I'm actually in Sweden, so we're quite close.
Okay.
We're not as far away as we are, but and we're on the same time zone as well, which is useful. But yeah, it's been a long day. Yeah, it's kind of tiring, but yeah, we're doing this marathon, so there's got to be a little bit.
This is early in the morning, I think. What time is it? 4?
For me now, it is 4:15. Yeah.
Right. Yeah.
Yeah, so it's early.
Yeah.
But yeah, I did this last year as well, so I'm kind of used to it. So it's not such a big problem.
Okay.
Oh, what's this we see?
Yeah.
Oh, a presentation.
Okay, so we have Professor Sar's presentation here, and yeah, maybe Professor, you can go ahead with this. Thank you.
Are you able to see the presentation now?
Yeah, we are able to see, and maybe you can put it in a full screen mode, and yeah.
Sorry, Ben and everybody else for this mess up, but you understand that this is not intentional at all, and I am not used to this Zoom platform. I'm really sorry for this.
Of course, it's no problem. No problem.
Okay, so shall I move on and start?
Please.
Yeah. So today I'm going to talk on microbial world of arsenic contaminated aquifer within the Bengal basin, particularly our study area is West Bengal, India. And as you possibly know, that elevated groundwater arsenic is a global threat. And it is considered to be the largest poisoning of a population in history, with very large number of populations are under threat, like more than 200 million people. through drinking the arsenic contaminated water or ingestion of the arsenic contaminated food. And arsenic is also having very, very severe health implications. So now, if we look at the contaminated areas, the groundwater contaminated areas in particular around the world, we see that the South Asian and South American regions are the worst affected. And if we look at particularly the South Asian countries, it is estimated that over 190 million people are currently affected by arsenic contamination in the groundwater. And these contaminated aquifers are mostly hosted by sedimentary basins and close to the modern mountain belts and deltatic regions. And to be more precise, over the Southeast Asian contaminated regions, which are hosted by young alluvial aquifers of Himalayan origin, what we see that the rivers which are continuously feeding these alluvial landscapes in and around the places of Bangladesh, India, and China, and all the other areas, including Pakistan and other places. So they have contaminated the underground system. And in particular, with reference to India, what we see that almost like, except a couple of states, it is arsenic contamination in groundwater is now detected in almost every corner of the state of the country, India. Now, the major questions of geobiological significance remain: what is the mechanism of arsenic mobilization in the aqueous phase? And what are the factors which control this aqueous arsenic level? Now, these are two very fundamental questions, and perhaps it is now going to be 2 decades that scientists are trying to answer, microbiologists, geomicrobiologists are trying to answer these simple questions. And yet these questions are not completely answered, maybe because of the heterogeneity, because of the sampling issues and many other parameters. And what is important here to understand is that these answers to these questions will allow us to gain a deeper insights into the relationship between the subsurface microbial life and the physicochemical environment of the alluvial aquifers. Now, if we look at the source of this arsenic, this is basically a geogenic arsenic. It is basically derived from the rocks which are originated, or the minerals originated from the Himalaya, and they are deposited in the alluvial landscape within different arsenic-rich iron, manganese, aluminium oxide minerals, and they are hydroxides. Now, if we look at this simple cartoon, we'll be able to understand that the presence of arsenic within these iron oxide, hydroxide minerals is a very common phenomenon within this alluvial landscape. And arsenic remains mostly as arsenic 5 arsenate ion adsorbed onto these mineral phases. Now, over the years, what we have also learned that the microorganisms, they play a very critical role in transforming this arsenic, which is bound to iron, either directly by transforming arsenic itself or by transforming the iron molecule present within it. Now, it is considered to be significantly contributed by microorganisms, and the indigenous bacteria, they transform this arsenic basically through redox transformation, which allows the alteration of the arsenic speciation. And arsenic 5, that is the arsenate, which adsorbs strongly on the mineral surfaces, gets into the aqueous phase as soon as it is reduced because of the microbial activities. Now, when we look at this particular process, though originally the arsenic is from these volcanic rocks and other through the weathering and erosions, it is now a part of the alluvial landscape. And this arsenic, which is deposited within the secondary source, we called, because it is not the primary source of arsenic. However, this arsenic which is present at the secondary source is continuously being transformed by the microorganisms who are present in these alluvial aquifers. And these transformations, both the oxidative and reductive transformations, they carry out the relevant reactions which facilitate the release of arsenic under the conducive biogeochemical conditions. So the cartoon itself explains that these microorganisms of diverse types, they colonize, they naturally live within these arsenic hosting minerals. And these process of arsenic we call geocycling by these microorganisms, they are highly constrained by chemical and physical factors. And this could be one of the reasons that the complete role of a complete picture of the arsenic geomicrobiology, or the role of microbes in arsenic contaminated aquifer, iron arsenic transformation, is not completely elucidated because of the spatial and temporal variations which we encounter in these aquifer regions. Now, what has been clearly established that there is a complex interplay of geomicrobiological processes in arsenic mobilization. So here you can see that the iron oxide hydroxide minerals, they host the arsenic mostly as arsenic 5, and primarily the iron reducing bacteria, which are also sometimes called as IRB, they reduce this iron as a terminal electron acceptor and allowing the arsenate to be mobilized or dislodged into the aqueous phase. These arsenic 5 is subsequently reduced by bacteria who are called arsenic reducing bacteria, because these bacteria, they contain a particular enzyme which is called cytosolic arsenic reductase that allow these bacteria to reduce arsenic 5 to arsenic 3. On the other hand, there are dissimilatory arsenic reducing prokaryotes which can directly reduce the arsenic 5 as a part of their respiratory metabolism and lead to the release of arsenic 3 into aqueous phase. In both cases, the arsenic 3 is released into the aqueous phase, and possibly that is one of the reasons that the aqueous arsenic is coming through the microbial activities. This is pretty well known for past, at least past decades. But here is another process which actually takes the process in the reverse, like arsenic oxidizing bacteria. They utilize arsenic 3 plus, they oxidize arsenic 3 and converts this arsenic 3 to arsenic 5, and arsenic 5 get adsorbed onto the mineral surface again. So essentially, this is a kind of very intricate and complex process where both iron and arsenic reducing bacteria, iron and arsenic oxidizing bacteria play a role or play their roles in the heterogeneous aquifer environment. However, this kind of reductive dissolution that is actually shown in this cartoon, it necessitates the absence of oxygen, because if oxygen is there, possibly iron and arsenic is not going to be reduced, at least in the dissimilatory process. Supply of organic carbon is required because thermodynamically otherwise the process will not be feasible. And of course, we must have appropriate biocatalytic bacteria who can, especially bacteria, because role of archaea is yet to be clearly revealed in this kind of aquifer environment. So the presence of these appropriate bacterial strains are also very much important. So we initiated this piece of study, this particular part of the study, in order to investigate the role of the groundwater microbiome, because throughout the last 2 decades at least, we see that there are some attempts to take into account the larger populations which are present in the environment. However, most of the studies were done using selected bacterial strain or selected bacterial groups. Microbiome or community level interaction studies were relatively rare. So we took these arsenic bearing sands from the alluvial aquifer and allow the groundwater microbiome to interact with this. Now this possibly allows us to elucidate the nature and extent of the geochemical alterations impacted or imparted by the groundwater microbiome. And for the purpose of today's lecture, I am going to highlight how this groundwater microbiome would actually show us that how the taxa recruitment, different organisms are recruited specifically to facilitate this interaction with the arsenic, and also the role of organic carbon in that. So what we did, we took the water the arsenic contaminated groundwater, and we collected the entire pool of microorganisms. That is, we call it the microbiome is extracted, and the microbiome was allowed to interact with the aquifer sediment under laboratory anaerobic microcosms, and with no organic carbon added, middle one, and with organic carbon added. Now, this extraneous organic carbon that we have added is actually a mixture of lactate, benzoate, and pentadecane, because in our earlier studies, we have found that these aquifers are often having this low concentration of these aromatic and aliphatic hydrocarbons, and the microorganisms also have their potential to degrade different type of aromatic and aliphatic hydrocarbons. So following a 2 months incubation, what we observed that there is a significant change in the dissolved oxygen as well as the redox potential. So the dissolved oxygen showed that the conditions become more anaerobic, and the redox potential data suggested that it becomes truly an anoxic environment as the EH value goes to like close to a minus 60 millivolt or so, particularly when we have the organic carbon added into this microcosm. So this was coupled with significant bacterial growth, particularly if we look at the bacterial and archaeal growth through a gene transcript assay by extracting the RNA from these microcosms. You can see there is a two orders of magnitude increase in bacterial numbers from 10 to the power 6 to 10 to the power 8 in organic carbon added sediment incubation, whereas in no organic carbon added sediment also there has been some increase, but not very significant. The number of archaea, however, did not change very significantly under the similar conditions. Now, this change in the DO, change in the reduction potential and the microbial growth, all these things were found to be well connected to the release of iron, particularly as Fe2+ within the organic carbon added sample, although no organic carbon sample also showed some amount of iron release, but considering the presence of the scenario where we have organic carbon added, the concentration of Fe2 release was very significant. And with arsenic, the data was very interesting, because with respect to arsenic, we found that sediment with organic carbon led to a very, very high amount of arsenic release. And the total rise in arsenic concentration, if we consider, compared to only 65 microgram per liter into only sediment and microbiome, when we add organic carbon, it goes to like 400 plus microgram per liter, signifying the role of organic carbon in helping the microorganisms to possibly interact with the sediment and the minerals, and leading to the release of arsenic into the aqueous phase. Now, when we look into the speciation of arsenic, over a period of time, we observed that initially, particularly in organic carbon added sample, the arsenate and arsenite was these two levels were showing some interesting trend. Maybe in the initial time and also in the final time, we see that arsenate dominant, but somewhere during the intermittent period, it is the arsenite which is also detected, but in the elevated level, slightly higher level, which might indicate that arsenate, which is released initially from the mineral phases, are actually reduced to arsenic 3, and essentially arsenic 3 is more toxic than arsenic 5. So therefore, in a long run, the bacterial or the microbial strains or microbial species who are growing there, they are reducing this, or rather they are oxidizing this arsenic 3 to arsenic 5 again, and therefore the arsenic 5 concentration is again rising. Now, when we look at the solid phase of this sediment, we found that the iron oxide minerals, particularly oxides, along with the iron and arsenic, indicate significant changes. So I'm not going into detail of this, particularly the XRD data, which suggests the mineralogical changes, indicate that the highest release of arsenic is from the crystalline and amorphous, because we did a sequential extraction of arsenic from the different phases of the solid phase arsenic minerals, iron minerals, and we found there is a strong impact of the groundwater microbiome on arsenic iron oxide minerals like hematite, geotite, etc., as you can see with the XRD data. We also did the X-ray photoelectron spectroscopy to identify the relative abundance of this arsenic 5 and arsenic 3, which clearly confirmed that it is indeed the concentration, as you can see, that arsenate concentration from the control sediments towards organic carbon added sediment is significantly declining from 88% to 57%. We also noticed that the residual organic carbon, which was there with the sediment, is consumed by the microorganisms while they grow, they interact with the sediment. With the GC mass data confirm that the different level of hydrocarbon residues and organic compounds which are being consumed compared to the sediment alone, or the sediment with microbiome but no organic carbon. In presence of organic carbon, the residual hydrocarbons are all metabolized very significantly. Now, when we look at the microbiome, we found that there is a considerable change in the species abundance, like the operational taxonomic unit counts indicate that there is a significant change. But the more significant would be the taxonomic identity of the organisms. Now, compared to the groundwater microbiome, which is found to be mostly dominated by proteobacteria, and interestingly, Deinococcus was also there and few actinobacteria in the normal groundwater microbiome. But following incubation with sediment, the gamma and alpha proteobacterial abundance declined significantly compared to the normal groundwater microbiome. However, the abundance of bacteroidetes, which are anaerobic, known to be hydrocarbon and organic carbon metabolizing, but we did not add any organic carbon in this set, particularly only sediment and groundwater microbiome were allowed to interact. And finally, within the organic carbon amended set, it showed entirely different set of organisms, particularly the alpha proteobacterial members were found to be highly enriched or highly abundant, along with few members of Firmicutes, actinobacteria, and some other members. So if we look at the data at the genus level, we will be able to find out that compared to the control sediment, which was having some members like Acinetobacter, Brevundimonas, Sideroxidans, etc., the sediment microbiome, when microbiome was allowed to interact with the sediment, the archaeal member, Thaumarchaeotas, Sphingopsis, and some other members, Pseudomonas, etc., were enhanced. Particularly when the sediment microbiome, groundwater microbiome, was amended with organic carbon, the presence of rhizobium along with E. coli, Pseudomonas were found to be very, very, very abundant. It was very interesting to note that the members of the rhizobiomes were not generally found in the groundwater microbiome, at least at a significant level, neither when they interact with sediment alone, but they emerge as a major abundant, superabundant population when organic carbon is available. Now, we tried to find out the correlation with the observed geochemical or physicochemical parameters that we measured into the microcosms. With the major members, major operational taxonomic units, what we observed that the members like rhizobium and E. coli, they correlated very strongly, positively with arsenic, iron, and also with DO and ORP. Whereas some members like Sedimenibacterium, which was less known, and also the members like Geobacter, which are very well known with respect to their ability to interact with arsenic and iron perhaps, were not found to be that significantly important in this case, at least this correlation pattern indicates. Now, the quantitative assessment of the qPCR of different functional gene transcripts from the extracted RNA indicated the expression level of various arsenic homeostasis gene, as you can see, particularly the cytosolic arsenic reductase like ARSC, gone up to like 2 order of magnitude increase, and also the efflux, arsenate efflux pump, and also the cytosolic and the periplasmic arsenate reductase has also shown significant increase. The BSSA-like gene, which is involved in the metabolism of the hydrocarbons, was found to be very highly expressed, And this could be attributed because of the presence of the hydrocarbons that we added through the organic carbon amendment. It is also found that the alpha proteobacterial member like rhizobium that we found over there, rhizobium belongs to alpha proteobacteria. The alpha proteobacterial members were indeed very active, metabolically very active, because again, a 2 order of magnitude in copy number only for alpha proteobacteria was noticed. Now the phylogeny of these major OTUs from both the sediment microbiome set as well as the organic carbon amended set were analyzed. So this is the phylogenetic analysis of the selected top OTUs from the groundwater microbiome sediment incubation. And these, the small black circle indicates the relative abundance of these. So the summary of this phylogenetic analysis is that the iron, manganese, and other metal reducing geobacter and rhodonula type of organisms predominate when the groundwater microbiome is interacting with the sediment alone. Lactate, acetate, formate, fermenting. So fermentative microorganisms are also playing important role when the sediment is exposed to the microbiome, but of course, no additional organic carbon is there. Short chain fatty acids, C1 compounds, and different monoaromatic compounds utilizing organisms are prevalent, and ammonia oxidizing or nitrate reducing strains or species were also found to be abundant within this group of organisms which are possibly recruited by the microbiome when they are exposed with the iron oxide type of minerals. Now, the similar analysis done with the groundwater microbiome sediment organic carbon amended set indicates something very interesting, that the rhizobium type of members that we found. So most of the abundant OTUs are affiliated to rhizobium. So a bunch of OTUs which are very abundant, up to 10% like that, you can see they are all rhizobium. And what is more evident, that these rhizobium-like OTUs are closely related to two other rhizobium species previously reported from different arsenic contaminated and selenium metabolizing ability. So this was very interesting, that facultative anaerobic arsenic, iron, and selenium reducing and hydrocarbon utilizing rhizobium species were surely there playing a very important role. Sugar and acid fermenting E. coli, lactobacillus type organisms, and polymer and hydrocarbon utilizing arsenic, nitrate reducing Pseudomonas, Thoira, etc. were also found to be the major populations when the microbiome was supplied with organic carbon. Now, we have also isolated a number of strains in order to validate that indeed these rhizobium type of members, they play a role in arsenic mobilization or in interaction with the arsenic bearing sediment. So we have isolated a couple of rhizobium strain from this organic carbon amended sediment. So I am just highlighting that part of the data. And what we see that these rhizobium isolates, so these are all rhizobium, different strains of rhizobiums isolated from the organic carbon amended set. And they show a very, all of them, they portray very significant lineage with these, again, this rhizobium arsenic reducens, which was previously isolated from arsenic contaminated groundwater, and it is capable of utilizing hydrocarbon and as well as reducing arsenic and iron, and also the selenium reducing rhizobium strain. It is also interesting that these isolated bacterial strains showed a very good agreement, phylogenetic agreement, with the OTUs, rhizobium related OTUs, which we detected through the cultivation independent approach. Thereby, we were able to actually claim that we are now cultivating those organisms which actually predominate the real the community when they are allowed to interact with the sediment in presence of organic carbon. Now, these rhizobium members were further studied for their abilities to utilize or reduce iron and arsenic, both as from the solid as well as the soluble phases. And it was found that they are both capable of utilizing the soluble as well as the solid phase arsenic and iron, and they accumulate those arsenic somewhere around the cells, as the ACM EDX data confirms the relative abundance of arsenic into the cell mass which is generated. So particularly the rhizobium species, those are tested, found to have the ability to almost like 80% reduction of the soluble Fe3 and AS5 and 40% reduction of the solid phase arsenate and iron, particularly when we provide the organic carbon as a source, carbon and energy source in the microcosm. So based on this study, we conclude that the groundwater microbiome of arsenic contaminated site are capable of interacting with the arsenic bearing aquifer sediment through specific engagements of microbial species. So there could be hundreds and thousands of species present in the microbiome, but it's not true that all the species are equally recruited by the community. So it's a community's collective decision possibly that they are recruiting few taxa when only sediment is available, or when along with the sediment we have the organic carbon available. The species recruitment was geochemically constrained. Particularly, organic carbon has a very important role in impacting the microbe sediment interaction. This surely validates the existing knowledge that organic carbon is playing a very important role, and it is easy to understand because it provides the thermodynamic resources, energy, the electrons, the carbons. Everything is going to come from this organic carbon. It is also found that the role of multipartite microbial processes, including the anaerobic utilization of hydrocarbon, which is found to be very unique in this case, because in other studies, we do not see much of the reports that hydrocarbons are being utilized during this arsenic dissimilatory reduction or arsenic mobilization processes. Microbial cytosolic arsenic reduction, iron transport, along with metal bioreduction and bioweathering of the clay minerals were also observed. Now, we also demonstrate the importance of the chemolithotrophic and heterotrophic members separately, perhaps, because during the no organic carbon amended condition, it is mostly the chemolithotrophic organisms who are playing important roles, whereas during the presence of organic carbon, it is interesting to note the role of rhizobium, which is perhaps not so much explored with respect to its potential in helping the release of arsenic. Along with E. coli and Bacillus. Bacillus members are already known to play a role in arsenic transformation. Geobacter, another very well-known species or member, is also found. And it denotes that the anaerobic hydrocarbon metabolism coupled to arsenic reduction by both cytosolic and respiratory reductase-based activities dominated the rhizobium and Bacillus members. And this part of our work is recently published in the Journal of Hazardous Materials. And with this, I end my talk. And I acknowledge my funders, the Department of Biotechnology, Government of India, IIT Kharagpur, CSIR, Government of India, Department of Science and Technology, Government of India, past and present students, and all the collaborators and the FAMES for organizing this meeting and giving me the opportunity to present our work to you. With this,
thank you, Professor Sir. Thank you, Professor Sir, for a wonderful presentation. And this is really loaded with information. Ben, do we have time for one or two quick questions?
Absolutely, absolutely.
Okay, Professor, I might have missed, what is the organic carbon you are adding in the experimental setups? You are muted. Yeah.
We are adding a mixture of lactate. We are adding pentadecan, and we are also adding some aromatic hydrocarbons. The aliphatic, aromatic, and these sugars are all there.
Okay. Okay. And do we have a study of, like, once the organics are exhausted, how the microbial community changes?
When we stopped the experiment, that is after 2 months, our GC mass data showed that organic carbon is nearly exhausted, though we are not claiming that we're not claiming that the organic carbon is absolutely exhausted. Okay, but that can be another interesting study that given a chance that the organic carbon was initially available, then it was completely depleted, how the community behaves actually. So
yeah, I guess once the carbon get exhausted, and also with the time the iron and arsenic will also get removed, like maybe co-precipitated and removed. Then because the toxic levels are reduced, maybe the other communities will pop up. Like, as you say, the taxa recruitment changes with the structure, with the microenvironment. So
so that is absolutely expected.
So if I'm curious, you have seen any methanogenesis in your study?
Methanogenesis, no, but the presence of Thomarchiota in no organic carbon added condition.
Okay.
That we have noted that during with no external organic carbon added, Thomarchiota members were there. So possibly some amount of methane may be generated, but one thing we have observed that C1 compound utilizing organisms are very abundant. So whatever low concentration of methane is generated, and you might be aware that recently the ONGC, they have found oils, petroleum oil reservoirs, and very soon the exploration of oils are going to be started in this region. So it is expected that methane would be there somewhere. And this methane may not be as a methane gas, but methane is transformed into alcohols. So it's basically the methanols and other C1 compound, methylated compounds. So we see a lot of bacterial strains who are capable of utilizing C1 compounds.
Right. Okay. Okay. That's pretty interesting. Okay. Thank you so much, Professor Sir. Ben, do we have any questions from the audience?
No, I didn't get any questions. But yeah. So yeah, with that, I mean, I'd like to thank you. Well, thank all three of you. I see that Saugata is also still here. So thank you very much for your contribution and for a very, very interesting session. And thank you for persevering with the technology. You know, it's not easy kind of in these kind of high pressure meetings like this, but I appreciate you kind of working through, persevering. We actually got to hear a lot of really, really nice information. So yeah, thank you very much. And I'll say goodbye to you now, and we'll move on to the next stage.
Okay, thanks a lot, Ben. Thanks
Thank you, Ben. Thank you, Devonita. Thank you very much.
Thank you, Professor.
Thank you.
Yeah, thank you. Bye.
Bye-bye. Bye-bye. What we're going to do now is actually going to have a short break in the live stream. So we're going to take, what is it now, 45 minutes? So we're going to come back at 1:30. So in European time, Central Europe time, that'll be 5:30. And we'll be back with a new host, Jason Tetro, who hosts the Super Awesome Science Show, and he is going to be discussing some very interesting, very, very new scientific data with a panel that he has chosen. So I'm very, very, very much looking forward to that. So I'll put on the holding card now, and I will see you in 45 minutes. Thank you very much. Hi, Jason. Hi, Claire. Can you hear me?
Hello there.
Hello, hello. Can you see me? Can you hear me okay?
Yes, I can see you and I can definitely hear you. How are you?
Fantastic. I'm good, thanks. How are you?
I am very good. How has microbiology day been coming along?
It's been going well. Yeah. It's we've had a lot of diverse and interesting topics being covered here, so it's been really fun. I'm definitely looking forward to this session now.
Fantastic. If you're good to go and everybody's happy, we can just get started.
Yeah, we are good to go. I'll basically hand over the floor to you, Jason.
Thank you.
Yeah, it's yeah, and I'm here if you need anything in the background. But yeah, Jason Tetro. I mean, you know, best-selling author, host of the Super Awesome Science Show. Take it away. We're very happy to have you.
Well, thank you very much. And thank you everybody for being here on this amazing International Microorganism Day. And I have to tell you something, when Ben asked me to be a part of this, I was just super thrilled, as I normally am when people ask me to do things live. Normally I do stuff taped, my podcast is taped. But there's something about doing something live that is just amazing. It feels like you're always doing an experiment. And for those of you who are in the lab, you know that feeling, right? You got the nerves going. You're kind of like, oh, I'm hoping it's going to go well. It's really awesome. But I do want to tell you something. And I mean, I know it's a late night talk show and it's a, you know, we're supposed to be happy-go-lucky all the time, but I'm kind of feeling a little bummed out. And you know what? Why don't I just show you why I'm feeling a little bummed out, okay? You see, we want to be living and loving our microbes. And Ben, can you see that? Excellent. And so one of the things that I've been doing for so many years is just trying to get people to sort of figure out how to have that relationship. Now you have to understand something. Relationships are hard, and I totally understand that. We have so many relationship therapy models. Getting the love you want, he's just not that into you, therapeutic attachment. The reality is the goal is really to best describe how the relationship exists and then figure out how you're going to be able to do that. And when I started writing my books, that was the first thing I wanted to do. And yes, the books are really a how-to on how to love germs. You got to get used to it. And really what it came down to was, if you remember in 1991, there was this wild book. It was called Men Are From Mars, Women Are From Venus, right? And what I did is I thought about it and I thought, okay, well, if we are humans and we're from Earth, well, where would germs be coming from? And we thought Mercury, maybe Jupiter, Saturn, Uranus, or maybe the planet formerly known as Pluto. Still upset about that. And we realized something. All you really need to do is just change the way that you pronounce something. So humans are from Earth and germs are from Uranus. And for those of you who don't speak English, it really is funny. Just believe me, okay? Anyway.
It is.
But the fact of the matter is that despite that microbes are in our GI for the most part, they do happen to be everywhere. And we do have this microbiome. And I think the one really cool thing about the microbiome that everybody really needs to understand is that the more that we get a feel for what the microbiome does for us, then we can understand how we can work with the microbiome. And a few years ago, I actually co-edited a book on this, the Human Microbiome Handbook. And at the same time, I was actually writing the germ file. So you've got the academic version in the one book, and you've got the public version in the other. And the whole idea was that whether you're a scientist or whether you're just someone walking off the street, you really want to figure out how you can create a love story with germs. And let me tell you something, it was going well. It was going so well. And then this guy came along. I'm sure all of you know who that happens to be. SARS-CoV-2, also known as the causative agent for COVID-19, coronavirus disease 19. Well, this thing just kind of messed everything up. It's a late night talk show, still not going to swear. But what ended up happening was that the masses of people who truly were starting to understand the relationship, truly understanding how we can love microbes, started running away. And where did they run away to? Well, yes, the underground, otherwise known as we hate microbes. But I'm showing the underground for a different reason. And that was after COVID came out, there was this very interesting paper that was published in the New England Journal of Medicine, which talked about how the virus could possibly survive on surfaces for a very long time. Interesting, I used to do the exact same type of research. It was a laboratory controlled study, but it kind of blew up in the media. And all of a sudden, well, we had people wandering around looking like Ghostbusters, except they were germ busters. And they were going around spraying everything. And the whole point was to try and spray everything that we were touching in the hopes of getting rid of COVID-19. There was only one slight problem with that. I know that we're probably blocking it a little bit, but if you look over here, you're going to see a little animal holding a hand sanitizer bottle. The reason this is important, that's a Banksy. Now remember, Banksy has sold paintings for like millions and millions of dollars prior to shredding them, but anyways. And the fact is that they actually got rid of Banksy paintings while they were doing this hygiene theater of trying to disinfect surfaces. It was a nightmare. But more importantly, it took away the one thing that we should have been paying attention to, which was the fact that the rats were showing us what we really should have been afraid of. Now, when I first showed this, I actually called them droplets. We now understand that it's probably airborne. So I'm just going to use the term aerosols, but by the same respect, it all means the same thing. Surfaces, airborne, absolutely. And of course, we did eventually figure out how to protect ourselves, right? And of course, you know, it is England, so they're going to protect themselves any way they possibly can. But the fact is that you have to understand that when you see this happening, you realize, okay, I've got to start thinking about how I'm going to be able to protect myself, but also do it in a proper way, which is, of course, the whole idea that I'm talking about when we're falling in love with our microbes. And here's where it gets really, really scary. I don't know about you. Have you ever taken out something from a restaurant and it goes into this takeout bag, otherwise known as a takeaway bag? Did you know that this poses a risk for you to get COVID-19? Why? Yes, according to that New England Journal of Medicine article, it does. But let me tell you something. If we do a risk assessment, it's a very low touch surface. It has no contact really other than the handles. And it doesn't really know, it's not really known to have any surface transmission from one person to another. But the most important thing you need to know is that it's not the bag itself that poses a problem, it is the food. Foodborne infections are the most common type of infections that we face. And that's one of the reasons why we have food hygiene ratings when it comes to restaurants, but I don't know about you, I've never seen a takeaway bag rating. This is what it means to have some kind of relationship with microbes. And just so you know, if we do end up actually getting a takeaway bag rating, it probably will be Germ Guy approved. And that's one of the reasons why I find myself so bummed out. And now the really interesting thing that you have to think about is the fact that when we do talk about our relationship with microbes, we do figure that it really is about us and the species that cohabitate that three-dimensional space with us. And we are going to get into that a bit later. But I do want to ask you all whether or not you believe that these microbes can have an effect not on our human to microbe relationship, but our human to human relationships. Now, this is a question that I asked someone a while ago, and it was in a very different context. But what I did is we talked about it on the show, and it was one of the best segments that we ever had. Like, it was just so good. I had people writing me going, oh my God, who is this person? We love her. And thankfully, not only was she loved in Florida, but she was also loved in the great state of Tennessee. And so I've asked her back for this particular show so that we can have a discussion about the aspect of relationships that germs have, not necessarily with us, but what the effect is on us and how that creates a relationship with others. And it's for one very important thing. Are you sure? It's the way we smell. Please help me welcome to the program, Middle Tennessee State University Assistant Professor, Dr. Jessica Gaby. Hi, Jessica.
Hi.
How are you?
Great. How are you?
I am absolutely fantastic. Thank you so much for joining us on the show. I mean, the last time we talked, we had such a great time. And honestly, you were the first person I thought of when it came to having this show. And so I'm so thankful that you were here and you're able to join us. I know it's a little bit late, so also thank you for that. Now, one thing I do want to bring up is that normally when I introduce people, sometimes I give them a little bit of a background on the history, et cetera, et cetera. And I just called you an assistant professor. And the reason I did that is because I want to start off our conversation with a very important point. You're not a microbiologist.
I am not.
Tell us, no, exactly. So tell us about your research program.
Yeah, so I study how the way that we smell impacts our social relationships with one another. And specifically, I'm interested in platonic relationships. So a lot of the studies that look at body odor have been done on mate choice, but that is such a small percentage of our daily interactions. I'm really trying to look at how does the sort of odor personality that we build for ourselves in real life impact other people's impressions of us. And so I've looked at that from a couple of different perspectives. I've looked at how perfume might impact it. I'm currently working on a study looking at how diet might impact that. And
I mean, come on, diet should already do that. Regardless of microbes, you eat garlic, it's going to be hard to really,
well, see, you would think that. You would think that, but the only study that has been done on the influence of raw garlic on body odor showed that people like the body odor more after people ate raw. It was men. It was women smelling men. So again, caveat that it's like a specific subset of judgments, right? But they found that the body odor of the men was rated as like more pleasant and more attractive after they ate, like, I don't know, I think it was something like 4 cloves of raw garlic.
I almost feel like I should have read the book Perfume before I started talking with you again. But before we get into any of the discussions about that, I do want to go back a little bit back to your PhD thesis from 2017. And the reason I want to do this, people, is because this is what it actually is called. Invisible Influence, the Role of Human Social Olfactory Cues in Ecologically Relevant Interactions. I don't even know what the heck that means, but I love it. So can you please just sort of take us through what that means and what it was that you actually sort of found out during that research?
Yeah, so, I mentioned earlier that I study these platonic interactions, right? And one of the main reasons that I do that is because I'm really interested in how these cues are functioning in real interactions, right? Because the majority of body odor research has focused on axillary sweat, so armpit sweat collected on either t-shirts or pads. And then sometimes it's even the body odor, the sweat of like multiple individuals combined. And when you come into the lab, so when you come into the lab, what you're smelling is just armpit sweat. And you would have to know someone pretty well to get unadulterated armpit sweat, because this is not just regular armpit sweat. I asked you to stop wearing deodorant for 2 weeks, and then I took your armpit sweat, right?
This is giving me memories of that movie Flirting with Disaster, where the guy literally had a fetish of licking armpits. So
well, you mentioned that movie Perfume. If you want to get into like weird small fetishes, that's the one.
That's a good point. Yeah. Also late night television, guaranteed. Okay.
But yeah, so, my sort of interest in body odor revolves around, well, okay, we know the way that this body odor functions in a lab when people have investigated this very removed type of odor sample, right? If I ask you to stop wearing deodorant for 2 weeks, I ask you to change your diet so you stop eating aromatic foods, chili, curry, garlic, asparagus. I ask you to wash only in unscented detergent and soap and shampoo and everything. I provide you all the things, right? And so I've eliminated all of these odors that actually especially in America, are really important to the way, I mean, not especially in America, across the world, right? Fragrances are important. And it's weird because America, you know, the American goal for body odor is to smell like nothing.
Yeah.
But that's not true in other places. And so what I want to know is, okay, we know these phenomena happen in the lab, but does it break through into real interactions, right? And this is like a whole different beast now that we're in sort of social distancing, masking territory. So,
oh yeah, absolutely. Now, the thing that I get a kick out of from your PhD thesis is that you were one of the first that I know of who was doing the t-shirt experiments that we're all familiar with, right? Like the MHC 1 and 2. Oh my God, are you attracted to someone because of the t-shirt? You actually did that. But more importantly, you did that with live donors. Like this wasn't you just collecting their sweat and then putting into a t-shirt. You actually got them to sweat into a t-shirt. How does that work? Like, do you just get them to sweat and then they take them off? Do you have like sexy music and slow motion photography? Like, how does that work?
I did do a study where I had to have a lot of women undress in front of me. It's not published yet. We're trying to get more funding to extend this project so I can get more women to undress in front of me. In that study, that study is actually a really great one for the topic tonight, but I want to just briefly cover. So the 2017 study, what's unique about it isn't the fact that we use t-shirts. What's unique about it is that we had people smell each other in real life, right? So we had a bunch of individuals in separate rooms, and they had blindfolds and earplugs, and they got a series of visitors who would come and sit down next to them. So they would cross in front of each person, sit down next to them for a minute, and then cross in front of them again and leave. And then the person would take off their blindfolds and answer a bunch of questions about the visitor that was just there. And so what we did in that study is we compared, we did a couple of things. We compared, first of all, if you meet the person a second time, do you get a similar impression of them? The answer is yes. Our impressions are very consistent in this context of this, what we would term a live interaction, right? So a more ecologically relevant interaction, something that's more akin to the kind of interactions we would have in real life, where maybe you would be sitting next to someone on the bus or in class, right? And then the other thing that we did is, we compared that perception to smelling someone's t-shirt, and we asked, well, how similar is it if you smell their t-shirt versus if they sit next to you? And the answer is similar, but not completely the same, right? Obviously, there's a lot more richness of information in a real life interaction. And then the last thing we looked at is whether or not we perceive people differently if we do have them do this removal of all the external odors,
right,
versus letting them show up the way that they really smell. And the answer is there's a drastic difference in the way that people perceive an individual if they come smelling the way that they normally smell in real life versus if they've done this, what we call a washout.
So in this, what I called in the show speed smelling instead of speed dating, do you feel that may actually have a much more influential perspective when it comes to not necessarily finding a mate, but as you said, just platonically hanging around one another? Oh
yeah, absolutely. So I got into this work because there was a person in my friend group who to me smelled like very aversive. I was never able to develop a close emotional relationship with this person because they smelled so bad to me that I couldn't get physically close to them, right? And so I started thinking about just how important these cues are in our social interactions. And obviously there's a big range of impact for different people. There's a lot of individual differences. I'm a person who thinks a lot about smell, so it's probably more important to me than to some other people. But certainly the sense of smell impacts us on a daily basis in ways we really aren't necessarily consciously aware of. And I'll tell you the other thing, which is that like whenever I tell somebody that I study body odor, the first thing they say is, oh man, there's this guy in my office.
I mean, there's always a guy in the office, right? That sort of brings me to the next question. I want to get to the women undressing in a second, but before we do, you are so passionate about this. Like even now, just talking with you, I'm just listening, completely enwrapped. What is the motivation behind this other than to try and find out why you couldn't have a relationship with someone because of the way they smell? Like,
I mean, I think what is exciting to me about the sense of smell is that we still know so little about it. So I was sort of going back and just reading some current papers in preparation for this. And it's so interesting to see how everyone's like, well, we don't really know the mechanism for how this works exactly. Like we don't know what it is in body odor that's important right? We're not sure from a chemical standpoint exactly which signals they are, and we don't really know how to measure them. It's just sort of looking into this abyss of unresearched space, which is very different from most other sensory systems, right? Vision is very well explored. Audition is pretty well characterized. And so the sense of smell is just wide open. And because I'm interested in this tiny little avenue, right? These platonic interactions that most people aren't looking at. For me, I can even look at a study that was done, recently where they had men, women smelling men. And I can say, well, okay, but does that work between women? Does it work between men? No one knows what works between men because almost no one studies male-male interactions, including me. Although, hopefully it's coming with more funding.
And do you actually because I want you on the show because obviously a lot of the smells are made by the microbes themselves. And that is really interesting. But I haven't seen yet anybody try and translate the microbiome to the sense themselves. And I think you kind of already answered that. We don't even know what the mechanism is. So
I love that you asked this because last year, pre-COVID, I did a really cool study. I started a really cool study when I was at Penn State as a postdoc. And we looked at the impact of a dietary manipulation. So we asked a bunch of Caucasian women who reported not eating curry on a regular, less than once a month. They had to say once a month or less. We had them eat curry for 28 days, and we took their body odor before and after they ate this curry. They had to come into the lab every single day, and they would eat just a quarter cup of rice with it's 10 grams of curry powder in it. So it's a pretty large amount.
Did you give them an extra toilet roll when they walked out?
No one really reported gastric upset. So what we were trying to do is sort of mimic, okay, if you ate curry every day, how much curry would you get in a full meal? And we wanted to not impact people's dietary intake because that might have its own influences. So we sort of tried to condense that into the smallest serving size possible that was still kind of, again, ecologically relevant. So we fried the curry powder in butter to make a curry paste, and we mix it with rice, which is mimicking at least some of the preparation steps that you would do if you were going to really make a dish of curry. But we really wanted to minimize the other influences, right? So we didn't add salt. We didn't add something like coconut milk. We told people they could add stuff on their own, but we really tried to so they ate curry. So these women who don't normally eat curry, we had to make our own spice bun because there's no curry on the market that's not spicy. And I was worried that if these women weren't eating curry, they would also not like spicy food. I didn't think I would be able to get them to eat a bunch of spicy curry every day for a month. So we collected their body odor on t-shirts, but also because we know or because we expected that diet would be linked with changes in body odor, we collected their skin microbiomes to look at whether or not we would see a shift in microbiome in conjunction with this dietary manipulation.
Okay. Well, don't just leave us hanging. Did you find anything? We got 2 minutes. We got 2 minutes left. I wanted to get to the naked ladies and also to the vanilla.
So these are the naked ladies. So every time, we actually took their body odor every week, and every week what they would do is they would come into the lab, and I'd apply. So we were also trying to collect skin volatiles, which it turns out is just really difficult to do, and we weren't very successful in measuring it. But we would put a little skin volatile collecting strip on them, and then we would have them, they would put on a new t-shirt, they come back the next day and they take the t-shirt off, and I would swab their skin and I would take their little volatile strip. And so every week, all of these women would have to come in and undress in front of me. And I was doing it, in a space. And anyway, what we found is that honestly, our results are inconclusive, and that's why we're applying for more funding. We found a borderline influence where people who hadn't eaten the curry. Right. So we asked the curry eaters to rate each other as sort of a measure of in-group familiarity. And then we asked a bunch of people who are unrelated to the study. And we found that for those people that were unrelated, there's a borderline effect where they find the body odor slightly more pleasant after the curry. And then what we saw is some shifts in consistent shifts in microbiome from pre to post, but it's just, we need more data. We just need more data.
Honestly, being in microbiome research for as long as I have, we always need more data. we could do this forever. We're going to have to hold off for the vanilla discussion until maybe I'll have you back on the show again in season 3. But Jessica, thank you so much for joining us. I'm so thrilled that you agreed to do this, and keep doing it. Congratulations again, assistant professor. I'm just so happy for you. I can't wait to see what you're going to do in the future, because I want to find out what those women are doing with their microbiomes. But anyway.
I literally just submitted a grant for more funding yesterday, so we'll see.
R01, obviously.
I need more data first. I need more data before I can even do that. So yeah.
preliminary data. All right. Yeah. Thank you so much, everybody. Dr. Jessica Gaby.
Thank you, Jessica. That was great.
Oh my goodness. Well, as you can see, Ben, when I bring on people onto the show, they are absolutely amazing. They are just so fantastic. And again, you also see just how amazing their research happens to be. And remember, Jessica, Dr. Gaby is not an actual microbiologist. She studies from a psychological perspective, and yet it's just so amazing what she's actually able to do. Now, the other thing that I wanted to be sure of is that when we start talking about the relationships, one of the things that people have been discussing from day one is, what does make a normal microbiome? What does make a healthy microbiome? And the fact is yes, indeed, we need more research. We need more data. However, I don't know if you look at the Nature papers and stuff like that on a regular basis. I actually found some incredibly fascinating research about the persistence of microbes in the first year of life. Now, for anyone who's done microbiome research, you actually know that this is like one of the most controversial things. So if anybody actually comes up with some data that shows us what it's like in the first year of life, it's amazing, okay? Because honestly, when you think about it, we normally associate that to perhaps the mother, maybe to the family, possibly to a pet. But that's not really the most scientific type of analysis that we can have. And so when I saw this paper, I knew I had to bring her on as my next guest. And because she has been studying the infant microbiome, and her results are amazing. Now, she's going to be showing us some slides that are probably going to be able to give us a little bit more understanding. I've looked at them. They're incredibly awesome. But before we do, I want you to give a warm welcome to the University of California, Berkeley doctoral candidate, and I'm sure is going to be a PhD in no time. Claire Liu. Hi, Claire.
Hey. Thank you so much for having me. Hey.
How are you?
I'm good.
Thank you so much, first off, for agreeing to come onto the show. And I mean, when I saw the paper, it was immediate. I just needed to be having you on the show because I wanted to hear more about this. And I was just so fascinated that, not only was it years and years of work, but it actually answered questions. Seriously, if I had you around when the microbiome handbook was being written, it would have been so much better. So first off, thank you so much that you've taken this type of research. To start off our discussion, before we get into the slides, when I asked you sort of how to, what to call you and that type of thing, you actually said that you use computational biology to study the infant gut microbiome. And so before we get into that, how did you go from computational biology over here, which is really systems biology, as we used to call it when I was young and had hair, to infant microbiomes? It's absolutely fascinating. Can you sort of give us a little bit of that journey?
Yeah, totally. So I actually, this all happened when I was undergrad at Berkeley. So I stayed at my institute for a PhD. And then I have always been fascinated with healthy diet. And the one thing I kept asking myself was like, why do I have to eat fiber, even though like knowing the fact that, my body can't digest that. And then I also, and then I interned at a couple food companies, and I asked people around and no one really know. And then I also struggled with stomach problem for the longest time. And it's kind of like a very kind of funny story where like I went to Tibet with my parents, and then we went to this temple and I saw this monk, and then he gave my parents this kind of kefir grain. And that was the first time I learned about kefir or kefir. And my parents actually made our own kefir at home. And I would say after maybe about 5-6 months, my kind of struggle with stomach would just kind of disappeared. And later I learned it's because like, you know, kefir is like microbes and probiotic.
Yeah.
So that got me really excited about this whole microbiome field. And I read some books and watched this documentary Cooked by Michael Pollan based on his book. And then I decided that I want to study microbiome. So I switched my major to microbiology and then applied for a PhD to just study infant microbiome.
It's just unbelievable. Now, as I said, we never had that kind of research when I was editing the book. So you have to understand, I am incredibly excited to find out about your experimental and analytical approach. So if you don't mind, can you bring us in and let us know what you were doing?
Yes. So I think maybe easier if I just like pull out a slide here. Let me just get my setup going and then I'll share my screen.
Sure.
Okay. So I'm
Nice.
all right, can you see my screen?
Yeah.
Awesome. So I'm very curious about like, you know, what constitutes early life microbiome and where the microbes coming from, and if they come, what's their fate? And also, is there any differences between, you know, full-term babies and preterm babies? Because we know that a lot of preterm babies, when they were born, they received routine antibiotics, so killed the germs that were colonizing, like literally the day zero. So to answer that kind of questions, we actually enrolled about 180 babies. And then in the end, we were able to follow 42 babies from birth until age 1. And then we literally collected their poop every month for the first 4 months. And then after that, every 4 months, we collect poop from them. And then we do what is called genome resolved metagenomics. Basically, you sequence the poop and then you look at who's there and what kind of things they're doing. So I was able to track what's there for the 1 year and also compare that between full-term and preterm babies.
And when you were doing this, and I was looking through the paper, you were talking about this idea of de novo constructed microbial genomes. I find that absolutely fascinating. But what I'm very curious about is, you know, was the amount of microbial species so small that you could just simply do this, or was there an actual computational trick that you used to be able to do this? I'm really curious.
So I'm gonna show this diagram. It's a little bit complicated. I would just like briefly walk you through like the main storyline. So basically, like I said, we take the poop, and then the poop is like basically full of the microbes that live inside of our gut. And then we just use the sequencing technology. I feel really blessed to be able to do this kind of work at this era because the advancement of sequencing technology. And then we just basically sequence a bunch of DNA. And then our lab is very specialized in genome resolved metagenomics, is basically recover individual microbial organisms straight from the poop, straight from the DNA that we sequence.
Okay.
So that's basically like everything all the way here.
That's amazing. So whenever we've been talking about the idea of, you know, infant gut microbiome, the first species that always comes up is bifidobacteria. And I'm just kind of like wondering, is that really the case? Or, you know, is it just one of many species that are around within that first year? Like, what really were the most persistent strains that you found?
So for the persistent strain, bifido is definitely one of the group of bacteria that we saw persisting in babies. And then another group is actually Bacteroides, which is another common gut commensal. And I think it kind of makes sense to me in the sense where both of them break down polysaccharides, carbohydrates, and then fed that to downstream microbes. So they're really important. So being able to persist in the babies are also really important.
That's amazing. Now, I have to ask you a question. Have you ever actually watched the show CSI?
No.
No. It's one of these shows that goes and uses specialized laboratory technologies to try and come up with an answer and identify a killer. And one of the times that they did the show, they were actually looking at the idea of being able to identify twins because of their DNA. And I mean, that's pretty obvious how you do that. It's pretty simple. I mean, it's been around for decades and decades. But here's something that I didn't realize, and that is you might actually be able to predict twins based on their microbiome. And when I read your paper, I was like, oh, I got to ask you about that. So please tell us the story about infants 7 and 133.
Yes. So I will quickly bring this over. So this is about this figure in my paper, and I will just focus on this particular one. So I was doing strain results study, basically like looking for the number of shared strains between all the infants in my study. And, you know, from this diagram, so the x-axis are all pairs of infants, and I have removed all the siblings. And, you know, most of the babies here share like 1 to 2 bacterial strains. And then I saw this pair here, infant 7 and 133, shared 11 bacterial strains. And to me, I was like, that is just biologically unexpected to see that amount of like, you know, high strain sharing. And then, so we have collaborator in Pittsburgh University where they enrolled all the baby and collected the fecal sample. And they also collected all the metadata. And then we talked to them, and then they went back to like all the files they have. And in the end, they actually found that they were like, oh, this is actually a pair of sibling, and they were born 2 years apart. They just forgot to document that on the metadata. And when I found out about that, and I was like, this is just so cool, because like we also collected poops from the same mom, actually 2 years apart. So I was like, we can like actually see like adult microbiome persistent or like sibling transmission, all sorts of kind of questions, even though it's just one case.
But what you're telling me is that they're not even twins. They're siblings.
No, siblings, actually.
Wow. It really does make you think about it, that the microbial species that happen to be within an environment are really that predictable that you can pick people out who happen to be related to one another. I think that's incredible. But more importantly than that, I think it brings up this whole question of, you know, genetics versus environment. And it definitely seems to suggest that perhaps it is the environment, because they aren't twins, so they don't have the exact same genetic makeup. Like, does that make sense to you?
Yeah, definitely. I think environment definitely plays a very important role in terms of like the microbiome, who's going to colonize the gut, and also how long that kind of species are able to colonize the baby's gut. Like one thing I spend a lot of time on is maternal transmission, and how strains are vertically transmitted from the mother. How long can they stay in baby, and is this kind of transmission, you know, continuous throughout the first year of baby's life? And this is a lot of stuff going on, but just focus on like the kind of line. At this time point, it's like the first time point where we detect a particular strain. And, you know, if they are detected all the way until age 1, you're going to see them cross all the way down. And these are all the strains that's maternally transmitted. And one take-home message here is just like, you can see this kind of maternal strains appeared in baby throughout different time points. And some of them, you know, they can stay with the baby for the first year of their life. And the orange one are actually referred to the Bacteroidetes, the Bacteroides strain that we found to be one of the most common bacterial species to persist in baby.
Wow. That is just amazing. But it's also so small. It's funny, not small in terms of size, but small in terms of the number. We always talk about diversity, diversity, diversity. That's the one thing. But perhaps maybe it isn't necessarily as diverse as it needs to be, but you have to have the right diversity in the gut, as opposed to just
Exactly.
a massive amount of diversity. So how many species are we talking about? Like in total, did you find really were persisting?
Oh, in total, we're persisting. So in total, I found like about 11% of everything that I looked at ended up persisting in babies. Very small amount. Baby in
Wow.
general have a huge turnover in terms of who's coming in, who's leaving, just because baby's gut is so, immature, kind of chaotic, not like adults microbiome where it's
Oh my
relatively
goodness.
stable.
Wow. All right, turn off your slides. I need to see you for a
Okay.
second, because
Okay.
I need to be able to see you in order to tell you this. We've gone through a lot of information just in these few minutes, and even right now, my head is just like, oh my goodness. Most of the time when people do research, it's really about the paper. And they just do the science to get from concept to publication. They don't really sense what's going to happen afterwards. Having been seeing microbiome research for the last 10 years for me, what you're showing is going to change perspectives. It literally is going to change the way we do research into the future. And I'm wondering, for you, as a PhD candidate, who obviously is going to get her PhD after this paper, what does it mean for you to know that even at your young age, you're starting to make a difference that's really going to translate into, a different way of thinking down the road, especially for mother and infant health?
Mhm. I mean, this paper is like very, very special just because like I think it's my first author paper, and also the kind of, the kind of influence I hope to have, like to make people realize that, the early life colonization is important, because if you accidentally have the wrong kind of microbes, they might stay with you for a really long time and might have like impact on the host health. So I would say during the writing process, I definitely went through a lot of challenges. Like half of the data is contaminated. So I have to throw half of my data away.
Yes.
And like you said, we need more data. And I totally felt that, like,
Yeah.
losing half of the data really changed a lot statistically. And whenever I felt down, I think about the kind of things I want to tell people, also the passion about the microbiome. It just keeps me going. And now looking backwards, it's a really rewarding process, and being able to put out this work out as well.
Oh, all I have to say is that you are, much like the sun behind you, a shining light in research. Microbiome research really needs to have people who are going to take them to the next level. And I really do sense that you are going to be one of the people who's going to do that. Is there anything else that you want to just share based on your research to anybody who might be interested in learning more about this whole idea of infant microbiome?
I would say if you're a parent, like a mom, dad, definitely pay attention to your baby's microbiome. And since your microbiome will influence your baby's microbiome, like we talked about environmental strain sharing, definitely be careful with your own microbiome. And one of the best ways to practice to keep a good microbiome is to eat high fiber food, high fermented food, which is like, this recent Cell study showed.
Yeah.
And also from my own work, I really witnessed the importance of breastfeeding, breast milk being very important for your baby's microbiome development and also immune system development.
Well, I cannot wait to see what the future holds. I will be watching you, and I so wish you the absolute best as you continue on. Like I said, you are going to be a shining light for microbiome research, and I'm sure that the people who are watching the show are not going to forget you anytime soon. So once again, thank you very much, everybody. Soon to be Dr. Claire Liu Yu.
Thank you.
Thank you.
Thank you for inviting me.
Well, Ben, I got to tell you something. I have been so fascinated by how microbiome research has been progressing over the years. And in the last, I'd say 2 to 3 years, we've seen some incredible research that's come out, and how it relates to not just our own humanity in our relationship with microbes, but also how we can look to not necessarily the human, because I mean, clinical trials, especially when it comes to poop, it's not always the easiest, and you're not going to get funded all the time. But we do have cousins. And I think that maybe we should be looking at those cousins in order to be able to give us some ideas. Thankfully, well, actually, no, not thankfully, because I would love to go to Kenya, but we do know someone who actually has done this. And so when I first was looking at how we could wrap up this show, I really sort of came upon this particular paper and I thought, oh my goodness, it would be so awesome to have this researcher on, because she has found out that microbes, much like we saw with Claire's data from mother to child, may actually not only be inherited in that manner, but may actually be inherited based on the environment that is around you. But unlike what Claire has done, the person we're about to talk to has done it in baboons, which to me is unbelievable and is why I needed her on the show. So I want to welcome to the show the most awesome postdoctoral fellow you're ever going to meet out of the University of Minnesota, Dr. Laura Greenison. Hi.
Hi, Laura.
Hi, guys.
Hi,
It's great
Laura.
to meet you.
How are you?
I'm doing well.
Thank you once again for joining us on the show. And I'm serious, when I saw the article, I was just completely and utterly fascinated. And the thing that I found so amazing was that you were doing this type of work, which for all of us, we were thinking we need to do either in hospitals or going into different communities, remote and urban, and doing all of this. You went to Amboseli National Park in Kenya. It's like, that's amazing. So before we go into any of your work, can you sort of give us an idea of what that felt like, what that was like going into this type of a habitat to do this type of research?
Yeah, sure. So I did my PhD studying the wild baboons in Amboseli National Park in Kenya. And this is a really cool project because it's called the Amboseli Baboon Research Project. They've been studying this same baboon population for 50 years now. So Drs. Jean and Stuart Altman started studying these baboons in 1971 with this idea that if you set up a long-term field site, you can get an idea of how an animal's sort of ecology and behavior and social environment change over time. And you can look at different life histories. So you can look at sort of what predicts like how long baboons live or die. You can look at their social relationships and how these change over time. So you can collect this amazing data set. And so what that meant for me as a graduate student joining Dr. Beth Archie's lab at Notre Dame is that I got to fly out to Kenya and go to this national park to do research, which was extremely cool. And Amboseli National Park, it's right near the Kenya and Tanzania border. And so you can look up and you can actually see Mount Kilimanjaro. So to study the baboons, there's a full-time Kenyan field research team who lives there. That they do the majority of the animal behavior observations year-round. And so they're scientists from various universities of the US and also with the Kenya Wildlife Service and also at University of Nairobi, who are also doing work here. But this field team is sort of like the core, collection group. And so, we'd get up at before dawn, get into the Jeeps, drive out, find the baboons, because there's a few baboons that are radio collared in every social group. And so remember, the field team would be out with their sort of antennas standing on top of the car, looking to detect the signal. And then you could sort of watch the sun come up and you'd see like Mount Kilimanjaro and there's like elephants out and giraffes and you're drinking chai. And it was just this really amazing experience. And then the other thing that's very cool about sort of seeing animals out in the field is one, so these baboons, they're individually recognized by the field observers. So at any given time, the project is following about 5 baboon social groups, and social groups have 20 to 100 individuals or so. And the members of the field team, they individually recognize every single baboon in the group. So kind of like you can recognize like your coworkers or your friends, they can recognize these baboons sort of by the angles of their tail and the way that they walk, and like they'll have like distinctive fur patterns. I can recognize like maybe 5 of them. And so, but that's okay, because I was just out there to collect fecal samples. So, you know, I could, if I saw a baboon poop, then I would tell the observer, I'd say, who's that one there? I see.
So
how donated a fecal sample to the project.
Oh my goodness. I could talk with you forever about this, but we do need to focus on the microbiome. So please.
Right.
So you have baboon poop, which to me is still like the most fascinating thing. How do you get microbiome information from that? What's the next part of the protocol?
Yeah, so with the baboon poop, so if you collect fecal samples and you could get them into ethanol within about 15 minutes, that preserves most of the bacterial communities, the microbial community. And so we do that and then ship them back to the US and then do for doing DNA extractions. And something that's cool with this project is the way that you preserve fecal samples. If you want to look at like baboon genetics, so how related the baboon individuals are for each other, turns out it's the same way that you preserve fecal samples if you want to look at microbial genetics, so how similar their gut microbes are to each other. And so that means with this project, they have decades and decades of fecal samples, the freezer that were collected to use baboon genetics, 20,000 samples. And it just so happens that we could also use this for microbiome work. So when microbiome research started to become a thing, you know, a lot of the PIs on the project were like, oh, well, let's get together. If we get a whole bunch of grant funding, we can sequence the microbiomes of 20,000 baboon samples that are from these individually recognized baboons that we have all this life history information from them. We know who they're related to. We know, you know, sort of who they socially interact with, so who their friends are. We know what their diet is. We know what's happening in the environment at the time. And so there's just this incredible longitudinal data set that really doesn't exist in humans yet and doesn't exist in many other species, because microbiome research hasn't been around long enough that we have like 40 years of fecal samples, you know, from an individual person or something.
And that to me is just unbelievable, because that brings up 2 very, very important questions. One is that heritability, like I talked about off the top, but more importantly, that means that you can get a really good perspective as to the genetics and whether or not that plays an influence on how that heritability goes, or whether it really is environmental, like Claire, who we just talked to, actually showed us. I have to ask you, what is it?
Right. So like anything, whenever you're asking a nature versus nurture question, the answer is always, well, both. But the environment's playing a much stronger role, we found. And part of that is, so as Claire was discussing, the microbiome changes a lot over time. And so a lot of human and other studies that have looked at this idea of heritability are based on just a few time points. And because the microbiome is just fluctuating, you know, on the scale of hours and days and weeks, depending on what you're eating and where you are and what you're doing, and if you're stressed out or not, it can be hard to find sort of these overall genetic signatures. But if you have, you know, up to 100 samples from an individual over their lifetime, and you have this over several hundred individuals, you can sort of tease apart, like say, okay, well, controlling for these changes in diet and controlling for social relationships and controlling for who your mom is, because as Claire pointed out, right, we do see maternal transmission. Do we still see that individuals who are more closely related to each other have more similar microbiomes? And we found out that yes, that is the case for most of the microbial taxa that we tested. But this genetic variation is like very, very small compared to environmental variation. So on average, although we did find that almost all the microbes have this element of heritability, so they have this element of host genetic effects, host genetics is only explaining like 6.5% of the total variation of the abundance of each microbe. So this paper, right, in a way, it's very cool because it's consistent with what we know about the microbiome, is that there are these overwhelming environmental effects. But it's neat because we could find sort of this low persistent underlying genetic signature. And the reason that that's interesting is it suggests that when we have selection on sort of the baboon genome and potentially on the human genome, we also are seeing natural selection that could be exerting influence on our microbiomes as well. So, right, so you have the selection on the host, it's also sort of manifesting also as selection on your microbes. which is just a very neat concept, right?
That
That these are
is,
sort of part of you, but not part of you.
somehow I would just love to see you and Claire work together. Now, when I did some research into your background, I actually came across something that for me was very important. You know, I am of a, you know, of a minority in terms of who I am. I know that I have my own health concerns and that I actually have to worry about my microbiome. And that's one of the reasons why I went into microbiology in the first place. An adenovirus can kill me, like seriously, forget about COVID. And when I saw something that you're involved in, I had to bring it up because I want you to talk a little bit about it. It's called the Microbes and Social Equity Working Group, or MSE. And from what I understand, it's about bringing equality across the board of humanity. Now, you're already talking about this from the baboon perspective. We've done a lot of non-human primate translation into humans, but maybe we can do this in a social perspective as well. So I'm wondering if you can just give us a little bit of background on MSE and tell us, you know, how the audience can gain more information about this.
Sure. So MSC is the, as you said, it stands for the Microbes and Social Equity Working Group. And this was founded by Dr. Sue Ishak at the University of Maine in, I think, 2018, but it may have been 2019. And the idea is that, right, so there's a lot of us who are studying the microbiome out there, and we would like our research, you know, to be used and sort of applied to looking at sort of human health concerns and human social equity concerns. But that's not really like our field. Like I'm coming into this from like ecology. I don't really know how what I do could necessarily apply to social policy. Sort of make these meaningful social changes.
Let me just cut you off right there. What you are doing will inevitably have perspective on policy development. I've been involved in policy development. It is amazing where we pull stuff from. But yes, sorry, continue.
Right. Okay. Well, I think the idea, right. So I guess it could have, you know, policy implications at some point, but the idea of this group is to say, okay, well, how can we sort of link policymakers with microbiome scientists, like in a more immediate sense? And so one way, if you want to learn more about this group, is that we do have, there's a mailing list and there's a website, which I don't know if I can send you the link and you could post that with the show or something. And there's a group newsletter. And we also have a journal collection coming out. So a whole issue devoted to this idea of looking at microbes and social equity, and talking about sort of 20 questions in the field. And so one of the things, the major questions in the field is something that, you know, almost Dr. Liu was talking about, is that do we see, how do these early life influences on the microbiome, and especially sort of these early life social influences, manifest over time? And specifically, for example, if you have sort of early life adversity, do we see a signature of that in the microbiome? And so one example of this, that this is actually the reason I got into the MSc group and interested in the MSc group in the first place. So I saw a talk a couple of years ago by Dr. Rashonda Jones out of California, that she was looking at, right, okay, so she was looking at how you're feeding infants. So if you're using formula versus breastfeeding, and she found that where she was, that the formula that was covered by WIC was this formula that had higher amounts of corn syrup solids and higher amounts of sugars in it. And so that meant that if you were getting, right, if you were feeding your kids on formula and you had to use WIC, that you're giving them formula that it could actually be setting them up to have more trouble with diabetes or obesity later in life, because it has this higher sugar content. And so her explaining that to me, I was like, oh, wow, this is like a very concrete example of how, you know, microbiome research and policy research should be combined. And sort of the goal of this group is to find, you know, both other scientists who are doing that work and other policymakers who are interested in that, and sort of, you know, trying to move forward.
I'll tell you one thing. We don't have much time here, but I'm just going to share this and then ask you a question. I remember being at a conference after giving a keynote and having a company, a multinational company, coming up to me going, "You're going to hurt our sales because you told them not to have blank." And I'm like, sorry? Not sorry. We do need to have that equity. Absolutely. Now, you have a perspective unlike any other researcher I've ever met. And so what I want to ask you is, let's go back to what my first talk was about, which was our relationship with microbes, and that can have a relationship amongst ourselves. How do you see your work with baboons with 50 years worth as being influential in how we can identify with our relationship with microbes and, of course, with each other.
Right. So I sort of see it as fitting in a way into the overall sort of One Health concept. So sort of the health of humans, the health of the environment, the health of the other non-human animals and plants within the environment are all sort of the same thing. And by looking at the microbiome, we can sort of extend this to even the microorganisms within the environment. So if we understand what's happening in wild animals, so if we understand what's shaping their microbiomes, and especially how these microbiomes are changing with changing environments. So this could be both like seasonal changes or diet changes or sort of bigger impacts. So we're seeing the climate changes causing these more severe environmental shifts. And so if we can see how that's affecting microbes and by extension, the health of individual organisms, that'll help give us sort of insight into the future to how that could affect human health, other aspects of environmental health, because, I study animal-associated microbiome communities, but there's a lot of microbes that are associated with plants and the environment and crops and other aspects that as we see climate change happening and more environmental change happening, we're probably going to see some shifts in those microbes too. And that might not be ideal.
Wow. Well, I wish you the best of luck. And if you ever need someone to just come out and help you scoop some poop out in Ambaselli, I'm here.
I feel like I haven't actually been out to the field now since 2014, because once a lot of the samples were collected, my whole postdoc, I've just been in the lab, and I'm like, one day I'll get to, go outside and do outside science again.
We'll go have some kafta in Sultanahmet. How's that sound?
That sounds great.
Anyways, ladies and gentlemen, Dr. Laurel Greeneisen, thank you so much.
Thanks.
Thank you. Thank you very much indeed, Laura. And thank you to Jason. It's been an absolutely fantastic session. Thank you for hosting it.
Absolutely. And I want to give you a special shout out both to you and also to Joseph Shuttleworth for making this day possible. Microbes are everywhere, but you two simply have covered it all. And anyone who's ever listened to my show knows that I want everybody to just enjoy the rest of the presentations, enjoy microbial diversity. And as always, well, make sure to show them some sass. Good night, everybody.
Thanks, Jason. See you next time.
Bye.
All right. So next on the agenda is traveling to Australia, where we're going to be having a tour of the Yangara vineyard. Is that where we are now, Susie?
It is indeed.
Wow. Fantastic. Hi there, Pete. How are you?
Hi, how are you, Ben?
Good to see you.
Welcome to the Yangara vineyard.
It looks great. I'm going to hand straight over to you. I mean, the weather looks great. The vineyard looks fantastic. Yeah, take it away.
Yeah. It's a bit windy, so I hope the noise isn't too bad. It's like a beautiful kind of 23 degrees, and we're in kind of the beginning of spring. The vines are starting to bud burst. This is kind of probably the most amazing block on the estate. Roughly about 95 hectares, and behind us is about a 2 hectare, 1.7 hectare little block, planted in 1946. This is our High Sands Grenache vineyard. So we're looking at vines that are nearly 80 years old.
Wow.
I suppose we're here to talk about not so much the vineyard, but the microorganisms that kind of play a really big role in actually growing grapes. And also later on, we're going to go down to the winery and talk a bit about microorganisms that are, integral part, good and bad, of making wine. So in about 2007, we started talking about organic biodynamic farming. We were all very, I suppose, conservative, soft, gentle kind of in our farming approaches. In 2008, we talked about that we wanted to basically remove all synthetic chemicals, and primarily they are in the form of herbicide. So glyphosate is being the number one kind of enemy of plant life and also those microorganisms in the soil, and then also your heavy-handed kind of diammonium phosphates, the high nitrate synthetic fertilizers. And so this vineyard has now been farmed since 2008 without any synthetic fertilizers, chemicals, and it's been certified since 2011, organic and biodynamic. Our organic biodynamic principles is based around a lot of different types of farming, very practical approach, both in permaculture, regenerative farming, all those types of things that are. We've got all sorts of practical ways that we like to do it, but it all forms around, I suppose, this really alive soil, where we're really promoting the microbes, the microorganisms in the soil to be doing a lot of the work for us. And within that, we're in beneath the soil here, which is actually sand, is 70 million year old geology. So amazing old rocks, and rocks all have their own personality with their own minerals. And what we want to do is the vines having access to those minerals that are native to that parent rock or that geology. And microorganisms are the kind of pillar for enabling that, which effectively the French use a word called terroir, and we want to kind of enable it the best way we can. So that's kind of the guts of why and what we do. And I'd like to hand over to our vineyard manager, Michael Lane. Come and join me, Michael. Michael's been managing this vineyard for 20 years. His off-sider, Sam Rogers, who's assistant vineyard manager and viticulturalist. So they're going to kind of talk about some of the microorganisms and things that are good and bad in the vineyard. So I'm going to hand over to them.
Hi, I guess with microorganisms, we're trying to enhance and use the beneficial microorganisms and minimize our disruptive microorganisms. Some of our beneficial microorganisms, mycorrhiza, soil mycorrhiza, trichodermas, etc., for nutrient uptake. But then I guess some of our biggest challenges are negative microorganisms. So a really common one is Eutypa lata, which is a dieback, which spreads about 2 centimetres per annum. And this is basically a dead piece of wood. We do things, cultural practices to mitigate it through pruning, late pruning, let sap flow, etc. And I guess we're trying to encourage our beneficial microorganisms and through cultural practices, composting, using a lot of molasses, humic acids, fulvic acid, fish, kelps in our applications.
You've got a great example of some compost here, Mick?
A lot of composting is applied. So we're applying around 1,000 cubic metres of an organic compost per annum to our vineyard, depending on requirements. And also our biodynamics, which I'll let Sam have a chat about.
Yeah, and that's the compost. Are you using wort as well as?
So a combination of both our winery waste through vintage. We'll compost that down and bring in, we always carry over an inoculum each year and incorporate that with our winery waste and keep building on that each year.
Yeah. So this is our biodynamic preparation called 500, and it's a composted cow manure, and we use it as an inoculum in the vineyard to help promote diversity and beneficial bacteria and fungi around. So we also add to our compost, on-site composting, minor preparations, and they're essentially used for a similar purpose to build up the diversity and in our soils. So you can see, so that they're additions to the soil. So what we do culturally as well. So we're always sort of slashing into compost our soils.
Do you use foliar sprays as well? What foliar sprays do you use?
Yeah, so we do a 501, which is a ground quartz silica. So that's more of an atmospheric spray.
So not
sunlight,
not so much more microbiological, but do you use like things like fish and kelp?
Yep. So with our foliar sprays, we'll add a fish humic mix.
Fulvic molasses.
Molasses, yeah.
So what does the molasses do? Does that give basically a food source?
A food source for bacteria. So yeah, it's that whole holistic approach and feeding our beneficial and feeding our disruptive microorganisms as well, but getting a balance.
Yeah. That sounds great. And I suppose that's all the good stuff. How do you the bad guys in the vineyard, the bad microorganisms, like powdery, downy, botrytis?
Yep. So botrytis is another big one, a bunch rot, our mildews, powdery mildew, downy mildew. So we're using elemental sources of sulfur and copper, minimizing our heavy metals and copper. But a lot of cultural practices, opening up the canopy, leaf plucking, shoot thinning, getting good bunch exposure and using Mother Nature in our environment to help control some of these diseases or microorganisms.
Yeah. And so it's interesting in terms of, how does that differ in an organic system like this vineyard versus a conventional
I guess
vineyard?
there's a huge amount of focus on being preventative rather than being reactive. So we don't have, in a conventional scenario, tools to go and eradicate. We're using nature and balance in the vineyard to help us control a lot of these microorganisms.
Yeah, a lot of decisions made for powdery mildew and downy mildew relate to weather events, so with humidity and temperature. So if we hit a certain series of events, we know to then keep a closer eye on it, and we can potentially get spores in the vineyard. And because they're everywhere, we're basically suppressing it and controlling it how we can.
Ben, have you got any questions of these guys?
Oh, you're on mute. Can't hear anything.
Could
hear
right,
me before.
Oh, there we go.
Yeah, I'm back. Sorry about that. I was wondering about the wort from the vineyard, and does that have any effect, or is that just you're using that to kind of find a good way of kind of using it up and using it in the growing of the new crop? But does it actually contribute anything like in terms of the microbes or the nutrients specifically to the crop?
Did you say watering?
So the leftover from the vineyard. So when you've kind of made the wine, you said you took the waste and you composted it, and you also used.
So yeah,
does that
more
contribute?
the bunch stems, the skins, the seed, the rachis, rather than liquid waste. So it's solid waste that we're composting back down.
Yeah, and what's interesting is you can see behind you this is a dry growing, a non-irrigated vineyard. So this is relying completely on the rainfall. Predominantly, we don't get a lot of summer rainfall. So these vines have roots down at least about 1 metre. And so they're sourcing quite deep from within this sandy soil. And one of the things that we didn't mention is that we are very aware of distributing our compost across the vineyard. Some vineyards, they ban compost directly under the vines, mainly because it probably suppresses weeds a bit. But we feel like if we spread it across the whole soil, that that compost is kind of in some ways inoculating the soil and promoting more microbiological activity in the root masses in the right across the whole vineyard, not just where the trunk of the vine starts, because as we all know that the roots of the vine are three-dimensional, and it's a bit of a human kind of nature thing is to think a bit two-dimensionally, and we tend to want to kind of put them in a single row versus kind of spreading it.
Treating the whole site as one rather than
Yep.
Yeah.
We've got a couple of other things that I'll probably jump back into screen. I wanted to talk about the microorganisms that come in to making the wine that come from the vineyard. So on the outside of a bunch of grapes, those who remember kind of probably don't get to see wine grapes very often, but those of you who buy table grapes will see there's a white bloom, especially on the dark. It's very obvious on the dark coloured grapes. And that bloom is the yeast and predominantly, and it's those yeast when the grapes are split or broken are basically fermenting the wine for us. And at probably in quite commercial wine, New World wine, the practice is to add packet yeast, but a lot of old world wineries in Europe and now around the world are wanting to use the native yeasts in the that come from the vineyard to actually make the wine. So there's this kind of makeup of the flavors from the organisms in the vineyard contributing to the flavors of the wine. And there's things that are very important to manage what microorganisms survive and thrive. So if the alcohol is too high, i.e. the sugars are too high, some of the good yeasts actually will have troubles converting all the sugar to alcohol, and you'll end up with residual sugar, which can end up providing a home to all sorts of spoilage organisms like Brettanomyces and other spoilage yeasts and bacteria. And so the other part that is also one of the very critical things that we do to manage these microorganisms in a natural way is picking the grapes at the right time when the natural acidity is there, which also then manages the types of yeast that are activated. Now we're going to head down to the winery, and what we might do is take some questions while we're driving down to the winery, and just a bit of change of scenery, and we can answer some questions. So give us 20 seconds. Great. We'll pile up all in the car, and cool. You can ask us questions.
Could you just spin us around before you go and just show us a bit of a panoramic of the vineyard there so we can see it? It looks spectacular. Wow. Amazing. Amazing weather as well. Is it always like this?
We wish.
So you had some summer rain as well. But
right.
Great.
You can go out the back window. So any questions from that so far, Ben?
Yeah. So you said you've got to pick the grapes at a very specific time, when the acidity is right. So how do you do that? Presumably it's a lot of testing. And what acidity do you need, and is it about reducing the sugar content?
So as a grape ripens, it accumulates more and more sugar. That's measured in a few different ways in various different countries. We use a measurement called Baumé. In a simple term, most wine is somewhere between about 12 and 15%. Approximately 12 Baumé would equal 12% alcohol. This is a very simplistic explanation. 15 Baumé would equal 15% alcohol. 15% is generally the upper limits of yeast tolerance. So you really don't want to be picking, and I actually prefer, and native yeasts that are coming in off the grapes generally don't like much more than 13.5 Baumé or 13.5% alcohol. And then while that sugar's accumulating, the berry metabolizes it's an acid called naturally forming acid called tartaric acid. And as that's being metabolized, it's also exchanging hydrogen ions with potassium ions. And the pH will start at around 3, and it will head north and head probably towards 4 Baumé, a pH of 4. And basically at less than 3.4, 3.5, and above 3 is a very favourable place for a yeast strain called Saccharomyces cerevisiae, and there's a few others, Baianus strains, that actually are very naturally occurring on the grapes, and those yeasts thrive in that acidity, and they are able to convert the sugar to alcohol without producing, I suppose, spoilage flavours like, or things like volatile acidity, which is, basically nail polish remover in high levels, acetic acid, which is vinegar, and other flavours, that are more complex and more detailed. But if you set the conditions right, you will end up with a lovely clean ferment with lovely flavours, and also they also produce some higher alcohols that have lovely mouthfeel and textural characters within the wine. So that's all the things that if you set it up right, you can have Mother Nature do her thing.
Great. And by doing that, does that give you consistency? Because you would automatically think, I guess, wild yeast would kind of produce a lot of variability. How do you manage that, or is that not the case? It's normally quite consistent.
I think, like I said, if you set up the parameters right, those native yeasts are fairly predictable. What you'll find is in kind of large commercial cheaper wine, they want a complete reproducibility and reliability. So you'll actually obviously then buy freeze-dried packet yeast of very particular strains, and actually make sure that it's all kind of reproducible and nothing can kind of go wrong. So so that's the general on the kind of using the microorganisms from the vineyard through to the winery. So we've just arrived at the winery. So Susie will pan around the winery and we can get into some more there.
Wow. I'm guessing these are filled with wine. Wow.
So this is kind of roughly where the fermentation happens. These are open top fermenters. We basically, where the yeast are converting sugar to alcohol, and at that same time extracting flavour out of the skins, especially for red wine, primarily red wine making. And we're about to go into our barrel store, which has got all sorts of barrels. We also use amphoras and ceramic eggs, which are very interesting in themselves.
Right. What are the differences between those? Why would you choose a barrel or an amphora or an egg?
It's interesting. They all provide different flavours. When a wine's maturing, it's basically the enemy of wine is oxygen. So it's important that the wine is contained in an oxygen-free environment. But when I say free, it actually also needs to have a little bit of oxygen. So at a micro level, so a barrel is great because air travels across the oak medium, and the same with these amphoras. There's some oxygen transgress that enables that to the wine to kind of continue to be alive. Basically, wine goes through a primary fermentation, which is driven by yeast, and there's been studies done. The Australian Wine Research Institute has done
a lot of testing. Our vineyard was used where they actually mapped all the different strains of yeast that were actually involved in fermenting our wine. And they also were able to showcase different flavours, different higher alcohols that have been produced by this different map of different organisms, microorganisms that have been involved in each of those fermentations. I can't remember exactly how many different vineyards that they analysed, but they were also analysing the grapes and also the wines. And they were also, there's also a lot of myths to say that a lot of the native yeasts actually were primarily contributed from the native yeast that lived in the winery. But they actually were showing that they were actually very reflective of what was native in the grapes. Wow. Which is really fascinating, and fascinating to see the diversity and complexity created in wine by actually using the native yeast versus the say a packet yeast, which is possibly more reliable, but might not give you the complexity and the interest that you got from the wild side. Then the next stage in wine is the bacterial fermentation, where malic acid is converted from malic acid to lactic acid, and that again stabilizes the wine. There was probably something only discovered in the early 1900s. I might be not quoted on that exact, but my vague memory is that it was a relatively new thing discovered 70 years ago, let's say. And that basically stops the wine, having another fermentation in bottle, which could actually spoil the wine if it happened when it was in the bottle. That also softens the wine, but is also responsible so that malolactic fermentation from those lactic acid bacteria, they, in a Chardonnay, for example, produce a buttery, waxy kind of smell and flavours. And also very useful in cool climate grapes where the acidity might be very high, and it helps to soften the flavours of the wine. In red wine, it's also very important because it literally stabilises the wine and sets it up for a long life. So fascinating how many all those things. I have one other thing, and then we could probably, because we're starting to run out of time, is some of the spoilage, guys. That Brettanomyces is a scourge on wine. And it's actually, you see it, I'm quite sensitive to it because we're always looking out for it. And it thrives in higher pH wine. It thrives in higher temperature, and it thrives in dirty barrels. So if you don't clean your barrels, wine throws something called tartrates, which is like a crystalline formation. And Brettanomyces loves living in tartrate in old barrels. And like I said, warm cellars and also stuck fermentations. So if you don't get everything lined up in the early part of your winemaking, Brettanomyces will come and hunt you down and make you look bad, because your wine will stink like horses and Band-Aids. So those of you who come across that smell, it's all about the Brettanomyces. But for the most part, there's a few other complex ones that mainly
oh, did we lose you there? The screen is just frozen. If you can hear me. Hello? Hello, hello, hello. Are you there? I hope we get them back. I was looking forward to them opening one of the bottles of wine there on the barrel. They've just gone, so maybe they come back just to kind of sign off, because we're going to be handing over very quickly to the next session. Oh, hello, I think we have them back. Hi there, Pete. I think you're on mute.
We
Hi.
back?
Yeah.
Sorry.
No
we just
worries.
had a
No worries.
freezing. Lucky it was at the end. But
Yeah.
lucky we can at least get to say goodbye. I think we've probably run out of time now.
Yeah, we're just right on the edge. So yeah, but it was great. Really kind of thorough kind of whistle stop tour of everything. I mean, the vineyard and the winery, and kind of just seeing how big it was and expansive. It was really, really great. And like we discussed when we talked earlier in the week, I'm definitely going to be looking out for some bottles wherever we can find them in Europe. So thank you very much. Thank you to the entire team. And we definitely have to do this again. I think spend some more time around the winery and the vineyard. But yeah, a huge thank you. It was very, very interesting.
Absolute pleasure. So enjoy the rest of Microorganism Day, which is also International Grenache Day. So that's why we set up.
Oh, well, that's a happy coincidence. So we know how to celebrate.
We get a
them.
double celebration. So.
Yeah. Fantastic. Well, thank you, and speak to you again soon.
Well, thank you. Thank you for having us.
No problem. Bye now. So the next session that we're going to go on to is the breakfast show. So we're going to be handing over to Joe, who is in the studio there.
Good morning. You can hear me all right then?
Yep.
Yeah.
Here.
Happy International Microorganism Day, Ben. It's now.
Happy International Microorganism Day. I was checking the time zones all night. I think most of us are there now. I just have another quick look.
So we're waiting. No, the East Coast of America is now in International Microorganism Day. So it's just the West Coast that needs to catch up.
Come on, West Coast.
Wonderful. We're here. The day has arrived.
Yeah.
It is International Microorganism Day. I have my coffee. We have my co-host, Michael Sawyer.
Okay. Well, then I'll bid you a happy Microorganism Day, International Microorganism Day. And I'll bid you farewell, and I'll watch in the shadows the rest of the...
Thanks a lot. I enjoyed the wine tour. I caught bits and pieces of it, but it was fun to see it.
Yeah, it was indeed. Well, thank you both, and enjoy the breakfast show.
Thanks very much. Right, Michael. I think we need to unmute you. We have you. How are you? Good morning.
How are you?
Yeah, not bad. I've had about 5.5 hours of sleep, which is better than none. But I wake up on International Microorganism Day of all days. So that is a fantastic place to start, isn't it? How are you?
I'm good. I just woke up. I had my coffee, and now I'm really excited about fungi.
Yeah, great. Because this year, you're my co-host for the breakfast show. Because last year, you were a guest of mine, and we talked about beer and yeast and baking and sourdough. And you had like a live sourdough culture on the table for us that we looked at. Now, this year, we're going to be looking through, lots of different things. And I'll just talk a little bit about how our breakfast show is going to commence, and then we can get on with it. But basically, we're going to be looking at our top 10 fungi. I mean, we might even have more than 10, but we're going to be working through a list of our favorite fungi for the first part. Then we have a couple of guests coming up. Our first guest will be Maria Martinova, who joins us from New York. So she's actually going to be joining us at nearly 2 a.m. there. But she's going to come talk to us about probiotics in yogurts. And also the Microbites platform, which is like an online kind of website, blog space platform of content for microbe lovers. Then we're just going to have a little recap of our favorite parts of the live stream so far. Celebrate the winners of the Microbe Art Contest. And then I think we've got a guest that you've lined up, Michael. Is it Paola Branduradi?
Exactly.
Yeah. And do you want to tell us a little bit about her background and what she's going to talk to us about?
Yes, she is from University of Milano Bicocca in northern Italy, but she actually is in Trieste now, traveling for a conference, so she will join from Trieste. She's a yeast specialist working on chemical production with yeast, and she knows a lot about really weird yeast. So very fun talking to her.
Well, that's going to be fun. I mean, yeast and fungi seem to be some of the weirdest microorganisms, at least in my experience so far. There seem to be lots of strange ones that do interesting and bizarre things. And yeah, I can't wait to discover more of them across the course of this show. But yeah, you joining us from Vienna, Michael?
Yes, right now, actually, I'm joining you from the western parts of Austria, but I am working in Vienna. So usually I'm in Vienna, working also at university there. But now I'm home at my parents' place. So when I watch out of my window, I see Switzerland, actually.
Oh, wow. So what town are you in now?
Oh, it's a tiny place called Mäder.
Okay. Is it anywhere near Salzburg?
No, it's much farther west.
Further
it's
west.
Essentially the westernmost point of Austria.
Yeah, okay. And yeah, how has Austria been treating you this past year and a half? I mean, it's been a very difficult year and a half, but have you been basically based only in Austria, unable to leave for the past few months, or did you manage to get some travel in over the summer?
Well, over the summer, I did some traveling. Yes.
Yeah.
I went to the mountains. I went a little bit to the sea, but otherwise, that essentially was the first travel I did the last 1.5 years. Yeah.
Yeah. Okay. Wow. Yeah. Just about.
Your home.
Yeah. Well, you know, Austria is not a bad country to be. One of the most beautiful places I've been. But yeah, I'm just going to recap to anyone who's joined us. What is International Microorganism Day? Well, it's a celebration of microbes for those who love microbes, whether you're a microbiologist, a student, or just someone who thinks they're cool. And the day was introduced by the Portuguese Society for Microbiology, and then the organization I work for and that Michael is involved in, FEMS, the Federation of European Microbiological Societies, have joined forces with the Portuguese Society to help try and internationalize it as much as possible. The date, 17th of September, which most people are now in, was chosen because it was the date that the first letter from Anthony van Leeuwenhoek reached the Royal Society, describing for the first time microorganisms that he'd observed with his homemade microscopes. So it's kind of, you know, the human discovery of microorganisms, I suppose, that we celebrate today. Although they have been used by humans, right, throughout all of human history, pretty much going back to the point of agriculture, I think. People have found, you know, beer brewing from thousands of years ago, fermented foods from thousands of years ago. They've obviously lived inside us and on us and, you know, amongst our livestock and plants for the whole of human history. So they're super important. We're here to celebrate them. If you're waking up somewhere in Europe or across the world today, drinking your tea or coffee or even having your dinner, thank you for joining us on The Breakfast Show. I guess we shall just move into our first section because we've got just over 30 minutes before our first guest comes, which doesn't give us long per fungi, but we have a list of our top 10 fungi, and we're going to work through it today and discuss some of the finer aspects of these organisms. So, are you ready, Michael? Are you ready for our top 10 fungi? Okay, so we put this list together, and I don't think we fully decided which were our top 10. So we might have a bit of argumentation or debate across the course of the next half hour about which should make it into the list. But for me, the first one on the list that I think is worth a mention is the cordyceps fungus. Now, are you familiar with this fungus?
Yes, a little bit.
A little bit.
It's so weird that I also looked into that one.
Yeah, because it is one of the weirdest fungi that I've ever read about. So if you haven't heard about cordyceps, let me just explain a tiny little bit about it. So this is a species of fungi that basically infects insect species, and it starts to grow inside the insect and replace the insect tissue with fungus tissue and hyphae and so on. But it doesn't just sort of rot the microorganism, it actually takes control of its brain and nervous system. And in a little minute, I'll have a couple of images for you, but what it essentially does is control the insect so that it starts to do strange things like walk up a tree or a plant and bite down into the top of the plant. And then once the insect's dead, the cordyceps fungus fully replaces the internal size of the flesh and then bursts out through the insect body with its fruiting bodies. So it's kind of a zombification fungus that also manages to control and then digest the organism. Do you have any fun facts about cordyceps, Michael, before I show us a small video?
Well, a fun fact that I recently came across is how wonderful evolution works, because exactly as you said, so it takes control, it makes the ant go up to the tree, what the ant naturally would never ever do, and then it bites there. And why does it do so? Of course, the fungus wants, let's say, to spread the spores as high as possible to get, a large area. And usually they are from tropical areas. So that's where they evolved, and there the ants bite into leaves. But they spread actually in areas where more colder weather is. And there it's not good to be at the leaf, because the leaf might fall down at the wrong time. And so actually it would be better to bite in a little, woody part of the tree. And actually it evolved that way. So there are cordyceps which make the ant go up the tree, but bite into woody parts and not into the leaf. And that's completely weird, because that means that this fungus can really control not only how the ants behaves, but also where the ants is, so depending on the climate.
And they are found everywhere, or in lots of different environments, right?
Nowadays in lots of different environments. I think in Europe they are not really present.
So here we have a picture of a cordyceps fungi bursting out of a dead wasp. So you can see these fruiting bodies. I mean, they're like little mushrooms really for the cordyceps coming out of the body of the wasp. But again, this wasp, as you say, has bitten down into a woody stem. And I guess this is to place it somewhere, in a high exposed position, so its spores can then float out over the jungle or the forest and infect other insects. But there's a few other weird facts about this fungus. And do you know about the role it plays in Chinese medicine?
Yes.
Yeah.
Well, the point is, it's such a weird thing, and that it's allegedly it has a lot of different roles, medical applications, and so it's extremely expensive, to be honest. So if you want to buy certain kinds of the fungus dried and milled, it's quite expensive.
Yeah, and in fact, in some places in Bhutan, villages in the mountains, one of their main sources of income is collecting these cordyceps bodies, which look a little bit like this.
Yeah.
And then selling them on to traders, and it's their biggest source of revenue in some parts of Bhutan. This is a paper, actually, from our journal FEMS Microbiology Letters that looks into the use of cordyceps fungus in Chinese medicine. But one of the things they highlight is that, like you say, they're so expensive and worth money that people have actually begun faking the cordyceps fruiting bodies. So of these 4 here, example C is actually a fake one. So these are real caterpillars with a fruiting body bursting out due to the cordyceps fungus. But this version here is simply a caterpillar and a grass stem that has been glued together to fake a cordyceps fruiting body to make some money. But you can't really tell, can you? They're very similar. But this paper I dug out from the archive from 2013 basically looks into, they're barcoding the DNA, the little caterpillar funguses here, and they found out that some of these are fake. So not all the fungus that you will buy in the Chinese medicine market will be the true cordyceps. So I think that's a great one to begin with. I think the second one on our list is probably the favorite, maybe of you, Michael, but at least of many microbiologists. So that one is the famous Saccharomyces cerevisiae.
Yes, exactly. So that's the brewer's yeast or baker's yeast. And the point is, first of all, I think it's good to talk about it after the creepy one, because that's the one that gives us lots of good things. So it gives us beer, it gives us wine, it gives us bread, and it's very useful. And actually, that's what microbiologists tend to call the conventional yeast, because when they talk about other yeasts than baker's yeast, they talk about unconventional yeast. I think it's kind of funny that only one is conventional, but that's why we know a lot about it.
So we've got one conventional yeast, and then everything else is unconventional.
Is considered unconventional, exactly.
Fair enough. And we have a lot to thank, right? I mean, so we have beer, wine, which just had a vineyard tour. So there would be no wine without Saccharomyces cerevisiae. Bread, it's involved in some way, yeah. I mean, I'm British, so...
Pizza.
Sorry, say that again?
Lots of cakes and also pizza.
Pizza, cake, pizza, yeah, okay. All of the delicious things, it seems, are thanked. We can thank this yeast for. And I was going to say, I'm British, so I love Marmite on my toast in the morning, which is made from the boiled up yeast bodies of the brewing process. So I think they boiled up all of the yeast at the end of the brewing process. They dehydrate it into a delicious, sticky black mess, full of salt and vitamin B12 and tons of other good nutrients. Oops. So here we have a little video again of Saccharomyces cerevisiae. So you can see, you know, it's fairly simple looking, isn't it, under a microscope? It's just a little budded yeast cell, which forms these little sort of colonies almost of grouped cells. But here we can see the budding process, right? So it doesn't reproduce like our cells or bacteria. It does a slightly different process. So I wondered if you could tell me a little bit about the budding reproductive cycle.
Yeah, that is extremely interesting. The point is the budding is an asymmetrical process. So it's not like bacteria usually divide in the middle and then you have two equal parts. And this is also quite philosophic because bacteria in this sense live eternally because, you know, every part can divide again. But in yeast, this thing already became asymmetrical. So what you can see here is that a little part is budding from, let's say, the bigger part. So the little part is the young part, we call it the daughter cell, while the bigger part is the mother cell. And so it's really not only asymmetrical in size, but also like the molecules are divided. Let's say all the new ones are going to the daughter and the older ones are sticking to the mother. And that means already that here the eternal life ends because the mother can only divide a certain number of times and then it dies. So yeasts do not live eternally. They have daughters and they go on, of course, but the mother is doomed at one point because it accumulates, let's say, the old stuff.
Okay, so like a well-meaning and I guess motherly parent, it gives the best of what it can to the new cell.
That's exactly what it does. That's exactly what it does.
So, I mean, we had a brewery tour last night, and they were saying that, yeah, they probably got out about 20 generations of yeast reproduction from their yeast before it started to perhaps decay a little bit and not be quite as snappy as it was in the past. So, I mean, that doesn't make too much sense to me at the moment, because doesn't this mean that, like, the yeast will reproduce for, 20 generations, but then suddenly it can't reproduce anymore?
Well, the point is you always have the daughter, right?
Okay, so it's just the original cell.
The mother, exactly. The original cell at one point eventually dies, but the daughter then becomes a mother herself and will start to bud again herself. And so you still reproduce, you still get more and more and more. So one can do it like 30 times. So you get, unlimited amounts of yeast cells, so to say.
Yeah.
But if you look at the individual, then there's a difference between bacteria and these yeast cells. So, and it's interesting because it's like growing from the really simple ones to the really complicated ones. Humans, at one point, age becomes a thing. And these cells age, and you can see aging, and you can study aging with yeast cells.
Because actually, I think you're an editor for one of our journals in Microbiology Letters, but we have a corresponding journal called Fem's Yeast Research, right, which focuses just on yeast and other similar fungi. But they had a thematic issue on yeast death and cell aging, and I guess we can use it as a model organism for studying human aging in some ways, isn't that right?
Exactly. Yeah. Because some of the mechanisms that happen for yeast cells are very similar to the ones that happen to us.
So hopefully, not only can yeast provide us with all the wonderful foods and drinks that we love, but maybe it will unlock the key to immortality, and it will make us live forever.
Exactly.
I think number 3 on our list is another yeast, which I didn't know about. So you've added this one. So that's Saccharomyces pasteuranius.
Yeah, the point is when we look at lager beers, they are actually not produced by Cerevisia, but they are produced by another yeast. And the interesting story behind that is that this yeast is kind of cold tolerant. So lager beer is brewed as a lower temperature than ales, for example. But the yeast has to be efficient. So selection actually led to a hybrid cell. So that's also kind of a zombie existence. So what happened is that two yeast cells fused and, let's say, built a new entity in the end. And what was known very early when we studied these lager yeast cells is that one part was cerevisia. So that's the part that brings in the good fermentation and the aromas and all this stuff. But cerevisia is not cold tolerant. And so genetically, what we could figure out is that there must be another part, but we didn't find it. And many people were looking long time until it was found actually the first time in Patagonia.
Okay, and that's the first time it was found, but it had been used for.
The first time it found that it was a big thing. So breweries were using that yeast and so on. So it's absolutely fantastic. And so there was a lot of debate because, of course, the yeast itself was selected in Europe. And so the question was, how would the yeast from Patagonia come to Europe? So there were a lot of weird stories about this. The point is, then this yeast was found also in North America. Then this yeast was found also in Tibet. So it's found in many places on the planet. But interestingly, so far it has not been found in Europe. So we still don't know exactly how this all came into happen. But it's really like a criminal story. And if you read the different publications from different groups at different parts of the world, it's really fun to read. Because, of course, everyone wants that his yeast is the ancestor of all our lager yeast strains.
Yeah. So found in Patagonia, but it is used in beer brewing?
Worldwide.
Now it is. Okay, but it hasn't. That's a very strange crisscross of the microbes across the world, I guess.
Exactly.
Yeah.
There seems a lot of traveling involved.
There definitely does. And I always wonder how these microbes cross the world because, individually they can't really move around that far. But I guess they hitchhike maybe on other animals or...
Oh, they hitchhike. For example, particularly talking about baker's yeast, we know that they hitchhike in bees and wasps. So actually, one of the questions is, how do they survive, for example, wintertime? Because in Europe, when everything freezes, how can they survive? And how would they come back to fruits and flowers? And what we figured out is that when these bees and wasps go to flowers and fruits, they ingest also the yeast, and the yeast survives in the bee. And when it goes back to the hive and stays there over winter, actually also the yeast stays. And then, like with a helicopter, they go back in springtime to all the flowers and fruits again.
Wow. Funky way of traveling.
Yes. Also, what is very interesting is that fermentation, for example, has a connection there, because a lot of these insects are attracted also by fermentation products. In this case, it's not like for humans the alcohol itself, but more some like aromatic side products. But the insects are attracted by it, and the yeasts do it. So essentially, the yeasts kind of make fruit and flowers attractive for the insects. So they tell them where to go, where they want to be, and where they want to mingle with other yeasts.
And how do they do this? Is it a chemical signal or something, or
yes,
are they just
signal? It's like smelling. You can imagine, really, it smells good for the animal.
Okay.
It's like the beer, the fresh beer, the fresh bread. It smells very good for us. So that's exactly what also the bee has. And so the bee goes exactly there. And then the yeast is happy because it arrives exactly at the point where it wants to be.
Very nice. I'm just realizing we're only on 3 and we have 17 more minutes before our next guest comes. So I guess we should move on to the next one on our list, which I think I put down here. And this one is acromonium. Which, if I just get up this small video, I put it on this list because it is a useful one. It basically gives us the cephalosporins, a beta-lactam antibiotic. You said to me that this might be a bit boring, but I think it's cool because in general, it provides us with, such a useful group of antibiotics. I mean, I think in terms of the biology, it's a fairly mundane fungi, right? It has spores and hyphae, and there's nothing super crazy about the way it lives its life. But that's okay. It lives an ordinary but useful life. So here you have just a few shots. So I guess it lives in blackberries or on fruits or, on different kinds of plants and so on. And you can see here the spores, right? Which are how it reproduces. It produces these small spores and then they go off and find new locations and then they eventually grow into a fully grown acremonium. But I want to just move us on to the next slide because we can have a little look at this antibiotic. So here we go. Here we have the cephalosporins. So I guess it's good to talk about this in comparison to penicillin, right? So all of these antibiotics have this strange nitrogen, carbon, carbon, carbon square with a double bonded oxygen, which is the beta-lactam ring. But I think, if I remember from my studies, what makes these antibiotics a little bit more durable for treating disease than the penicillium or penicillin, which we were used to, is it has this extra ring here with a sulfur atom. And this makes this molecule much harder for sort of beta-lactamases, which are enzymes that break down antibiotics. It makes it much harder for bacteria to be able to break this molecule down because this sulfur gets in the way and this ring is a bit different to what it is in penicillin. So it provides us with a useful alternative. And I think that's really cool because, as we know, antibiotic resistance or antimicrobial resistance is a huge problem. And it's little chemical changes like small little bits of sulfur that you add into molecules, which might make these molecules last a bit longer. So for me, that's why it's on the list. I guess as a biologist, perhaps you say it's not the most interesting.
Oh, no.
Or at least.
No. Poor acremonium. I didn't want to say it's boring in itself. It was just that when we compare it to penicillium, the point is also penicillium gives us the antibiotics and penicillium is the historic one. And penicillium gives us wonderful cheeses and other things.
I mean, it is number 5 on our list.
Exactly. That's why I thought, okay, let's talk about penicillium to save time, but not to avoid acremonium, which is, as you say, the point is what we need is diversity, not only in our societies, but what we need is diversity also in antibiotics. And that's exactly what the more fungi we have, the better it is.
Yeah. And of course, the first kind of true antibiotic came from penicillin, discovered by Alexander Fleming, Florey and Chain, and I'm sure many other scientists at the William Dunn School of Pathology helped turn this into a product we could mass produce during the Second World War. I actually had the honor of being a laboratory intern at the William Dunn School of Pathology at Oxford University for 3 summers, working in the same building that they did all this process in, which was amazing. And, you really feel like you're walking in the footsteps of giants. And yeah, I mean, you find these molecules, you might find them produced by fungi or other microorganisms, but then turning it into a product that is safe and effective and also mass producible is the next challenge, right? So, we need scientists looking for molecules like this, but then we also need to work out if they're going to be therapeutically useful. So hopefully fungi can provide us with more examples of this. And here we go. Here's a nice little example of some acremonium growing on some agar. You can see the nice little hyphae here. But penicillin gives us the original beta-lactam antibiotic, but it also gives us blue cheese. And in fact, it's Penicillium roquefort, which John Warhol, back last night, was saying should be the official microbe of the town of Roquefort, where they make the blue cheese itself. And I think that's a great idea. So if you're listening from France and you're near that town, please petition your local legislators to make Penicillium roqueforti your official microbe. But there's another one, isn't there? Penicillium camemberti, which I think the name gives it away, helps us make brie and camembert. So from saving lives to giving us cheese, we really have to thank penicillium. Right, okay, we've got 10 more minutes.
Things, right?
Say that again?
They can do a lot of things.
Yeah, they can. it's amazing. I mean, I had cheese for breakfast, so it's even powering me through International Microorganism Day. Yeah, we've got 10 minutes to do 5 microorganisms, so I guess we better get our move on. So the next one is, it's a, let me just get this ready here. It's, Malassezia. So do you know about this one, Michael?
Not too much, actually. You brought it up, and then I had to look it up. And the point is also there is a lot of philosophy behind. The point is, this is a fungus that grows on our skins. And it's there for all of us, essentially. Most people have it. But then the question is, what happens?
Yeah, exactly.
Exactly, the film.
And it grows on our skin, and usually it's beneficial. And much like yeast, it does budding as its process for reproduction, and we can see a nice image here under some scanning electron microscope. But when this fungus gets out of balance, I guess, with its neighbors, you can see a little more images here, it can cause some problems with your skin. So one of the things it can do is it can start to change the pigmentation of your skin. And if you have a weakened immune system, or some kind of perhaps nutritional deficiency, it can cause hypopigmentation, which is a reduction in the pigmentation of your skin, or hyperpigmentation, an increase in the pigmentation. So I just think that's a very strange and local fungus, which can have a huge effect on people's complexion and skin. So it's a very human-centric way of looking at it, but it is quite an amazing thing that it can do. So that's why it's on my list, because it can, completely change the color of your skin. And it's normally living on everybody, right? So it's something that we all have somewhere on our skin, in our hair, and so on. You can see a little bit more of the structure here. It has a thick capsular layer and a thick cell wall. So it's pretty well defended against the things around it. Next one is Aspergillus, which is quite a famous one, actually.
Yeah, and that's one of my favorites.
Do you want to give us a little introduction while I get this image ready?
Yeah, get an image ready, because
here we go. This is what we like to see.
Exactly, that's very beautiful. Yeah, Aspergillus can also do a lot of things, but one of the things that it's really useful for is for citric acid production. So when you drink a lemonade, or when you cook your jams or something like this, and you add citric acid, that's actually usually not extracted from lemons anymore, but produced with an Aspergillus strain. And this fungus is extremely efficient in eating actually sugar and converting it into citric acid. And that's what hardly anybody knows.
Wow, okay. So, I mean, just to put it in context for anyone listening, citric acid is that kind of tang-fastic flavor that you get on sweets. So if you ever have a very sour, tangy sweet, it's usually been dusted in citric acid. But our bodies use it, right, in their metabolism in many different ways.
Everywhere. It's absolutely everywhere.
Yeah. And so they turn sugar into citric acid. Okay, that's a cool fact I didn't know. And it's a mold you might encounter, I guess, if you leave a tomato to go moldy in your kitchen and you see it go white and furry. That could be Aspergillus. And in fact, here we have a close-up, and then I guess these are the fruiting bodies ready to spore and then infect the rest of your tomatoes. And do you work with Aspergillus at all?
Yes, we do work with Aspergillus in our lab.
Yeah, okay.
We're actually looking into it, and the point is, they are always so why we as humans, we always try to explain something with our our ideas, and most of the time these questions might be even stupid because there is no really a why. But the question is, why do they produce so much citric acid? It seems completely futile, because they can really eat the sugar and convert 95% into citric acid and grow only very little with it. So the question is, why do they do that?
So that's an unknown. So if you're a young budding scientist looking for problems, there's one to investigate.
Exactly.
So another favorite fact of Aspergillum is, and we might see this later today, because after this we have a session on traditional Japanese beverages, including a tour of a sake factory. But the koji mold used in Japan to ferment soybeans, but also to make sake, is actually an Aspergillus species, Aspergillus oryzae. So it's also used in making sake. So we have another fantastic use for this fungus. I think I'll just...
They produce a lot of enzymes, and so they're very useful. Like sake is produced from rice, right?
Yeah.
And rice is very starchy, but the yeast that actually produce the alcohol wants sugar, doesn't want starch. And so the fungus actually can degrade the starch into sugar that the yeast uses.
Fantastic. And actually it gets its name from this structure, I think, called the aspergillum. Is that right? Yeah, which is the fruiting body. You can see all the spores here in this close-up. Okay, that's number 7 on our list. We have a few to pick from. Do you want to pick a top 3 or the next one from this list? And we'll try and get through the last 3 in the next 5 minutes.
Yes, let me open the list myself. Yeah, why not go directly to the next one, to Yarovia lipolytica?
Yeah, okay. So I don't have a video of this, but if you could give us a little introduction, that would be great.
Yes, because Yarovia, we talked about like when yeast gets not eternally living anymore. So there are a lot of these steps how organisms develop. And Yarovia is kind of a thing. Yeah, there you see it. You see actually that the middle picture is fantastic.
Ooh, let's go back. This one?
The one below, this one.
This one.
Good, yes. What you can see here is that the cell can grow as a yeast, like you saw it before from baker's yeast. So you have these little cells and they are budding and they look exactly like the yeast that we saw. But then you see this long hyphae also, right?
Yeah, I can see.
Much more like the acremonium or like the aspergillus. And so Yarovia is kind of an in-between on the way between yeast and filamentous fungi. So it can do both. Depending on the conditions, it can completely grow as yeast, or it can completely grow as hyphae, or it can do both at the same time.
Wow. Okay.
It's cool to study because also we don't understand how exactly that's regulated. And so that's a good thing to study it.
Okay, so at some point, some genes are getting switched on and off in different environments, and we don't quite know the process.
Exactly, and the hyphae cling to something. The other really fun fact about this is lipolytica already tells you a little bit. So it can actually degrade fats and oils and grow on them, what is special. Many things cannot.
So it might have a role to play in clearing up oil spills, perhaps, or something, although it's not aquatic, but it can definitely eat lipids.
Yes, it can eat lipids, which is biotechnologically interesting. And for example, in our lab, we are working with a strain that has been isolated from the tank of an airplane.
Wow, okay. So it's living in there somehow.
Exactly, it's living in there, and it can even degrade something like kerosene.
Whoa. So it can survive off jet plane fuel.
Yes.
And it can pretend to be a yeast type thing or a hyphae type thing.
Exactly.
Okay, that's definitely number 8. So we've got 3 minutes to do 2 fungi. So any on this list that you really, really want to get to?
So let me look. Wait a second. Well, later on we have Zigo, so that we can. So let's go to Trichoderma very briefly.
Which one? This one.
Number 10.
Yeah. Let's see if we can find us a nice image.
Because again, fungi give us a lot of molecules like antibiotics, but also a lot of enzymes. And in our quest to produce things, for example, from woody biomass, we need enzymes to degrade wood, for example.
Yeah.
And this one is one of the best producers of enzymes degrading wood. And it has been found actually in the Second World War from American soldiers on the Solomon Islands, because what they realized is that their canvas and their paper was falling apart under their hands. There was something growing on it and degrading it. And it was actually this fungus. And then we figured out that it has fantastic enzymes. And with these enzymes, we can degrade a lot of things in our environment and we can use it. And so from looking at some damage somewhere, we could get a strain that is now one of the worldwide #1 producers for enzymes.
Whoa, okay. So from degrading soldiers' papers in World War II, it's now an industrial giant.
Yeah.
So here we have a little image of it growing under the microscope, but then this is a more macroscopic sort of big spore colony thing with fruiting bodies as well. Okay, we've got 1 minute, one fungus. What's our 10th fungus on this list?
The thanks that let's go to Exophiala dermatidis.
Yeah.
It's black yeast, and that's a cool one because it's so sturdy. It grows extremely slowly, but it grows everywhere. So there's hardly any condition that it cannot grow in. It's black, it's cool, it really looks nice, I think. And it can grow, for example, in your dishwasher. So you wash your dishes with 90 degrees, you have very, very aggressive dishwashing agents, but this yeast can survive that and it will not die, it will grow. It can grow on glaciers, it can grow everywhere. And that makes it very interesting for us because really life is everywhere.
Whoa. So it can grow in dishwashers at 90 degrees. It can grow in glaciers at minus whatever.
Yes.
Okay, that is a hardy fungi.
Be happy. The point is that's a problem, of course, because you don't want a yeast in your dishwasher. So, but for the yeast, obviously, it's cool.
I mean, that's a huge range of environments that it's happy in. And it's this. So, does it make these black colonies, these black kind of groups?
Exactly.
Whoa.
Are they cool?
They are really cool. I didn't know about this one, so I thank you for adding it to the list. Unfortunately, we've run out of time. I mean, we could go on talking about fungi, it seems, for hours. And we've just managed to cover 10 in the last half an hour. We have our next guest arrived in the call. So I'll just invite her and stop sharing my screen. And we'll just check that all of her equipment is working. But yeah, that was fantastic. I mean, we could have gone on for hours, I believe. We really had to rush through just a very small group of fungi. There are just so many cool fungi out there. So we have coming up now Maria Martinova, who I think is based in New York, although I think she's originally Russian, who's come to talk to us about probiotics in yogurts and also to talk to us about the Microbites initiative. So we'll just wait a few moments to see if she can join our call. I think it's taking a little longer than normal, but we'll see. But yeah, we're going to sort of have a look at some other uses of microorganisms in what could be your breakfast. So probiotic drinks, yogurts, and so on, all require microorganisms. And I'm trying to think what other breakfast foods. Well, I mean, yogurts. There's a particular kind of yogurt here in the Netherlands called quark, which is a very thick yogurt, which again is going to have its own fermentation process. And obviously, cheeses. I mean, what I love about cheese is that it can be made by so many different microorganisms, and yet we still call it cheese, even though it's kind of an entirely different organism working around it. What is your favorite cheese, Michael, if you had to pick one?
Oh, that's really a hard choice. I don't want to choose because cheese is really, but I really like the mountain cheeses from the high mountains that are really old and you know.
Yeah. Okay. Do you mean they've been matured for a while, or they have been? Yeah. You see, again, I think it is hard to choose a favorite cheese, but one I've really got a lot of love for is Comté from France. Very like nutty, hard, delicious cheese. Really full of flavor. You can taste the salt crystals in it. Maria, can we hear you? I think you might have to unmute, but you've joined us.
Yes. Hello.
Hey, how is it going? Are you in New York? Is that correct?
Yes. Yes, I am.
Fantastic. Let me just get us up on screen briefly before you begin. And yeah, thank you again for joining us so late in the day in New York. I believe it's nearly 2 a.m. there.
Yes. There's a lot of light around me, so it seems like it's a day.
Yeah, fantastic. And well, you've come to talk to us about probiotics in microorganisms. So yeah, I will give you the floor, as it were, and let you begin. So if you just want to share your screen,
of course.
You can give us a little introduction to probiotics, and we'll have a little discussion at the end. And you're welcome to begin.
But I mean, great.
Fantastic.
Okay, you can see my screen, right?
Yeah, we can.
Awesome. Okay, so I want to start my presentation with a little bit introduction about myself. My name is Maria. I am an editor, writer, and translator in microbiotes, and I hope we're going to have time to talk about microbiotes and what I'm doing. But what I'm going to be talking about today is probiotics and yogurt and the truth that hides behind the label that you see on the pack of probiotic yogurt. So before we dive into probiotics, I created this infographic to talk about history of yogurt, because it's truly fascinating how, long ago it was created by accident, I think, and we're still consuming it up until this day. So I think, as I said before, I think it was created by chance. It was an accidentally discovery during Neolithic period, because there's an evidence from ancient pottery showing that people in that period of time were using pots to store milk. So obviously they were aware of milking cows, and they had cattle. So it was pretty, easy to figure out. But the real history of yogurt starts in Middle East, when herdsmen were keeping their milk in goatskin bags that were hung across their camel's backs. And while they were traveling, the heat from the camel's back and the juices coming from the bag, they're all contributed to the fermentation process. And, the stories about nomadic tribes traveling around the deserts reached Rome, and Pliny the Elder, who you could call a journalist, mentioned yogurt in his notes, saying that nomadic tribes knew how to thicken the milk into a substance. And Turks were aware of medicinal uses of yogurt. They were using it for treating illnesses and symptoms such as diarrhea and sunburned skin. And a funny story, my grandparents, they would use sour cream when they had burned skin, because, it heals burned skin. Yogurt was used as a cleaning product and beauty lotion. And going forward in time, yogurt made an appearance in Europe only in 16th century, 1542, and it was brought by King Francis I, who was suffering from diarrhea, and it was believed that the yogurt cured his condition, but as we know now, probably did not cure, just alleviated the symptoms of diarrhea. But it was 16th century, so people believed a lot of things. And in 1904, scientist by the name Ali Mechnikov developed a theory that aging is caused by the activity of toxic bacteria in the gut, and lactic acid bacteria that were found in yogurt could potentially prolong life. And he made this observation by looking at people who lived in Bulgaria, because they were pretty young. They looked very young, and they consumed a lot of yogurt in their diet. So he kind of, made this conclusion, and we're going to talk about Mechnikov a little later. And in the 1930s, Daniel Carasso started a yogurt company that later became Danone, world famous company. And pretty much from 1930s, yogurt became commercialized, and people had access to it. They could make their own yogurt at home, but they would also, like the variety. And today you could go to any store and you will find different types of yogurt, different flavors of yogurt, whatever you like. So let's go back to my presentation, and we'll talk about probiotics in yogurt. So probiotics definition of it is, let me just do this. Okay. So probiotics is, probiotics defined as live microorganisms, which when administered in adequate amounts confer a health benefit on the host. Pretty much any organism can be probiotic, but it's a really tricky definition, because in order for an organism to be defined as probiotic, it has to go through many, many stages of identification and defining properties, health properties. But usually usually it's considered Lactobacillus, genus Lactobacillus and genus Bifidobacterium are considered as probiotics. Probiotics must be alive when administered and must have documented health benefits. Now, when I talk about health benefits, it either means drug type effect or food type effect. And drug type effect means, for example, alleviate symptoms of diarrhea. So it's something similar to when you take a medication for diarrhea, for example. And health type benefits, for example, it helps your immune system to fight diseases. So doesn't mean that everything is a probiotic. The short answer is no. The long answer is absolutely not. It does not mean that everything that has some sort of bacteria in it means that it's probiotic. A lot of people think that probiotic equals live active culture, which is not true. Live active cultures are used in fermentation, and unless those cultures that are used for fermentation define as probiotics, these two things cannot be equal to each other. Same with native beneficial bacteria. They cannot be really native because probiotics usually taken orally through yogurt or through medication. So they're not really native to our bodies, but they are greatly digestible. You know, we readily, unless you're lactose intolerant or, you know, any other condition that you have, but probiotics usually easily digestible in our bodies. And probiotic must be defined, and it's a really long process of defining probiotic on a bacterial species, on a genus species. Plus, if a scientist decides to say that certain bacterial strain is probiotic, so it means it has probiotic properties, this bacterial species needs to be added in an international bacterial collection, so other scientists would be able to reproduce the results that the first scientists received. So it's a really long process. So this is a brief history of probiotic history, timeline of probiotic history. We can see that probiotics came to play around 160 years ago, roughly. It's 1857. Pasteur was experimenting with lactic acid bacteria. He determined the fact that lactic acid bacteria contribute to spoilage, spoiled food. And then a lot of scientists started diving into this topic. Lister in 1878 isolated lactic acid bacteria from milk. Mechnikov, the one that we were talking about, described Bulgarian bacillus associated with health. In 1930s, after commercialization of yogurt, Japanese microbiologist Shirota isolated a bacterial strain that was called Lactobacillus casei Shirota after him. But then the name was shortened, and Lactobacillus casei is still used in fermentation processes when making yogurt. And then from 1930s, you know, up until nowadays, people really started diving into the whole topic of probiotics. In 1953, Koloff but he used the term probiotica. 1965, definition of probiotics. 1989, Fuller defines probiotics. And only in 2001, which is only 20 years ago, World Health Organization gave the official definition of probiotics. And then in 2003, when people started applying genetic tools, sequencing bacteria, all that stuff, the first genome sequence of a probiotic bacterium was done only in 2003. So it's 18 years ago. Knowing that lactic acid bacteria were known for 160 years, and only 18 years ago, they actually started to, you know, sequencing stuff. That's interesting. So I would like to quickly talk about yogurt market in Europe and the US because it's quite interesting. I grew up in Russia, so I'm really familiar with Europe yogurt market. But in the US, it's quite interesting to see that not a lot of people consume yogurts here. They consume a lot of cheese, that's for sure. But it's not yogurt, and definitely not a probiotic yogurt. Pretty recently, people became more aware of health benefits that yogurt has, and they started to consume Greek yogurt and regular dairy yogurt. Top brands in Europe are Danone, Lactalis. I think that you're more aware of them than I am, but even in the US, Danone is something that people consume. Chobani is a very interesting brand that we're going to talk about in the next slide. And leading market in Europe is Germany. And in the US, it's New York State and California. these two states consume the most amount of yogurt, so they produce the most amount of yogurt. So what is interesting about Chobani, when you go to their website, is the amount of product that they have. If you look at it, they have 0 sugar yogurts, they have milk, they have yogurt, they have creamers, they have fermented plant-based drinks, aka probiotic, they even have cold brew coffee, which is a lot. And what is interesting about this brand is that it takes 45% of the whole market, of dairy market in the United States, which is a lot compared to the previous brands that I talked about over here, 45%, and the rest is shared by these 3 brands. So what is interesting, again, about this brand is the way they advertise their products. They have a great variety of products. They're very appealing to an eye. They write this really big labels. Oh, this is a probiotic yogurt, like here, peach mint flavor. I haven't seen really a lot of those in the market. They have very colorful bottles, and it's just interesting to see the marketing of it. So based on the marketing alone, there was this study done in 2012, and the goal of the study was to determine what makes a consumer buy dairy products, because they're heavily advertised, and obviously people buy them for their health benefits, but they also buy them because they're advertised properly. So it was determined that individual attitudes and beliefs towards health have become important factors in food purchasing and consumption decisions. And these are components of the belief, of the model called health belief model. It was proposed by Eugene Rosenstock in 1990s. He was a social psychologist who studied consumer's choice, and he studied why people tend to choose certain things out of the whole variety on the market. And it turns out there's many reasons for milk consumption and yogurt consumption. HBM believes, oh, I want to be healthy, that's why I consume yogurt. Or, oh, I think that food plays a great role in my diet, so I will be consuming yogurt, so I will be healthier. Or people who believe that they don't consume enough yogurt, for example, they are willing to pay more for additional nutrition information and vitamin enhancement in yogurts. For example, additional calcium. Sometimes you see on the package, oh, it has additional calcium. Oh, it has billions of bacteria. And people who don't do their research and maybe who don't know a lot about probiotics and yogurt tend to buy it. And there are many more other reasons listed in that paper for why people buy certain products. And what is interesting is, despite the advertisement and proclaimed health benefits, do probiotics actually work? Because for probiotics to work and be considered as a probiotic, it has to survive the transit through gastrointestinal tract, acidity in the stomach, recovered from feces usually, because that's how bacteria are extracted for analysis. And all of that should happen in a pretty short amount of time, because bacteria tend to, disintegrate in the body. So if you want to really see how viable it is, it has to be extracted fast. So there was this study done in around 2010, where a group of volunteers was consuming yogurt for 7 days, and at the end of the experiment, there were these 2 bacteria extracted as Thermophilus and Lactobacillus bulgaricus. These are the most commonly used bacteria for fermentation processes, especially when someone makes yogurt. So they concluded that since they were consuming yogurt and it has those bacteria, and the bacteria that they extracted were alive, it means that it works. Which is true, but it has a lot of discussions around this topic of how beneficial, or actually like how powerful probiotics are when they're consumed from a yogurt. So great variety of controversy. I'm going to talk in the last couple of slides about two products by Danone, Activia and Actimel. I think that you guys are familiar with it. Again, when I was living in Russia, that was my chobani. I would always get a bottle of Activia because it's easy to carry around. It's, good for my body, all of that stuff. On the bottle, you can see it says billions of live and active probiotics. People tend to buy it because they don't really know what a probiotic is. They're not really familiar with the concept of probiotics. So, as I said, Activia is a brand of yogurt classified as a functional food by Danone company. Danone developed a specific strain of bacteria called Bifidobacterium animalis, and it's marketed by Danone under the trade names of all of these bacteria. So if you go to a store and you decide to buy a Danone product, you're going to see all of these bacteria listed on the label. But I think that if you're going to buy a dairy product, a yogurt, for example, from a different brand, you're not going to see this bacteria. So they're very specific to Danone products. And in 2003, the probiotics market was worth almost $10 billion, which is a lot of money. And right now it's been 18 years. I think now at this point it's worth so much more than $10 billion because people tend to, take care of themselves more, and they prioritize diets and healthy eating and clean eating. And it is a fact that Activia, because it is advertised as functional food, it costs more. On average, a bottle of Activia costs $2 more than a regular yogurt because of this Bifidobacterium animalis that was developed by Danone. But obviously there's controversy around this. There were a lot of lawsuits around, actual health benefits of probiotics. 2010, Federal Trade Commission said that Activia packages are deceptive. Danone exaggerates proposed health benefits. 2008, class action suit accused Danone of mounting a massive false advertising campaign to convince consumers to buy Activia products. I don't want to take sides here. Obviously, I'm just reporting what is known, but I was really surprised about reading about this stuff because I would never think that someone would, say, oh no, it's not true what's written on the label. The other product, Actimel, also Danone product, it is advertised as probiotic product. And it was invented in the 1980s when Danone took the first steps to develop a fermented milk based on Lactobacillus casei. We talked about it, the one that was discovered by Shirota in 1930s, all that long story. And then it's claimed to have benefits ranging from reducing the incidence of diarrhea to the improvement of immune function. Obviously, when you hear this combination of probiotic and then benefits and then reducing the incidence of XYZ, obviously you want to buy it. Even British Medical Journal 2007 said that Actimel could help avoid antibiotic-associated diarrhea. We all know, antibiotics have a tendency to disrupt our gut microbiota. So obviously the sales for Actimel skyrocketed because no one wants to deal with consequences from taking antibiotics. But again, there's a controversy around it. Alexa Mayer, who works in the Department of Nutritional Sciences in the University of Vienna, compared Actimel drink with a normal yogurt and found no significant difference in the effect of Actimel and normal yogurt. Moreover, she said that if a person would regularly wash their hands, take care of themselves, exercise, eat healthy, and consume normal yogurt every single day, that would have exactly the same benefit as Actimel. So you don't have to spend extra money on Actimel drink because you can get it for free, just by taking care of yourself. But again, another controversy in 2005, group of scientists said that there is a measurable health effect, measurable health benefit linked to the presence of live Streptococcus thermophilus and Lactobacillus bulgaricus in yogurt. So it's really hard to say where the truth is. In my opinion, I just want to say my last words: eat your yogurt, do your own research, do not solely rely on advertisements. if you would like to choose a yogurt for your special health benefits, for example, you want to improve your digestion, maybe you have to look up for some interviews with dietitians and nutritionists, and they will help you to choose the right yogurt brand, or maybe will help you to learn how to study the label on the yogurt. And that's it from me about yogurt.
Well, thank you, Maria. I hope you're doing okay over there, because it is now 2 a.m. But it does mean that you're in International Microorganism Day, just about by a couple of hours. So congratulations, happy International Microorganism Day. And thanks for delving into the murky world of probiotics, because I guess it's one of those situations where you have big business, but also perhaps quite a useful supplement. So there's always going to be a bit of back and forth between the truth, but also there is a lot to get out of it. One question I had was, why don't Americans eat that much yogurt?
That's a great question. I really don't know. I think that because it was not as common here, I don't really know. Like, they are big on cheese and big on other dairy products, not yogurts. It's really became a very recent thing to consume yogurt.
Yeah, okay. So it's more of a cultural thing. It's like a new trend rather than an old way of eating.
Yes. I would say that maybe, immigrants, they would make their own yogurt, but it wasn't as commercially available and people would not pay as much attention to it as it is now. They would consume smoothies. I mean, not smoothies, milkshakes.
Yeah, okay.
But,
the American milkshake is quite an icon. So we have 5 minutes for our next guest comes. So I just wanted to know if you wanted to chat about the Microbites initiative in our final few minutes.
Yes, that would be great. So basically, there's a group of people who love science, or love microbiology, and they want to write about their research, and they want to, tell people about microorganisms, and this group is called Microbites. I am writing articles and editing articles, and I'm also a translator. So I fluently speak Russian, and I am trying to reach Russian audience by translating articles into Russian. But there are a lot of other languages available, German, Spanish, French, that people could, explore more in different languages, which I think is great. There's a lot of undergraduate students, graduate students, people who are getting their masters and PhDs, and who just want to share their knowledge about bacteria.
Mm-hmm. And so we have all these blogs. Do you have any other output from the initiative? Do you ever have events, or is there a Microbites meetup that ever happens?
No, there is our internal Microbites meetup, because the last time we tried to have a meetup, there was problems with internet connection, and we got really upset. So we're looking forward to it, and we're going to be discussing new stuff that's coming out. So probably after that meeting, we're going to have something interesting. Okay, great. And what's your plan for International Microorganism Day? When you wake up tomorrow, how are you going to celebrate? I'm gonna, I don't know. That's a good question. Probably gonna get probably gonna eat a bowl of yogurt. I have this beautiful new yogurt that I bought, and I'm dying to try it. So can't wait. And what brand of yogurt's this one? Oh, there is this store called Trader Joe's. I don't think you've ever
I've heard of it. I've never been.
Oh, no. Okay. So what I like about it is that they sell their own produce only. They don't really have other brands, and they have their own yogurt. And as a microbiologist, I studied the back of the yogurt. Yeah. And I really like it. So. But any good species in there that we should know about in this yogurt? I would say Lactobacillus casei. That's the one that, usually stands out. I also take probiotics, like pills, like individual every single day, because I don't think I consume enough yogurt, and I don't want to pay extra money for extra nutrition in the yogurt. Fair enough. I've just seen our next guest arrived. Maria, you're welcome to hang around in the call for the discussion at the end, but I appreciate if you need to sneak off because it's late in New York. I might have to, yeah. So thank you so much for coming and giving us such a good overview of probiotics in yogurt.
I hope I get
Thank
to
you
eat some
so
yogurt
much.
today and have a
Please.
fantastic time. Sleep well
Thank
and
you.
enjoy International Microorganism Day.
Thank you so much. It was a pleasure.
No worries. We'll hopefully do some blogs or something for IMD and Microbytes crossing over somewhere. That would be really cool.
Okay, I will pass it by.
Yeah, awesome. Right.
Thank you.
Bid you farewell. Have a fantastic International Microorganism Day. Our next guest, Paola Branduardi, or Branduardi even, joins us from Milan, I believe. So we'll just bring her into the call. And she's here to talk to us about a very, very hardy and tough yeast. So we'll just check we can hear and see her.
Hi, can you hear me?
Yeah, we can. How are you doing, Paola?
Good morning.
Good morning. Happy International Microorganism Day.
Thank you. Same for you.
So you're here today to talk to us about a very, I don't know, tough, hardy, bizarre yeast. And I'm going to let you get on with it because you're the expert here. But you're going to give us a very fascinating talk and then stick around at the end to just chat about fermented food in the final few minutes of the breakfast show. But I just want to check with you, have you had your breakfast this morning?
Not yet.
No. What will you eat when you get round to it?
Yeah, I don't know, but yeah, I mean, it was a good opportunity anyhow. And so, you know, when you wait a little bit longer, then you will enjoy even better. So
Yeah.
this is what
Fair
I
enough.
will do later on.
So,
And
I mean, I had
actually then
cheese.
I also have to attend a project meeting, so they will, okay.
Cool. Okay. Well, let's pop
Yeah, I did
ourselves.
the same.
Yeah,
saved
okay.
my breakfast for after the show.
Yeah, and then you
So
will
I'm
really
looking
deserve it.
Forward to it, exactly.
Nice. Okay. Well, we'll get out your way, Paola. And if you want to share your screen, if you have slides, then you can tell us all about this yeast.
Yeah, well, thank you. And let's see. I mean, I have some slides, but let's see how it goes. I mean, I'm also in your hands because, yes, I want to talk about this yeast, but also, I mean, you said before, it's also a story of a yeast and a human. So, you know, it's really connected in a sense. And you said before that I'm expert in this yeast. I would say more that I'm beginners with this yeast because I keep on learning from this yeast every day. So I'm not sure I'm really an expert because it's.
But compared to others, it's 20 years we are laughing
It's only
about
that
that
this yeast
sentence.
is putting me
I love the
in
modesty
corner
of scientists.
more or less every day. So this is why maybe. Yes, sorry. Yeah, you were a little bit frozen. Yeah, maybe it's my connection.
I think we have you back. Yeah, you froze a little bit, but we have you again. But like I said, I love how modest scientists will be. They'll study something for 35 years and be like, oh, I'm just a beginner in this subject. Forgetting, of course, that no one has anywhere close to the knowledge that you have about this organism. So let me see if I can pronounce the name of this organism. So is it Zygosaccharomyces bailey?
Yeah, or Bailey. I
Bailey,
don't know.
okay.
But Bailey, yes, it's only. I mean, it's all accepted. Let's put it in this way. He, in a sense, I mean, he will not complain. So no, but what I want to say also is that let's say very often. With a microbiologist, it's also, well, let's say I talk about me now. It's a story of prejudice also. And I want to shortly say that I became a microbiologist despite my initial idea. And so this is what I think I like most of my job now, because it was a discovery. It was like, serendipity. I would have never guessed to be so fascinated about a microorganism. I was studying, let's say, the development and differentiation of neuronal systems before approaching to yeast. And so for me, it was just a continuous discovery. And then the part on metabolism came in the scenario even later. And that was also another discovery. So what I'm saying is that what I like about microorganisms is that they put myself in front of my tendency to have prejudice, despite I'm thinking that I'm an open person. And so this is what I like most of my job. So just let's say message that I wanted to give you. And yes, so I mean, I can also try to show my screen because, yeah, I don't know if you can. Yeah, I don't know, but I prepared that.
Oh, wicked. Yeah, we can see those well.
You can see my, yeah.
Yes,
So, but I prepared that just because, the story with this yeast, it's also a story of friendship because of this yeast, I met Michael. So in a sense, I have to say that the first picture is for him because we always laughed a lot about the point that this yeast has been isolated from pickles. And for us, it was always very funny. And indeed, because this is known as a spoilage yeast. So it was also isolated from other food. And then you might say that our initial question of having breakfast with Zygosaccharomyces bailey maybe is not a good idea because it will spoil our breakfast very soon, in a sense. And it can also spoil our evening, let's say, pleasure moments, because it can also spoil wine. You know what I mean? So then this is how it starts. And also, this is why I said it's also a story of prejudice, because, this is only our vision of a story. I mean, this yeast is happy when it's spoiling our food because it's eating. And so obviously, if you stop thinking about the damage that you have to your food, to your drinks, then you would say, yeah, this is a bad yeast. And, it's always obviously an attitude that you can have with many microorganisms, because first you think that they are dangerous or they can bring some, pathology and so on. But in a sense, this is also nice because if you isolate this pre-concept from that, you might start thinking, okay, but so why is it so, let's say, robust that can survive within the pipeline of production of our food and fermented food? Because obviously, very often we add additives that are preservatives. So how is it possible that it can survive out of that? And like in jam, marmalade, you have a lot of sugar. Usually this inhibits microorganisms to grow because it's too much pressure for growing. So then if you start thinking about that, maybe you can consider from a different perspective, let's put it in this way. And to add another image that I think is my favorite funny image, this yeast is so good in fermenting, very fast food, even when there is a lot of sugar, is that produce a lot of gases, and it can be even exploding can containing food. So this is another funny concept that I always found. And, so when I started to work with this yeast, I was thinking, okay, but is it dangerous? What happened when I start my kinetics? But then, as I said, I think it's very nice to think that if you consider that it is recognized as the most preservative-resistant organism known, then you can think what these preservatives are. And could it be that we can turn, the coin to our favorite side? And this is what comes my story and also the story of Michael, actually, because we started to work together with this yeast. And we started to think, but can we turn this potential foe into a friend? And why am I saying that? Because Michael and we are really fascinated about the point that these microorganisms, you mentioned before, you were talking about citric acid before. So these microorganisms can produce this chemical platform. This is what we call them, because, it's like these Lego pieces. And with these things, we can do so many things. And if you think about producing them from oil or from other fossil resources, then this means that you are depauperating the resources of the planet and producing, a lot of impacts on the environment while you are producing, while you are let's say, processing them, and also at the end of life of these products. But if you think back about what I said before, that these microorganisms can really eat a lot of things, produce a lot of things, and then these things goes back to nature. So then maybe we can really think about it in a different way. And this is why we were thinking about using these yeast as a small cell factory, and possibly engineering it, and then maybe Michael might have produced a lot of these organic acid that you mentioned before, because you can see they are different, but they can really go in different sectors that are really important for our society. So if you can convince this yeast eating, maybe we can think later on what this eating is, then you can produce a lot of organic acids, and then these you can use. And so this is what I somehow meant. The message was that very often we consider microorganism, let's say from our point of view, but our point of view very often is limited to the things that we are doing in that moment. But if we try to enlarge our vision and then to see, then you can really even turn things in a different way, and you can learn a lot. And what I mentioned before is that you keep on knowing these yeast. And what I can tell you is that at one point we thought, okay, we understood. So we can start manipulating this yeast, engineering that. And then from our collection, we took out our favorite one, because these microorganisms have many different strains, and then everyone had his own favorite. But then it turned out that our favorite strain was a hybrid. And he was laughing to us, because every time that we were trying to engineer that, at the very beginning, we were completely puzzled. And it's just because obviously his genome was like a mess. And this is where we are now. We understood that. So we understood another point. Let's put it in this way. And I think that this is, I can even skip. I think that anyone is a little bit like bored about having presentations, but I think that some images were nicer than my face. But the message is that it's really a way in which we are, let's say, looking at microorganisms. And also another thing that it's funny to think is that, for example, you have a microscope in front of you. That it's really the instrument that is, it was the way in which von Leeuwenhoek made our unknown unknowns evident, because before we didn't know what we didn't know. And so you go really in the very, very, in the world of the very, very little. But then you can only understand microorganisms if you can have at the same time the very big picture. And this is also another lesson from one of, let's say, the more relevant scientists of our field, that is Winogransky. So he was saying you cannot understand anything if you bring one microorganism in your lab and you just look at these microorganisms isolated from everything else. And so I think that the lesson that I learned more from microorganisms is to always have like a narrow bell ringing against prejudice. And this is what I wanted to say.
I mean, that's part of why we're here today, is to try and remove any prejudice people have against microorganisms. So I wanted to know, is this yeast, Zygosaccharomyces bailey, present in all pickled jars, or is it just we have found it in pickles and that's cool? Or does it love living with pickles, or would it rather be somewhere else?
Well, obviously this I cannot really know, and it's even not the only one. It belongs to a genus where you have a lot of other yeast that are resilient against, for example, high concentration of salts. But what it is true is that obviously it might happen that during the pipeline of production, you have this kind of infection. And this is why they are really, let's say, considered like spoilage. And there is a lot of research actually trying to understand which their, let's say, weak points, because then when they start entering in the production chain, then you have, I don't think that, yeah, it's so common that if you have your own pickles at home, you might have that in. But yeah, it's also a story of competition, you know, because very often it was also found in fruit together with Saccharomyces cerevisiae. You mentioned it before. And so how they can cope with that. So Saccharomyces cerevisiae goes first. It ferments first, but then you have also some bacteria and they accumulate acids. And so Saccharomyces cerevisiae has to stop. And then this is when Bailey comes. And so it's really a question of balancing and evolution, and also how they evolved. For example, sugar transporter. Bailey prefers fructose. So yeah, and cerevisiae prefers glucose. So this is, yeah, nice story of how they can combine in nature and create niches. That is what makes our life possible.
Yeah, it's fascinating. I want to know, does this particular yeast, has anyone tried using it to brew anything or to make products that we could drink or eat or anything like that?
Well, yeah, there is something around. Actually, there was a company in one of our European projects that was interested to try something. Also because you know that, yeah, there is a moment, some concerns about the fact that because of the climate change, a lot of times then you have grapes, when they are good to be collected, then they have a very high ethanol content. So you might be interested in brewery or in winery, I don't know the term, in producing wine with some yeast that produces not so much ethanol as cerevisiae. And so Bailey is something in the middle, let's put it in this way. So there is some interest, but at the very moment, I don't think there is a product produced with that.
Okay, well, maybe one day. And I wanted to know, is there a thorough understanding of the mechanism it uses to remain hardy? Or are there parts of it that are still under investigation that a young scientist could decide, this is my thesis area?
Yeah, I mean, if someone is thinking about that, yes, please come to my lab. Yes, there is still a lot to do. I mean, I think that it's also good that I think that this day has been promoted by the University of Lisbon. And there is a professor that is Isabel Sacoreia, and she is one of the most, one of the experts on this yeast, and she worked also a lot. And so it's also, that is also nice because phenomena like robustness are something that we as scientists define like emerging properties, which means that are properties of the system. So if you isolate the single pieces, you cannot understand. So you understand one part of the problem, but then you cannot reproduce it. So this is the beauty. So you just have to understand how the different pieces belonging together then develop this trait. And this is robustness. So there is something related to the membrane. There is something related to some enzymatic activities that preserve something. There is something related to some specific transporters of ion, energy, and yes, but there is still a lot. And then when you come to hybrid as our favorite, what you also see that then they start to recombine the traits as for the Pastorianus that you mentioned before. And this is also something that we are at the very moment investigating, because then the two genomes contribute differently under different stressful situations. So yes, there is a lot to do. So yeah.
Fantastic. Michael, do you have any questions for Paola?
Yeah, the point is, one thing that I wanted to talk about, so you talked about robustness, But the point is, we are also very much involved in microbial stress. And, you know, like we talked that yeast can age, yeast can be stressed too. And that's very interesting. So can you tell us a little bit about stress in context of yeast cells? How can a cell be stressed? So how we are stressed, we know.
Yes, so the point is that, again, let's put it in this way. You can look at this question from, let's say, looking at yeast staying in nature, and then if you think about using these yeast as cell factories, as we want to do. So obviously, if we look at in nature, so we know already that they had to face stresses, and especially it's because of competition, so they have to try to find their own niche. And so their genome has already adapted, or the potential that was in the genome was already expressed to counteract this stress. And again, if we compare that to what is happening to us, is like really a moment that, when you acquire this kind of trait, is not a continuous moment. It's one of these jump off, you know, it's like we talk a lot in this year about resilience. And so the resilience in principle is the capability to grow within the stressful condition. If you have some abilities. So it's really the moment in which there is something that emerged. And this is nice because if we keep this concept in our industrial processes, then we know that we are stressing the cells in a way that is completely different from the stress that they have in nature. Because, for example, we want them to eat like, you can still hear me because my connection seems a little bit weak.
Ooh, I hope we get you back. Yeah, we've just lost you for a moment. But this is the way with live streams. As you have people jumping in and out from all over the world, you might expect a little bit of a technical delay, but I think we've still got you. Yeah, okay. You're back.
Good.
There she is.
Okay, sorry, but yeah. And there is also a thunderstorm. So here in Trieste,
oh, wow.
Yeah. So then the point is, for example, when we go for industrial bioprocesses, we want to have something that is sustainable. So usually we want these yeast to eat what we discard out of our. They are not in ideal condition because maybe we want a specific temperature, pH, or whatsoever. And then we want them to produce one specific product at a very, very high concentration. So it's really something that they are not used to. So it's all a stress. And so if we manage to balance in a way that the stress can evoke something that is in the genome without killing them, so then maybe we can make one of these jump and produce something that was hidden in the genome, but we can, let's say, make appearing with our processes. I don't know if I was, that was in the direction of your question, Michael.
Yes, exactly. The point is that's very similar to what happens to us, right? So instead of roaming around in nature and be happy, we are sitting in an office under really unnatural conditions, and we should be productive and, for example, write a single report all day long. And so then still people think we should be efficient. So that is stressful. And that's exactly what we do to our microbes.
Yeah. So we're living in our equivalent of a strange petri dish, I suppose.
Exactly.
So I just wanted to move us into the final part of the breakfast show, which is when we're going to discuss fermented foods. And one thing I wanted to share, because it is so relevant, is that we have a blog on the FEMS website, in the FEMS Micro blog, called What's in Your Kombucha? So kombucha is this fermented tea drink, which comes from the East. And one of the things I wanted to draw our attention to is the microbiome of kombucha. So it is a yeast bacteria hybrid or a mixture. And the kind of slimy, rubbery surface gloop that forms is usually called SCOBY, which I think is an anagram for something like surface, blah, blah, blah. I can't exactly remember it. We can Google that in a second. But one thing I wanted to draw your attention to were the species that they find. So there was a study put together here, which I think we might get. But anyway, and they looked at the different species in kombucha. And this study found Candida was the highest prevalence, but in another study, they identified many fungal species, including Zygosaccharomyces bailey. So it sits there in our kombucha, helping to ferment this tea into a delicious drink. And then another one, which I have a lot of love for, is Schizosaccharomyces pombe. And the reason I love this particular one is the name. So this species of yeast was discovered in East Africa, and pombe is the Swahili word for beer. So that's why it was named Schizosaccharomyces pombe. What are your guys' favorite fermented foods or drinks? I mean, kombucha is kind of a new one. We looked at yogurt earlier on in the breakfast show, and I just had one more thing to show you about yogurt, which is to remind everybody that for International Microorganism Day, we have had produced a wonderful set of education resources. Can you see this now on your screen? Yeah? Okay. Including how to make your own yogurt at home. From last year, we had how to make sourdough, which Michael and I discussed in the previous breakfast show. Here we're using, I think, baker's yeast to kind of proof the dough and turn it into a sort of more acidic dough. But here we have instructions on how to make yogurt. So you need a very simple set of things, a little bit of yogurt, and then you can use this with your class. So, I mean, if you're a teacher waking up today and you haven't got a lesson plan, here's a nice way to teach your children about yogurt. So we talked about yogurt earlier on. We've talked about kombucha. We mentioned cheese a little bit earlier, but, you know, what are your guys' favorite fermented foods? And I have a few blogs as well from the Microbites Initiative, which dig into this, but I just wanted to check with you, what could you not do without in terms of the foodstuffs that we love?
Oh, that's very difficult. You're always asking for difficult choices.
I know. It's to make you think and select.
Well, I can select only one.
I'll let you have two.
So if it's two, I would say it's cheese and beer.
Okay. And Paula?
If it's two, I would say cheese and bread. And for me, there is an additional challenge because it turned out in 2013 that I am celiac. So then there is also a lot of research in that because the flavor without gluten are more difficult to brew, to bake, and to make a dough that is anyhow excellent as the one that you have with gluten. So I think also this is a very nice field of research.
So do you still eat bread?
Yeah, yes. I mean, that is something that I would not call bread.
Yeah, that's a question of debate if she's eating bread or what that is.
Do you just have to limit the amount you take? Yeah.
Yes. But anyhow. Yes, so bread and whatever is around bread, let's put it in this.
So we just lost you for a second, but I think we've got you back. So again, we heard from the Microbites Initiative earlier on in the call, and they have a series of wonderful blogs actually on sourdough bread and its aromatic microbes. And this is the process that we know and love now about making sourdough, a big craze during lockdown, but it produces all of the gas and raises the sourdough bread. And we have here a nice little infographic on, I guess, both our foods involved beer and bread from your choices. They all use Saccharomyces cerevisiae.
So at this point, we are still looking at the yogurt thing.
Yeah, well, we have it over here. Wait, oh, sorry then, I haven't switched over. This is always the way with all the windows I have available. Here we go.
Yeah, there we are.
So this was the Microbites website. Maria from earlier helped put together the Russian section, which unfortunately I can't read, but it looks very nice. There's no Italian section, Paola. I'm sorry. Maybe you could help them put together one. But yeah, so we have some nice infographics here about creating sourdough and the active starter culture. And yeah, a nice little infographic about Saccharomyces cerevisiae. So there's some cool facts here. And yeah, I mean, its age of use is fascinating. So if it dates back to 7000 BC, it's been used for over 9,000 years, which is probably one of the oldest microorganism-human relationships that we've cultivated. And not only do they have it on sourdough, they have it on cheese, which I think you both chose as one of your favorite foods. So there's plenty of information here. And then there's a few more interesting and perhaps lesser well-known facts about fermented foods. One I found very interesting was that you have to be careful about bacteriophages, because if too many bacteriophages get into your culture, they can kill off all the bacteria. Now, I know that bacteriophages affect bacteria, but Paola, is there a yeast equivalent? Is there some kind of yeast virus that you have to be careful of when you're looking at brewing things with yeast? Or do they happen to, you know, have a little bit less vulnerability to viruses? Ooh, could you hear me? I think you might have frozen. Michael, do you know the answer? Oh, we've got Paolo again. Yeah.
Yeah. Yes, no, they are less sensitive to that. Usually this is typical for bacteria, but then this is usually one point that is in favor of the people that work with yeast when they suggest to use them as cell factories for production, because they are less sensitive. So usually, really, this is something that distinguishes bacteria and yeast. And let's say implemented yeast, there are no, let's say, examples of phages that attacked that. So, but again, also, yeah, I mean, I think at the very moment anyone knows, but also the story of phage infection turned into something that we now can use, because the story of this immune system that the bacteria has against the virus turned into the discovery of the CRISPR system. And so now that we know that together with these endonucleases can be used for targeted editing of genome. So again, it's another example of something that can turn into something useful, or something that can open our mind to new things.
Okay. So yeah, in yeast's favor, their ability to survive viruses, which are a bit better than bacteria then.
Yeah, it is relevant. So for example, like after World War II, there were a lot of fermentations for butanol production with bacteria. And so there were different reasons why we are not doing it anymore. But one of the reasons was phage infections, because if a production plant was once infected with a phage that would kill the production organisms, it would be hardly possible to take this plant into action again, because they couldn't get rid of these phages. They would always disturb the production. And that actually does not happen with yeasts. So for example, for baker's yeast, I don't think anything is known that could do that.
No, okay. Never heard about that.
That's nice. Good, healthy yeasts. We had a talk last night by John Warhol, and an initiative he was trying to get off the ground is the idea that every location should have an official microorganism. So I wanted to know, Paola and Michael, in the area you're in Milan and Vienna, where you normally are, Michael, what makes the most sense to be the official microorganism of that city or that province?
Well, I think for Vienna is quite clear. It should be baker's yeast.
Okay, yeah, from all the beer brewing and bread making.
The point is like special, like lager beer brewing, a special way was actually invented in Vienna. So then like how to produce yeast for bread making, the system to make these cubes, you know, that was invented in Vienna. And like many specialties of the city, like Vienna is a city of wine, so that's not possible without yeast. And there's a special cake that is produced with yeast. So actually, what we tried since some years is to get a monument. I want to erect a monument in Vienna to commemorate the yeast and how important yeast is for our society. So far, we didn't get the money together to do that, but we are still working on it. I want the monument for yeast in Vienna.
That would be very cool. Would it be, in your mind, a kind of giant sculpture of yeast? Like a huge yeast cell?
The point is, I'm a scientist, so my mind would be to find a good artist and talk to these people and let them decide how that should look.
Fair enough. I mean, if you do get this together, let us know, and perhaps you can bring them on the science art session next year. Paula, did you have an idea for Milan's official microorganism?
I was thinking, and for sure the Milano area is not as famous as Michael was mentioning for Vienna for wine. I mean, in Italy you have wines, but not really from Milano. But what we have that is quite typical of our area are some cheese. Actually, Gorgonzola, that is not distant from Roquefort, something like that. So I would say that maybe one of these
So it would also be a fungus.
Producing this blue vein. It's a fungus. In this case, not maybe in the east direction, so not unicellular, but again, it's a fungus. Yeah, I would say so, because, I would say so.
Mm. You see, I have a small idea for what the Netherlands could choose. I remember reading that Antoine van Leeuwenhoek observed Volvox cells at 1701 in the canals of Delft. So I think it would be very cool to pick Volvox as your national organism, because it is such a weird, strange one with like its babies growing inside its circular or spherical shape and then burst out to produce new ones. So I think Volvox for the Netherlands.
It's, by the way, the same thing with aging. Here you have now the concept that someone has to die to give birth to the new ones.
Yeah. And this is very much how it does its reproductive cycle in Volvox. Yeah. So earlier on, Michael, we did a top 10 of different fungi. I think we have time to do maybe a couple of runners-up for the final few minutes of the breakfast show. So one of the ones you put on the list is Pichia pastoris.
Yes.
I wondered if you could tell us about this. So I have an image of it, which I can get up on the screen for people. It is another lovely looking yeast.
Yes.
And
well,
here we go.
Its major attention comes from its biotechnological exploitations. The point is, a lot of the products which we are interested for example, pharmaceutical industries, are proteins. And what we want is that these proteins are produced well by the organism and then secreted, let's say, to the outside of the cell, because you don't want to break the cell to get your product, but you want it outside of the cell. And Pichia pastoris turned out to be extremely efficient in doing that. So we can teach it, let's say, or we can program it to produce pharmaceuticals of use very efficiently. And it's much better than many other yeasts for that purpose. And this is why it's used widely also industrially. It has been actually isolated from trees, from tree sap.
Or is it living inside a tree or outside
of the tree? Outside.
Okay.
So if there's a wound or something to the tree, for oak trees, then you can, or even in the bark, you can find it. And since particularly the lab of my colleague is totally dedicated to this yeast, so they also went out to nature to isolate their own strains, and it's possible. So you can find it if you choose the right trees. You can find Pichia strains.
Very cool. And now,
yeah.
Sorry,
Go
I was going
ahead.
to say, what pharmaceutical products does it produce for us? Anything that we'd heard of?
Oh, it can produce vaccines, but it can produce like antibodies. It can produce essentially everything. So.
Oh wow. Okay, very useful.
Yeah.
Any final?
By the way, it's really cool. It looks like moss.
Yeah, it does look cool, doesn't it? It's got this lovely texture, and you can kind of see the budding formation here, which has been highlighted in red.
Well, actually, that's the bud scar. That's what remains.
Oh, that's the scar. Ah.
Yeah, so you see, if you look at the upper cell, so on the left side, you have that one, exactly. That's the daughter cell. And on the other side of the cell, you have the scar, what remains when the daughter cell left the mother cell.
And does that stick around?
Yes,
or does
that
it
does stick around. And that already tells you that this, for example, the one in the middle, the big one, is the oldest one. So this has already budded at least 4 times. So I don't know what is on the back side of the cell. So 4 times. So this is older. And there you can see that it can't go on forever, because if it's full of scars, it cannot bud anymore.
Okay, so does it cover its surface in scars and then that's it? No more reproduction?
Yeah, I think it will stop before it's completely covered. But yes, essentially you can see it that way.
Oh, that's amazing. And I guess that's just the beginning of a budding formation. Then you've got a couple here fairly midway through.
Yes, yes.
And then this one's nearly done.
Yes, exactly.
In fact, over here you've got one that's basically just about to leave, isn't it?
Yes.
And then probably a scar will form there. Do we know much about the ring structure and how this scar is structured?
I guess we know, but I don't know. Do you know anything about it, Paula?
Well, yeah, there is it is known that in this moment where you need to have the division, there are some specific molecules that accumulate. And this accumulation is somehow guided by a concerted mechanism between, let's say, the cell wall and what is like the cytoskeleton inside. And so it is there is a recruiting of specific molecules, and then they can create this scar. So it's really another example where you see that you have to think about a very complex mechanism despite they are single cells. So it's also fascinating the fact that for budding yeast, you see that, as Michael was mentioning before, there is an old mother inside, and every generation the mother becomes a little bit bigger of the generation before. So you have heterogeneity within the population that is already also talking about the different traits that they can have. For example, for brewing, this is really important because they figured out, this happens also with Cerevisiae, and they figured out if in the repitching, they take the 2 young cells or the 2 old mother, this is not good. So they have to take something in the middle to preserve the capability of fermenting malt, but at the same time also the ability of a phenomenon that is like a clogging, and then that favors the collection of yeast at the end of the fermentation. So that is, it's something that is started like an art, and then we figured out later on that it was based on some molecular and cellular mechanisms.
Very nice. I think we've got time for one more fungus. And I chose this one, but again, Michael told me off for saying it's a bit boring. And that's Eremothecium gossypi. And I have some images of it here. And I just love it because, it has a nice role to play in research. And yeah, it's very easily tagged by GFP proteins. So, this video, here we go. Also, interestingly, it has like 2 names which are common. So I think it used to be Ashbia as its original name, but then they changed it quite recently to Eremothecium. And we'll find out why in a minute. But yeah, here we go. We have some lovely fluorescently tagged versions of this. So you can see